humans. And we've actually seen that from the research, land use change is one of the biggest drivers of these zoonotic diseases. They are not just affecting livestock, they are also, some of them are even affecting wildlife. So that's a big challenge. The land use change is coming because of things like deforestation, agriculture itself, demand for animal food, and of course, urbanization. So all these issues are contributing to land use change, and that's contributing to new diseases, new zoonotic diseases, and that's a big challenge. But with the diseases, we've also seen that there's quite a lot of challenge in terms of the um, antimicrobial resistance. So the microbes are developing resistance to different drugs. It's not just microbes. When we talk about livestock, quite a lot of diseases in livestock, more especially in Africa, and specifically in East and Central Africa, are transmitted by ticks. I will again come back to this point at a later stage. Ticks are treated quite a lot by acaricides, but we've also noticed that a lot of ticks or a lot of species of ticks are now developing resistance to the acaricides, which is another big challenge. Talking about all these challenges, climate change and heat stress and all that, there's also the problem of water stress. I'm sorry for that spelling mistake there. It's double burden. It's not a new word, double. It's double. Um, the scarcity of water in the region is really becoming a big problem. In this picture that I got from the University of Hohenheim, where I used to be professor and now I'm honorary professor, really depicts a very good scenario there. Good in terms of illustration. The same place where humans are collecting water is the same place that livestock are also having a drink. So we can already see that it's not just the bio genome that's interacting there, but also transmission of disease. So pathogens are part of that whole system. Another, excuse me, one other challenge that we haven't really been talking about quite a lot is the increase of carbon dioxide in the atmosphere. In the past 400 years, as in the graph that's depicted here from NASA, carbon dioxide hasn't increased a lot. But in the past 50 years, we've seen a very drastic increase in carbon dioxide. Why is that important as a challenge? It's important because we all know that plants get their carbohydrate from carbon dioxide in a process called photosynthesis. And because of that, we are ending up with increased carbohydrate production in some of the plants, more especially in C3 plants, such like wheat, rice, and potatoes, which we all depend on, and we also grow those crops in Central and Eastern Africa. These elevated levels of carbon dioxide are not just affecting the structure of the crops, they're increasing the yield, which may seem like a good thing. And there has been research that has shown that sugars and starches have increased by about 30 to 40%. Sounds like a good thing, but actually, this is not really good because this increase in carbon dioxide means that something has to give, which then affects human nutrition. What is it that has to give? It's the micronutrients that give. So concentration of nutrients like calcium, potassium, zinc, iron reduces because of this increase in carbon dioxide, in carbohydrate. But even the concentration of protein in those plants reduces, which then changes the whole ecosystem in these areas. The other challenge that's there is when we look at the planetary boundaries, we know that in some of these 9 planetary boundaries, we've really gone past what we can afford to lose. So the green part shows that we are all right, but as we go towards amber and red, it shows that we actually have a problem. In this work, which was published in 2023, it actually shows that one of the problems we're having, or areas where we've really shot up the planetary boundary, is the area of biosphere integrity. And when we look at that, one of it is actually genetic diversity. We're losing a lot of genetic biodiversity, which is a big challenge. These losses are not just global. When we zoom in into the region, into East and Central African region, we can actually see that from this study, even within local areas, we've lost some plants, we've lost some fauna and flora, which then really brings the problem home that it's not some other people's problem, it's our problem, and we need to do something about it. This table here just shows some of the specific examples of the losses that are happening in the region. So it's not just about plants, it's also about animals. It's not just about animals, we are also losing a lot of insects. The other thing that is a big challenge is the environmental stress itself. So environmental stress to livestock, to crops, such that we are now starting to look at resilience as one of the major issues that may bring in mitigate some of this environmental stress. Is it enough? We'll come back to that. An example of all these environmental stresses is also the occurrence of, or continuous occurrence of, some of the tropical diseases. An example I have here is East Coast fever, which most of us know, and the map of Africa shows the distribution of this disease, East Coast fever, which in Africa is caused by Threlia parva. The map at the bottom also shows that this problem is occurring elsewhere. So in Asia, it's occurring, but that's not Threlia parva. It's Threlia annulata that is causing a problem. The life cycle that depicted in that image is how this disease evolves. So that's the life cycle of East Coast fever, which involves ticks. And like I said earlier on, ticks are also developing resistance to the drugs that kill them. So you can actually see that this cycle is being perpetrated by even the resistances that we are getting in terms of acaricides. Another crisis that's coming up is heat stress, which is coming quite a lot due to climate change that I talked about earlier on. This graph here shows areas where livestock can live as in the comfort zone, but more and more that comfort zone is being broken, such that we are having a lot of heat stress, again affecting not only the livelihoods of those animals, but also the fertility, which means what type of genes are being transmitted. Can we utilize the power of genomics and genomic biodiversity to mitigate some of these problems. As we have already discussed, some of these physical mitigation strategies are not working. Is genetics and biodiversity genomics having a role to play in this? The answer will be in the next slides. So when we talk about genomics, what we are really looking at is to identify the specific SNPs that are within the different livestock. So the discovery work is really important in that aspect. It's not just about finding the SNPs, it's also about looking at the functional variants that sometimes may not actually be within the genes. They may be next to the gene, or they may be functioning together with the gene that we are talking about. So it's really important to discover and investigate these functional variants because they might be where the solution is. In terms of biodiversity genomics, really important because we are then starting to look at the variety of life. So the biodiversity itself with the advanced DNA sequencing. This is really important because we know that all these plants, all these animals, all these humans in these areas are not existing in a vacuum. They are interacting. So tracking the environmental changes is really important. Understanding how these organisms evolve and adapt to the environment with all those challenges that I talked about is really important. Some of the core tools that are used in this area, and I'm pretty sure in the practicals you will be dealing with some of these, issues like DNA barcoding, developing of a reference genome, very important, and also the environmental DNA, collecting and using environmental DNA from water. You remember I talked about water stress and all that, and that image that I talked about depicts how some of this DNA is exchanging at water holes, DNA from soil, from air, even from the plants and animals themselves. So really important to think in a holistic manner and making sure that biodiversity genomics becomes a big part of what we do in making sure we can conserve these species and also mitigate them from the challenges that I talked about. This image here, which I got from the paper from General et al. 2016, depicts this procedure of sample collection from different species and subspecies, and then utilizing that information. Now, earlier on, I talked about creating the reference genome. It is important as well to think about not only one species in this case, it's thinking about all the species. So the creation of a pangenome is really important as a reference point in this environment. Why is it important to look at this holistically? It is very important because that's what will give us the strategies for conservation. That's what will give us strategies for monitoring and tracking the ecosystem, so biomonitoring. That is also what will give us the strategies for health and safety, identify the diseases and what is causing them and what's carrying them, just like I indicated with insects. So earlier on, I talked about East Coast fever. An example we have from CTLGH is the discovery of the gene for tolerance to East Coast fever. Now, we know that East Africa has a lot of livestock. As we speak, Kenya is one of the highest producers of milk in Africa. This is being used. This is being done using different breeds. So in that image there, on the extreme left we have the Holstein Friesian, whilst on the extreme right we have the East African Shorthorn Zebu, with the Brahman and the Ankole in the middle. One of the things that we discovered in CTOGH in our labs in Erie, Nairobi, and University of Edinburgh is that as we move from Holstein Friesian to the East African Shorthorn Zebu, the tolerance changes, the level of tolerance changes. Holstein Friesian, high milk producer, but highly susceptible to East Coast fever. The East African Shorthorn Zebu, not a very high milk producer, but actually tolerant to East Coast fever. So even the breeds that are utilized currently in crossbreeding and pure breeding have different tolerance levels to this disease. We could not come up with that statement if we did not go into the lab and look at what's really happening in the genome of these animals. So using genomics and some of the biodiversity genomics, we actually found that yes, there is tolerance in different breeds, but even different individuals within families. So the picture there is showing one of the sires, a Boran sire, whose daughters were all tolerant to East Coast fever and survived and kept going, whilst other Borans, their daughters would perish in the same challenge. This is another slide. It looks busy, but the idea is not to go into detail of each one of the issues. It is about identifying what's really making these animals tolerant. So we discovered a haplotype that is responsible for this tolerance. Again, we could not do this without applying bioinformatics, without applying genomic tools, such that we can also look at the tolerance on this loci across the globe. So the graph on the left is showing the tolerance in different regions of the world. So we have the East Asian indicus, we have the South Asian indicus, we have the East Asian cross, the Taurine cattle from Asia as well, and of course the African zebu. All this kind of work could not be done by just looking at the phenotypes. It could be done by really getting into bioinformatics and utilizing those technologies. The other example I have is about the origin and domestication of chickens. One of the points that we came up with in CTOGH is that we cannot just look at adaptation and also tolerance at this point in time, but to start looking back to where these animals have come from. The graph that the map on the left is showing us where and how the routes of migration for the chickens. We know that chicken was domesticated in Southeast Asia, but we could only know that by studying the genome, because the time these animals were migrating, these maps never existed. So the genomics work is really revealing the origin and domestication of chickens, and then giving us lessons on why some chickens in Africa are adapted to heat stress, why some chickens in Africa are adapted to uh, low environment stress or high environment stress. It's because of where they are coming from. And we could only find that using genomics and using bioinformatics. Another example that I have from CTLGH is on the ecological niche modeling that has been done in chickens in Ethiopia. We know that Ethiopia, just like I indicated with the Rift Valley example, has some of the highest places and some of the lowest elevations as well. And using molecular biology and genomics, we found that um, there's a good reason why some of the chickens that are in the lowlands cannot be taken into the highlands. They'll be heat stressed. They'll be cold stressed rather. So there's quite a lot of work that's showing that the environmental adaptation of these animals is not just because of management and the rearing of those animals. It's actually the genomics and the genome that they are carrying. But are we really harnessing the power of artificial intelligence and even the power of bioinformatics to the maximum? At the current point, I would say no, we are not harnessing the power of artificial intelligence fully. Here are some of the barriers that we are having in Africa. And specifically, we can even link this to East and Central Africa. The high cost of equipment. The impact is that this limits the use of this technology. Can we find solutions? Leasing models, maybe, substays, um, and even working together, collaboration. So this is where I pay tribute to the BioGenome Project. By working together, we can actually reduce some of the costs that come with these equipment and technologies. Poor connectivity in quite a lot of countries in Africa, and Eastern Africa is not immune to this. There is quite a lot of poor connectivity, which reduces real-time monitoring and operations. Maybe we should start looking at offline first computing capabilities. Low digital literacy. Again, this workshop, even in the introduction, it was mentioned that part of this is for training, part of this is for early career scientists, and I dare say even some of the late career scientists need this. We own the day we will all say I've stopped learning or I've finished learning. That's the day that you are done. So really important that these workshops continue going on because that's where we can share new technologies, we can share new skills and utilization of the data that are there. Talking about data, one of the challenges is data governance. I'm really happy that even in the introduction, it was mentioned that there will be a talk and discussions on sharing, benefit sharing, and governance, because there is a gap there. The impact is there's usually low trust, and people don't want to even talk about things or even adopt them when there's no proper governance structure. There is need for clear ownership and sharing policies so that we can really harness the power of artificial intelligence and biodiversity. We need to harvest this power or harness this power because that's what will help us even understand the biology more. That's what will make us understand what we have on the ground. And then that will also help us master the language or the techniques or technologies that are used to harvest this power. It's only when we do that that we can then gain comfortable skills and use them comfortably when we are doing this work. We need to embrace AI and automation. Time is gone when AI was packaged as just one uh, chat box. It is a tool that can really help us do quite a lot of this combination of biology, statistics, and computing to unleash the power of the the biogenome that we have in the region. And remember, artificial intelligence is nothing than a collection of models. They need data to be trained on. They need data to operate perfectly. So generating those data is actually in our gift. In that sense, then, we can only unleash and harness the power of, of bioinformatics and AI when we get involved. And on that note, I would like to also pay tribute to some of the initiatives that are happening in Africa. The only thing that I would actually say is yes, let's continue these initiatives, but at the same time, universities should embrace and change some of the curricula to embrace these technologies. Short courses are good, short courses enhance capacities, but we also need to really have courses that are ongoing in different universities. Because as we can see, there are not really a lot of universities that are doing this. This is an example of the Arua, which is really a good example of connectivity between African universities and those even outside of Africa. But we need more of this. We need more of working together, generating this data, generating the capacities that we have. So, in order to harness and uh, utilize the full power of artificial intelligence and bioinformatics in Eastern Africa, we need to make sure that biodiversity and the biodiversity genomics is not just viewed as part of the solution. It should be looked at as not just as a subject, rather, but it should be viewed at as a part of the solution to the global challenges that we talked about earlier on. Biodiversity should also not just be looked at. We have all these flora and fauna. We should look at that as part of the local development agenda and local wealth in the region. Artificial intelligence is here to stay. Let's embrace it. Let's utilize it effectively. Let's build the guardrails that will make sure that the governance around it is strong, the capacities around it are sharp, and the capabilities that can utilize this are there that we can actually move forward. And in all this, I would actually say collaboration is key. And that's one reason I really thank the organizers and the conveners of this workshop for doing a very good job. On that note, I thank you very much for honoring me with your attention. Thank you.
Great. Thank you very much, Professor Chagunda, for setting a very good foundation to the workshop. And please allow me to take two questions from the audience and one from online. After that great presentation, we cannot go without hearing a bit more. So do we have any questions in the audience? Okay, we have one.
Thank you, Professor. And that was very exciting discussion. I was just, fascinated, I actually did not know how increased carbon dioxide can lead to reduced micronutrients, which we, even as physicians, we can now see in the clinic. We're seeing patients, coming with, deficits in selenium, zinc, XYZ, and if you look, they're eating healthy. So I don't know. These problems look like they are so big. How? Because they are like at the planetary level. I don't know. What is your take, what can we as researchers do to sort of mitigate? Are we getting into, should we sort of be, emphasizing on genetic engineering of plants, of animals, so that we can sort of mitigate these, challenges which are at another level that, thank you.
Thank you very much for that question. So there is a saying from which is not my I didn't create it, but I find it very important that with all these challenges, we should think global but act locally. So these challenges, yes, they look like they are at planetary level, but they are actually affecting us locally. So we should really start thinking local. And the example you gave in terms of deficiencies in micronutrients, the point that we should raise then is how do we deal with this problem using the science that we know at a local level. So even when we are talking about sequencing the material, the animals, the plants that are within the local environment, we shouldn't sequence just for the sake of sequencing them. We should be sequencing because we want to find a solution for a problem that is existing locally. That's how I would actually address that.
Thank you very much. Any other question in the audience or online? All right. Thank you so much for that great presentation. Africa is beautiful, has great diversity, and Africa is adapting, is evolving because of climate change and all these things that are happening. So we do need to apply and adopt these technologies to build solutions that are lasting for us. So thank you very much, Professor Chagunda. And we'll move on to the next session. So we are at the Mount Kenya University in Kenya, in Thika. And this is our second time at the Mount Kenya University, a very great host for us. And I'd like us to be officially welcome. We are taking over as if we are home. So I'd like us to be officially welcome to the Mount Kenya University. And please allow me to welcome Professor Jesse Gitaka, who is a physician scientist renowned for his impactful contributions at the intersection of molecular discovery and public health implementation. His academic journey includes advanced degrees in tropical medicine and medical science, where his doctoral research focused on critical aspects of genomic diversity in infectious diseases, and further postgraduate training honed his expertise in point-of-care diagnostics, solidifying his foundation in translational research. Currently, Professor Gitaka holds pivotal leadership roles, directing multiple strategic units that encompass the Center for Malaria Elimination, the Center for Research in Infectious Diseases, and the Directorate of Research and Development. In these capacities, he leads multidisciplinary teams spanning fundamental science, rigorous clinical trials, and innovative health system advancements. His research portfolio is extensive, featuring leadership and co-leadership in numerous competitively funded projects. These initiatives address critical global health challenges, including complex interactions of infectious diseases and the development of highly sensitive diagnostics for maternal infections. His work underpins significant research efforts across maternal newborn health, malaria eradication strategies, surveillance of AMR, and application of genomics-guided point-of-care technologies, with over 90 peer-reviewed publications and a strong presence in international conferences. Professor Gitaka consistently generates evidence that shapes policy, informs product development pipelines, and his recent contributions include pioneering work in placental malaria immunopathology and diagnostics driven by genome mining. Prof's profound impact has been recognized through his election as an affiliate of the prestigious Africa Scientific Academy and selection at the next Einstein Fellow, and membership of a prominent global professional organizations. He is deeply committed in fortifying African research ecosystems, cultivating the next generation of scientific leaders, and ensuring the cutting-edge scientific advancements and are translated into equitable and accessible health solutions for all. I'd like to add, he is leading one of the genomics facilities existing in Africa and one of the high throughput sequencing facilities in a university in Kenya. We are proud to be associated with you, Dr. Jesse, Professor Jesse, and please, tukaribishe.
First, thank you, Cecilia, for the very kind introduction. And so thank you very much. I really want to take this opportunity to, first of all, follow the protocols. Our Vice Chancellor, Professor Deogratius Jeganyi, the Deputy Vice Chancellor, Academic and Research Affairs, Dr. Marceline Kamande, Professor Bobit Badawi, Chair of the African BioGenome Project, Professor Anne Migai, Chair of the East and Central Africa Node and Deputy Vice Chancellor of the National Defense University of Kenya, Dr. Thank God Beneza, our keynote speaker, Professor Mizek Chagunda, who we just listened to, representatives of the National Research Fund, and our industry partners, colleagues joining us from across the continent in this hall and online. Good morning. Yeah, so on behalf of the Directorate of Research and Development, I welcome all of you to Mount Kenya University, and I'm super happy that actually this is our second hosting of the conference. We had a very successful one last year, and I know that's one of the reasons why Professor Anne Mugai early this year tapped on us again and said, let's do this again. So we are really excited to have it. And most importantly, I really want to, just reiterate. I know my seniors will be talking about this, but I just want to reiterate the commitment that Mount Kenya University has in advancing genomic sciences, biodiversity. And as you will see, from the discussions, when we look at whether you're looking at the humans or the animals or plants or microbes, the tools that we use are basically the same. Okay, the tools that we use, the sequencing platforms, the pipelines, the knowledge is not different. It's not because the language of life, the basic units of the DNA, the ACTG, is what you find in animals, in plants, in microbes, is the same. We all written with the same alphabet, So the tools are the same, and that's why we actually need to create these multidisciplinary engagements, interactions, where we come together, exchange ideas, because it's at the level of ideas meeting and, pollination of ideas that actually innovation exists. If you just sit somewhere in a corner alone, hoping to innovate, sad news for you. It's very difficult. You only innovate when you meet other people from other disciplines. A good example I like giving always is the use of ultrasound in the hospitals. This has actually revolutionized maternal care because we are able to scan, women during pregnancy, and we're able to see whether there are any challenges with the fetus, the presentation of the fetus. I mean, that's how the orientation is the head down. Is the fetus actually sitting in the mother's womb? Are they lying, transversely? Okay, all this actually affect or impact the decisions we as physicians do or make for the well-being of the mother and the baby. But doctors alone could not have discovered the ultrasound. We needed physicists, who have some knowledge on sonography, on sound hitting an object and coming back and telling us the nature of that object. And the physicists needed to have learned from nature, because we know animals like bats use sound waves to be able to see in the dark with a lot of precision. The animals, the bats, they are, as we all know, they're almost blind. And when they come out of the caves, they are giving some ultrasound, the very low wavelength sounds, hitting on branches, on trees, and knowing this is not the way to go. This is the other way to go. So you can see this one innovation that is actually helping our mothers. It has a lot of multidisciplinarity to it. So this is a key aspect of our understanding, and this is demonstrated in the composition of our teams. Sometimes when we're in a lab meeting, people are wondering, there's too much diversity here. We have mathematicians, we have bioinformaticians, we have food microbiologists, we have physicians, we have immunologists. Okay, we have geospatial guys and so on. And now we even have social scientists in the group, because the problems in the world are so multidisciplinary. Okay, we as people belonging to one discipline will not be able to solve them, because when you come to the real world is when you discover you may have a great innovation, but if you do not understand the social aspects to that innovation or to the problem, you'll be turned down, and it will be a waste of money and time. So I really want to take this opportunity to thank the local organizing committee, Professor Anne Muigai, who has been actually very key in spearheading this endeavor for many years now. Dr. Kuja Josiah, who was actually one of our own postdocs here before he went to Copenhagen and Harvard and then came back. Dr. Cecilia Arohio and Dr. Paul Kagame. And I really want to take the opportunity to thank Cecilia and FNS for when we were getting our NextSec Illumina sequencer, actually, they gave us a serious discount. I remember haggling with them. And when I told a friend of mine in Germany the cost that we got the equipment at, he could not close his mouth. He was telling me, you have the interesting dishonor of having the cheapest, the most affordable equipment in the whole of the world, because that price was very discounted. We really thank you, and I'm also very happy for the support you've given us. Whenever we are doing a sequencing run, we call on them, they come and support our team. So this is super cool, and let's keep doing it. So I now want to take this opportunity as I finish my remarks to actually challenge all of us. Make sure when during the breaks or during the discussions, the hands-on practicals, engage with people you actually do not know. Do not. Is our nature to always cramp with people we know, but as I told you, we need cross pollination of ideas. So that means if you are from MKU, look for somebody in JKUAT or in KU or some other place, and vice versa, and then ask them about their name, their research interest, and so on, and see how you can collaborate. So I want to now take this opportunity to welcome Professor Anne Muigai to make a few comments before I invite our Deputy Vice Chancellor, who will then invite our Vice Chancellor. And I really, I think I'll just make her biography very short by just saying, we've been cooperating over the last 3-4 years, and I don't fail to remind people that she's a Deputy Vice Chancellor of a military university. Okay? Yeah, and we were at a certain meeting, and I see military guys in full fatigues, and she's coming out and saluting to her, and I was very excited to see this progression, Prof. I've known you for many years working at JKUAT, and now it also shows that we can inject, this kind of skills in our military service. And we thank you for the kindness you've extended to us over the many years. Early this year, actually, was when we had the professor from, was June this year. It's just the other day, actually. Yeah. She invited a professor on our behalf on plant biology, and we had a great time, exchanged some brilliant ideas, and had dinner together. And we really thank you for that, for pointing people to us. This is one of things a good friend can do, is to actually expose you to other good people, isn't it? Yeah, so we really thank you for that. And I really want to take this opportunity to welcome you, to say a few comments before we proceed. Karibu sana Professor. Tumpigie makofi akija.
Thank you, Professor Jesse, for your kind words. Vice Chancellor, Mount Kenya University, Professor Deogracios Jegani, Deputy Vice Chancellor, Dr. Marceline Kamande, Professor Jesse, distinguished directors of Mount Kenya University, good morning. My name is Professor Anne Muigai. I'm a Deputy Vice Chancellor at the National Defense University, Kenya. But more importantly, and for this function, I'm the founding chairperson of the African BioGenome Project. And now we're distinct to have, very, very happy to have Professor Boabid, who will be addressing us as the chairperson, because I felt it's important that the chair also gives room to others to lead. And it's also given us an opportunity to now focus in-house in East and Central Africa and see what we can do ourselves. We are extremely delighted as African BioGenome Project to be hosted here for the second year running by Mount Kenya University. Last year's decision to come to Mount Kenya University was not based on anything else other than the fact that I felt it was important to put action to words. We had been telling institutions in Africa to invest in genomics and bioinformatics. We've been saying how important collaboration is, how we as scientists need to have an open door policy to utilize instruments and sequencers that had been placed in university. Then what does Mount Kenya University go and do? They go and buy an Illumina sequencer and they place it here. So I was like, I have no choice but to actually have the third regional workshop in Mount Kenya, first of all, in show of support that you have gone and done what we have been saying should be done, and also to be able to highlight the fact that this instrument is now in Mount Kenya. It's a facility that's available to us as Kenyans, and through collaboration and through partnerships, there should be no reason why there should be any barriers towards that. And thank you, Professor Jesse. But last year we had superstar treatment, and now we found it hard to go anywhere else. So early this year, I was telling Professor Jesse, give us a date, even if it's in December 24th, we will be able to come. This is the place that we want to have our conference. And thank you very much, Vice Chancellor, for facilitating that open door policy. Your staff have been able to extend a great sponsorship to this project, and only through that sponsorship have we been able to be here. I also want to remember that we have partners who have been able to provide resources because holding these workshops is expensive. One of the things we did as African BioGenome Project is to say that we will not charge for access. We want our early young career scientists, our graduate students, our young lecturers, our middle-aged lecturers, and even older professors to be able to have access to information, to be able to have access to new tools. And therefore we said we will not charge registration fees, we will not charge anything. We'll have an open door policy, but it costs to run a workshop like this. And through our sponsorship of partners like Mount Kenya University, who have provided conference room facilities, we have been able to run this. We also want to recognize the role that Illumina have placed. Not only do they have an instrument at Mount Kenya, but they continue to partner with African BioGenome Project to provide resources, to become exhibitors. And on top of that, they are also running a practical at Carlo, and their practical is called Accelerating Breeding Pipelines with Illumina-Powered Genotyping by Sequencing. And thank you very much. We also want to recognize Incaba Biotech, who are running 2 practicals with us. They are also exhibiting, and they are running a practical called Bioinformatics Without Tears. I mean, maybe you've all, Cecilia, did you cry when you are running your scripts? And yes, so Illumina is going to show us how to make bioinformatics easy and accessible. And they're also doing Sanger sequencing practical. We also want to recognize Project Mocha, Excella Biotechnologies, ICAWorks, and our Vixen technologies, who are providing the video conferencing facilities. Like I mentioned earlier, we are running 9 practicals, and these practicals are based throughout East and Central Africa. From Wednesday to Friday, we will give an opportunity to the scientists who have been selected to go to these practical sessions and have hands-on experience. So we have a practical that's being run by Pwani University all the way down in Kilifi by Dr. Sami and Dr. Anubak Khan, and this will be an introduction to wildlife genomics. We have a practical that is going to be run in the middle of Laikipia at the Impala Research Center. It's a collaboration between Impala and University of Copenhagen, and this will be hands-on wildlife conservation genomics workshop. We have a practical that is being run in Uganda by the National Animal Genetic Resources Center and Data Bank Entebbe. This is NAGRIC by Dr. Joel Ogwang, and they're going to do a genomic prediction of adaptation to future environmental conditions. This is timely because of the climate change that is being faced. We have other universities outside Kenya. University of Addis Ababa has partnered with Bioimaging Technology Institute in Ethiopia, and they're going to run a practical on Python-based bioinformatics and data science, a hands-on approach. Professor Dewan, Hawassa University with Professor Degu is going to be running a practical on DAT sequencing, DAT-generated GBS data filtering and analysis. And finally, but never the least, we are going to have a practical for the 3rd time running in University of Kinshasa, who have partnered with Laboratory of Molecular Biology to run a machine learning with R for genomics and bioinformatics. This is an excellent show for institutions, and we're very, very grateful. We had 1,000 applicants to this workshop. Out of 1,000 applicants, 700 applicants wanted to attend this workshop in person. We extended the invitations, and we reached 100. As you can see, this room is full, 100 in person. I hope that shows you the demand that we have. It would be nice to have 1,000 people, but it's logistically, it may not be very easy to do it. We may be able to do it if we develop satellite, and that's why we have an online presence. Right now, online, we have about 300 participants who have logged in. And for the practicals themselves, we had about 500 applicants. So the demand is there. We are grateful for the institutions who have partnered with us, and we ask those institutions who can to please forward your request to us, and we would be very, very happy to partner with you. So thank you very much to Mount Kenya University. Thank you to all of the participants who have come, and we wish you a fantastic 5 days of the regional workshop. Professor Jesse.
Yeah, another round of applause to Professor. Thank you, Prof, for the very insightful and kind words to Mount Kenya University. Now, it's my pleasure to welcome our DVC, Dr. Marceline Kamande, who is herself an economist to make her comments and then proceed to welcome our Vice Chancellor. Karibu, Dr.
Asante sana, Professor Jesse, for that. And a good morning to all of us, both of us who are here and those who are online. But maybe I can, we have done it for the sake of those who are online. It's really my joy to be here and to be part of this workshop. I know I always say that when I come to Professor Jesse's workshop, he does the science, I do the administrative. But I think today I quite resonate with what you're talking about, because when you touch on issues of climate change, I think climate change is really cross-cutting. It's something that we are seeing everywhere. And I was quite excited as I listened to the first presenter talking on things that we can really relate and problems that we can really relate to in the communities where we are. And all what I can say is that this is very encouraging that we are having this discussion at Mount Kenya University. Allow me to just start by saluting my Vice Chancellor, Professor Jaganye. Thank you for always supporting us to be here and making these people comfortable so that they can come back. Professor Baduli, but I hope my pronunciation is good, the chair of African Genome Project. Professor Muigai, thank you always for being here, and thank you for being such a partner. Dr. Ebenezer and Professor Chagunda, I think I heard him speak this morning. Distinguished speakers, partners, and sponsors, colleagues, students, and friends in the room and online. I want to take this opportunity to really welcome you to Mount Kenya University on behalf of the academic division. I am privileged as the Deputy Vice Chancellor, Academic and Research Affairs, to be, or rather to oversee this team. And it's always good as opposed to oversee a winning team. So they always make me very happy. And I say they always run faster than me. Sometimes I have to catch up with what they are doing, but indeed, as a university, we are really happy for what Professor Getaka's team is doing in trying to do the networks and having all of you in this place. And again, also, thank you for choosing to be in Mount Kenya University. I have heard the campaign that Professor Muigai has made. Being a deputy vice chancellor in her own institution, she has chosen to bring it to Mount Kenya University, and we cannot be grateful enough. I'm one person who believes that we have, as universities, now it's about collaboration. You can never exist alone. And so when I see all of you here, and in fact, as I sat there, I saw the Africa, and I was like, how much Africa we are? And I will not ask that question, Professor Muigai, because I think you have been able to profile how big this conference is. It's actually bigger than the room that we have here. And for that, we are really grateful for choosing that Mount Kenya hosts this. And for us who are here, let us take opportunity to gain knowledge, because knowledge is something that you can never buy. Actually, if these guys were to sell what they are giving you, I don't think many of us would afford, but thank you for the scientists who have gone ahead of us. And all what they are doing is that they are reproducing themselves into us as young scientists, faculty, students, and the best that we can do is really to take advantage of what is here. I've listened to Professor Getaka give a brief of what the workshop is, and I cannot emphasize more that on the importance of this workshop to us as academics, to us as researchers, to us as upcoming researchers. Because the best that we can do with science is when science meets the needs of the community. And for that, I want to reiterate what he has said, that let's take advantage of this opportunity that we've been given and really gain that extra skill that you need to take it both to your institutions and to be able to network with whoever else is in this room. Again, networking is very important, because when we network, then we are able to benefit with our next door neighbor on things that we would not have done alone. So the Africa BioGenome Project aims to sequence more than 100,000 African species within 10 years. And this is where I reach and say that this is the description of the project, courtesy of Professor Jesse. And that's a genomics target, but it is also an education target. Every one of those genomes needs a person on this continent who can design the sampling, learn the instrument, build the pipeline, and defend the results in the literature. And behind every such person is a supervisor, a curriculum, a degree program. And as I read that statement, I reflected on what individual things that we do in our own respective disciplines. And unless we are able to learn from those who have gone ahead of us and those who are mentoring us, to really be able to get into the literature and see how do we utilize this literature in what we are doing, then we may not have achieved as scientists or as researchers. So the AfricaBP's Open Institute has already trained more than 800 African scientists in hands-on genomics and bioinformatics. And I think, as Professor has said, we had this one earlier this year. And these are opportunities that we are given, and I think they have described where even those people who are online. At first, I was worried, Professor Muigai, when I saw your numbers online, and I am like, these people will not get the practicals. But and I can see you have really done good to be able to ensure that even those who are not able to come anyway, we may not be able to host the 700 here. But when you use other institutions the way you have done, I think to me that is really commendable, and I really want to appreciate the organizers for this workshop for being so insightful to involve other people. And this is really remarkable for a young consortium like this one, and it tells us that the training pipeline, not sequencing capacity, is the binding constraint on African genomics. And I think that is really true. It is about us being able to train and to diversify this training among other people is what is going to really make us what we want to be. And this is where universities come in, and it asks 3 things from us. And that is both ourselves and those universities that have been mentioned. So the first one is that we teach we teach the frontier and not the history. I think that is quite powerful. This year's theme puts artificial intelligence at the center of the biodiversity genomics, because that is where the field is. If our curricula still teach bioinformatics that was practiced 10 years ago, our graduates are life already behind. And what this project is doing, it is putting you ahead of everybody else who is doing what they are doing, so that you can become competitive in the fields that you are working on. The practicals from Wednesday to Friday in Python, in R, in machine learning, in genomic predictions are precisely the content our programs must absorb. And I could really relate on this. I think he's just said I'm an economist and precisely an econometrician, and we do a lot of these informatics. And when we do this now from the science point of view, then we bring the socio-economic and science meet together. Then we touch on our economy. I ask our academic staff and postgraduate students attending this week that to treat this practical as curricular dissonance, and to bring back what should change in what we teach. It is not only for us, it is for the students whom we are teaching, it is for the colleagues whom we are linking up. If you are a postgraduate student, as you sit on that computer, as you sit in that lab doing what you need to do, can you be able to really have a spillover effect to the people who are near you, so that it is not only about us, but it is about the science. I think we cannot afford to be mean with what we know. We cannot afford to be mean with the knowledge that we know, because having knowledge and keeping it to yourself then will not help the science that we are trying to help. Second, we want to request that we widen the doors. Women remain underrepresented in computational biology, and early career scientists at smaller institutions rarely get instrument time. Tomorrow's session on gender, indigenous knowledge, and inclusive implementation of the coming Montreal framework describes who gets to do the science and in whose terms. The hybrid model of this workshop, which keeps a student in Goma and in Hawassa or Kisumu in the room, is part of the answer. Thank you for using technology to diversify what we want to diversify. Because if we do not affect that person in Kisumu, if we do not impact them, then we will still remain a small team of ourselves, and it may not be very helpful. We want that multiplicative effect in what we do. That we treat research governance as taught content, access and benefit sharing, digital sequence information, biobanking ethics, and Kenya's own framework under the Narcotics and Data Protection Act are things every genomic graduate should understand before collecting a first sample. I am glad that the first African Congress on Digital Sequence Information is happening on a university campus. And I hope that some of what is said tomorrow finds its way into our course outlines. I am in charge of curriculum development at this university. And I really want to say that one of the things, even as we prepare, as universities now we are thinking CBE, we'll be having a team that has been trained to do things by touching, practicals. And I think that is what, as universities, we are having a conversation on. How can we be able to advance what they have been trained at the lower levels, at the university level? And this could not have come at a better time. As I see you people trying to bring artificial intelligence into the science that you do, and bringing the data science into the science that you do, that just brings in now, it breaks the barriers in disciplines and allows us all to come together for a common cause. And to me, that is quite commendable. So Mount Kenya University's commitment is concrete. Under the Vice Chancellor's leadership, we have invested in the Directorate of Research and Development, the Center for Malaria Elimination. I believe you will be able to have an opportunity to see that facility, which has trained quite a number of people. And I am glad that even this year, we have been able to provide that opportunity. So the Center for Research for Infectious Disease, which is housed at that lab, and in international partnerships that bring instruments, data, and course supervision to our staff and students. The academic division will ensure that those who complete this week's practical receive formal recognition, and that the connections made here are followed with joint supervision, exchange, and co-authorship. One of the things that my Vice Chancellor is very, very bold to tell us and, push us about is to ensure that whatever you publish gets to important journals. And that is why that co-authorship, that supervision, being supervised by people who have gone before you, then it really makes you get into the top of the top. The Scopus journals, the Web of Science, those who are our students, they know that narrative, that for you to publish, then we must go to the top journals, because it does not help you and it does not help me as a university to produce publications that do not go anywhere. So that's something that we are giving as a commitment. And maybe just to add something, recently as a university, we have actually also acquired another very important something that is going to help the researchers, the certification for the good financial grants practice certification. And that tells you that even the grants that you do in collaboration with Mount Kenya University, they will be able to be guaranteed of a higher level of success despite the competition because of the GFGP certification. And I think we are among the very few universities in Kenya who have got that. And that just shows you as a commitment, as a university, that we are committed to make your work easier as researchers. You do your work, and we are here to ensure that you have the right environment, you have the right support, administrative support, for you to be able to do what you need to do as scientists. And we are happy to partner with you in whatever you do. So therefore, if you are a partner institution interested in an MKU student or have a student who would benefit from time in our laboratories. My office is open to that conversation. Please feel free to come to us. And the good thing, even with what you're doing, is that as much as we have the science lab, it is supported by a very robust IT architecture. And even with the other sciences. So we have the genome lab, but we have quite a number of labs here, and we'd be happy to really be part of the journey of all the students that will be able to be co-supervised at this project. And I believe we are open for that conversation. So to our students, this is your week. The people speaking over the next 2 days are writing the papers. You will cite. And so for you to benefit from that, then ask them questions, interact with them. Don't go and just sit with the person whom you spend time with. Spend your time in the practicals and be attentive. And I think with that, you'll be able to gain what you need to get from this. Let this week not just be 5 weeks out of work. 5 weeks maybe to get a few per diem to bring you out of your office. That is not our goal. Our goal is for us to be able to use this work very properly and to gain the most that we can do. And with those many remarks, allow me to take this opportunity to invite our Vice Chancellor to address us. I always say he's the scientist. Me, I'm not a scientist, so he resonates better with what you people you do. Karibu sana, Vice Chancellor.
Good morning, everybody. Let me do the obvious, the salutations. Professor Baobid Badawi, Chair of the African BioGenome Project. Professor Anne Mugai, Chair of the East and Central African Node and Deputy Vice Chancellor of the National Defense University, Kenya. Maybe we should salute you. Because I hear that whenever you walk there, it's just salute all the way. But I must say thank you very much for bringing this conference to MKU. We much appreciate it. Dr. Thank God Ebenezer, co-chair and founding architect of AfricaBP. Professor Chagunda, our keynote speaker who has just spoken. The National Research Fund of Kenya, head of partner institution from Kenya, Uganda, Ethiopia, Democratic Republic of Congo, Rwanda, Tanzania, South Africa, Morocco, India, and Denmark, industry partners and the sponsors, my colleagues, Dr. Kamande and Professor Jesse Gitaka, members of the local organizing committee, ladies and gentlemen in this hall and joining us across the continent online. Good morning and welcome to MKU. It is genuinely a pleasure to host the African BioGenome Project again, and particular pleasure to host it in a year when the workshop is joined by the first African Congress on Digital Sequence Information, Infrastructure Development, and Policy. Let me offer 3 reflections, and then I will do what a Vice Chancellor is here to do and declare the week open. My first reflection is about sovereignty. When Dr. Benaza and his colleagues founded this project in 2019, they set a target that most people thought was odious. 100,000 African plants, animal, and microbial genomes in 10 years. But the more important part of the ambition was where the work would be done. Not sampled in Africa and sequenced elsewhere. Not sequenced in Africa and analyzed elsewhere. The full chain from field to freezer to flow cells to publication on this continent in African institution by African scientists. That's what this is all about. time has come where we don't just have to give our samples to global north and then we become recipient. We are still doing it in terms of our agricultural products. We take our coffee beans to Europe, they grind it, they send it back, costing us 1,000 times more. So this was an ambitious project. Things to be done in Africa for Africans. That is a statement about scientific sovereignty. And everything about this week, the practical in our laboratories, the facilitators from Kilifi and Kinshasa and Entebbe, the industry partners setting up instruments down the corridor, is that principle made concrete. I want to acknowledge the National Research Fund for backing this workshop. As you have heard, that it is not cheap to hold this kind of workshop. You need somebody else to give the support, and we got the support. So we definitely are very grateful. Public research funding that stays on the continent is the foundation of which the rest is built. My second reflection is about the theme. Artificial intelligence is the frontier of biodiversity genomics. I'm a physical chemist by training, and I have watched computational transform my own discipline over 30 years. What is happening now in genomics is faster and more consequential. Models trained on billions of bases can now predict gene function, annotate assemblies, and flag variety of concern in hours rather than months. Professor Chagunda has spoken to what this means for livestock genetics and adaptation. And we listened to that particular presentation, which was a fantastic presentation. And a lot of things which people take for granted were really touched on. So, artificial intelligence is here to stay. We may all make noise about our student using AI to answer exam questions. And we simply say that, come up. And I've told my DVC, come up with questions that AI cannot answer. But otherwise, our students must be AI savvy as they go out there. And as an institution, we are in partnership with the Chinese to make sure that all our graduates are AI experts. So, DVC, you have a job, and other DVCs, you have a job. I don't know if the army people are using AI, but good luck. Dr. Gagutko will speak to pathogen surveillance. Dr. Fankola will share hard-won experience from real assembly project. But I want to add a caution that a university leader must voice. Artificial intelligence learns from data, and the data is learned from determined what it sees and what it misses. If African genome, African pathogen, African livestock, and African crop are absent from the training set, then the tool the world built will be blind to Africa. When you look at the data, the publications from Africa, and you compare it with the rest of the world, we are still very, very far behind. And that's the reason why I keep on telling all the academics at MKU that you publish in predatory journals, you're digging your own grave because you'll never be promoted. Because it's of little value. Publish in Scopus journals, publish in the Web of Science journal, publish in those kind of things so that the African knowledge can be referenced by other researchers. You look at the publications, South Africa is leading, and then you go all the way to the top of Africa where you find those publications. So we need to be able to add on our own data, because the artificial intelligence will only be able to work for us if we have our own data available. The way to change that is not to complain about it, it is to generate the data, curate it well, publish it, and build the compute and the skill to train and evaluate models here. Africa must be a builder of these tools, not only as a consumer of them. Time for us just to consume other people's knowledge has come and gone. And you have saw it in terms of the COVID. You'll be locked in your own region. If you don't have the data, you don't have the knowledge. A lot of people expected Africa. I think there was an expectation that Africa will be wiped out by COVID. But we seem to have a different type of gene. We survived. And maybe a lot of people want to understand how we survived when the rest of the world was facing a lot of problems. That means investment in a high-performance computing on our campuses, in data stewardship, and in the mathematical and computational training of our students. It is a job for universities, and we, as a university, accept that. My third reflection is about the Congress that begins tomorrow. And it is the reason I am glad this conversation is happening on a university campus. Digital sequence information sits at the intersection of science, law, and justice. We missed out economics. She's an economist. The Convention on Biological Diversity, the Nagoya Protocol, and now the coming Montreal Global Biodiversity Framework are attempting to answer a question that Africa has been asking for a very, very long time. When the genetic resources of a country generates value, who benefits? Who benefits? When the genetic resources of a country generates value, who benefits? When Kenya decide that it's going to give its medical data to the Americans, who benefits? Who benefits? Are we the one benefiting? Are there drug companies in America who are benefiting? What's the motive? Why are you interested in our data? I leave politics to politicians. For decades, the honest answer has been usually not the communities and the countries where the resources come from. The Congress will hear from NACOSTIC, from the Wildlife Research and Training Institute, from negotiators for the DRC and Rwanda, from the Ethiopian Biodiversity Institute, from ILRID, and from our biobanking colleagues in India and Kenya. I hope it produces more community. I hope it produces a shared East and Central African position, practical templates for benefit sharing agreement that scientists can actually use, and a clear account of what infrastructure in biobank and in data platforms the region must build so that our sequence data and govern here under our laws and our benefit. I would like to add that no region has more at stake in getting this right than ours. East and Central Africa holds the Congo Basin, the Rift Valley lakes, the Ethiopian highlands, where coffee and teff were domesticated, and the Indian Ocean reef of our own coast, and the dry lands where pastoral livestock have adapted to heat and drought over millennia. Those genomes are a scientific inheritance, but they are also an economic one. Crops that will feed us under a changing climate, livestock resilience to diseases, and natural products with medicinal value. Whether that inheritance is developed here and whether the returns come home depends on the policy and the infrastructure choices this Congress is convened to shape. So we wait and hear what this Congress will be able to come up with. Are you going to give our inheritance away, or are we going to keep it within the same continent? Let me turn to Mount Kenya University's commitment. In 2025, this university was appointed the United Nations Academic Impact Hub for Sustainable Development Goal 9. We have been a holder of Goal 10, but now we are a holder of Goal 9, which is industry, innovation, and infrastructure. We take that as an obligation, not as a decoration. It's an obligation, not a decoration, to just simply put it on the United Nations website that MKU is a hub, is a holder of the hub, but it's an obligation. It's an obligation to the rest of the continent. It's an obligation to the rest of the world. Under our Directorate of Research and Development, our Center for Malaria Elimination, and our Center for Research in Infectious Diseases, we are already running genomic surveillance, molecular diagnostic, and clinical research with partners in Europe, Asia, and across Africa. Through the Vice Chancellor's Research and Innovation Grant, we fund our own staff to start the projects that external funders later on scale them up. To AfricaBP and to the partners in this room, I want to say 4 things. 1, our laboratories, our computing, and our campus are available to this consortium as a training and analysis node for the region, not only during this week, but between workshops. So we are opening up to anybody who is interested. Take advantage of it. 2, we will support our academic staff and postgraduate students to take up AfricaBP projects, and we will recognize that work in appointment and promotions. 3, we will work with NACOSTIC and with our partners on the governance side so that Kenya's framework for research licensing, data protection, and benefit sharing are ones that enable good science rather than obstruct it. 4, we will help to bring the next generation in, because those are the future. That's the future. It's an issue of continuity. I stand here as a Vice Chancellor of Mount Kenya University, but I know, and I always tell my students, I'll be very proud when I retire to see one of them standing here as the Vice Chancellor of Mount Kenya University. So, Professor Gitaka today is leading the malaria elimination group. In his group, there should be somebody, a pipeline of young scientists who will be able to continue with that work. So, from our side, we will be able to make sure that these young people are part of the next generation of the scientists. I wish the politician could do that. They don't. Look around this room and you will see a lot of young people, which is a sign that the future is bright. Many of the facilitators for this week were themselves participants at the earlier workshops. That is how a scientific community reproduces itself. And Mount Kenya University intends to be one of the places where it happens. To our guests, some of whom have traveled a very long way, thank you for coming to Thika. To our organizing committee, our staff and our students, thank you for the work that we'll never see, but without which nothing here will have functioned. And now, on behalf of Mount Kenya University, and with great pleasure, I declare the 4th AfricaBP East and Central Africa Open Institute for Genomic and Bioinformatics Workshop and the first African Congress on Digital Sequence Information, Infrastructure Development and Policy in East and Central Africa officially open. I wish you a productive, argumentative. Academics must be argumentative. If you're not argumentative, then you are in the wrong place. As an academic, you must always try and defend your space. And you defend your results. So, I wish you a productive and argumentative and a useful week. Thank you very much and welcome to MKU.
Another round of applause for our VC. Very inspiring, stimulating, energizing opening remarks. And we are very happy and very grateful for the kindness that MKU has continued to give us as a community. And we are super excited for this and very grateful, Vice Chancellor and the DVC. So we'll take this opportunity to move down for the photo session for posterity. Yeah. So actually, we'll do it at the stairs, just at the entrance, and then after that we'll have tea break, and then we'll proceed from there. Thank you very much. Let's move for the photo session. Yeah. For the people online, I don't know. 30, we'll be back in 30 minutes, right?
Yes, for the people online, bear with us. You can also grab a cup of tea. We'll be back after 30 minutes.
Thank you very much.
said. So I want to welcome you all to our second session after the tea break. So we have 3 speakers, and I've just been informed that Professor Badu is online. So allow me to read his bio. Professor Badu is a prominent Moroccan scientist and a professor of genetics and genomics at Mohammed V University. So this university is based in Morocco. So he's highly recognized for his leadership in advancing African genomics, currently serving as the chairperson of the African BioGenome Project. He is a his research blends molecular biology with computational science, focusing primarily on genetic diversity, phenotype-genotype interaction. So his core areas of expertise include landscape genomics. So I just mentioned a few. He studies how environmental changes, climate gradients, and geographic factors shape the genomic pathway and adaptation of native animal breeds. So this include Moroccan sheep and goats. So he's also into animal production genetics, where he analyzes the genetic determinism behind milk composition, proteins and lipids that are present in milk, coat color, demographic shifts, and thermal stress response. Then finally, he specializes in bioinformatics and biostatistics, developing data analysis tools to map massive data sets for quantitative genetics and computational biology. So ladies and gents, let's put our hearts together and welcome Professor Badu.
Thank you very much. Just to check if you can hear me. Thank you.
Yes, we can hear you.
Yes, we can hear you. Go ahead.
Okay, I will try to share my presentation. Could you see my presentation, please?
So it is visible. You can put it on presentation mode.
It is already. You see it small?
Yeah, so Boabit, it's in presentation mode, speaker mode. Can you put it on presentation mode rather? So we can only see the slide, but not in the presentation mode.
Let me check.
Or you want to swap it, like sometimes you can flip it.
Okay.
Yeah, click that. Let me see. Setup. Click setup. No, click setup in that at the bottom where you have the play button. Yeah, click setup.
Yes,
Yeah.
yes.
Yeah,
I mean,
exactly.
yeah.
Exactly.
Yeah. It's okay?
Yes, carry on, please.
Okay, great. So good morning, everyone. First, thank you very much for giving me the opportunity to present AfricaBP, specifically AfricaBP Open Institute. Before starting, I would like to thank all the people from the MKE University, organizers, professors, students, and all the people that organized this very important event. Specific thanks go to Professor Anne Mogay for the great effort and the commitment for this event and for AfricaBP generally. So, as we know, AfricaBP is home to extraordinary biological diversity, but much of that biodiversity remains genomically undocumented. In this sense, AfricaBP was created to change this situation through African-led science, capacity development, and responsible data government. What I will try to do in these few minutes, I will try to explain our mandate and how our model have developed mainly for Open Institute, the progress already achieved. And just to give a hint why biodiversity genomics can become a foundation for health, food security, scientific sovereignty, and prosperity generally across Africa. So, of course, at the center of AfricaBP is the sequencing. There is a clear continental mandate to sequence approximately 1,500 endemic African species. Of course, as it was explained in the article published by our colleagues here from AfricaBP, this is not sequencing for its own sake, because we need to use this genomic information to help us to understand what species exist, how the population are changing, and which biological resource may be vulnerable to climate change generally, or specifically to habitat loss, disease, and other pressures. This is the first thing. The second thing that we would like to get from the sequencing is to create a knowledge, mainly practical knowledge, for ecologists, conservationists, and for plant and animal breeders that works to develop a more productive and resilient food systems. So biodiversity is seen in the context of AfricaBP as a strategic resource, but it cannot be protected or used responsibly if its biological baseline remains incomplete. And AfricaBP has as a goal to build that baseline and ensure that the knowledge, skill, infrastructures remains available and connected in the African countries. So just to give you a hint, in few words, how AfricaBP began. It began as a grassroots governance. I mean, for about 2013, African scientists start to recognize the need of a greater genomic independence and shared continental approach. That community of animal and plant breeders and scientists aligned to the formal launch of AfricaBP in 2021 with the objective of sequencing African species to build capacity and to translate this information that we get from the sequencing into evidence for the African policy. Between 22 and 25, the initiative evolved through the Open Institute model. And this is what we think it marked the transition from a centralized concept toward decentralized one. Because Open Institute, as I will explain later, is a hub and spoke structure operating across African 5 geographical regions. So this evolution is in our eyes is very important, because from Open Institute, AfricaBP has grown from a scientific vision into an operational continental network. Of course, project of this scale requires governance, and solid governance that is both coordinated and locally responsive. AfricaBP in this sense used decentralized structure. As you can see here, we have the steering committee that provides the overall direction and coordination. And we have the advisory board that contribute to scientific, the ethical and the economic oversight of the AfricaBP. The regional coordinator on the other side, they translate the continental strategy into local actions because they support the implementation and capacity building, taking in consideration the specificity of each region, because being there, they are able to know what is happening there, and they are in a good position to know how to apply the direction of the steering committee to the local community. So we believe that this structure balances shared standards with regional ownership to create a democratization, but one that is well governed. It also allows decision to reflect local priorities and available structures and the needs for national scientific communities. So we think that this strategy is needed for the well-functioning of AfricaBP. We arrived to our main area, the Open Institute for Genomics and Bioinformatics, which is the capacity building engine of AfricaBP. Its first mission, of course, is knowledge transfer. We would like that Open Institute bridge the gap in biodiversity genomics, help institutions develop the infrastructure needed to participate fully in capacity building. And the second pillar of these institutions is the standardization of the curriculum. We want that the training be equal over all the African regions. So this training that combines genomics, bioinformatics, molecular biology, and biobanking, among other, it uses intensive hybrid format that connect theory with practical applications. And the third pillar of Open Institute is the grassroots exchange. Instead of concentrating our expertise in small number of institutions, we would like to involve a maximum number of institutions. So to give a maximum opportunity to a maximum number of Africans over all the continent. The distributed hub and spoke models of Open Institute, it's a core specificity of this institute. So AfricaBP works through localized regional hubs and public-private partnerships. Because we want that the training will be available for all the Africans by reducing the need for scientists to cross borders repeatedly for training or laboratory access. This is especially important because we know that travel costs and visa restrictions are real barriers to the movement of scientists. We have some hub centers like Muhammad V University in North Africa, the Port Harcourt in Nigeria for West Africa, the Mount Kenya University that is holding this workshop now, the South African Medical Research Council. All these hubs are the anchors, but these hubs, they are not the only sites where the training is happening. Many other institutions that are involved according to the trainings that is offered in each workshop. The growth in the trained human capital shows quickly how this model is in success. In 2022, AfricaBP has trained 21 scientists, and this number got increased till 2025. Over the same period, more than 45 localized workshops were deployed across all 5 geographical regions in only 36 months. This number, we think that this number will represent more than attendance. They indicate a growing community with practical skills in sequencing, bioinformatics, molecular biology, among others. We think that the knowledge that one could acquire during this workshop could pass to other colleagues and students. So the human capital is therefore the essential infrastructure behind every genome generated and every future scientific conservation, health, or economic application. And we believe in the core of AfricaBP that training a human being is the first step for building the future, the genomic future of Africa in the context of AfricaBP or in the context of other initiatives. The first Open Institute doesn't start from scratch, from zero. It lies on 6 landmarks in the context of AfricaBP. So, of course, the first landmark is the sequencing, is the biological blueprint that was described by Ebenezer and colleagues in 2022. It established the foundational roadmap for sequencing African biodiversity and aligned that ambition with the goals of the Convention on Biological Diversity. So the second landmark is bridging the genomic capacity gap. The work cited here by Sharaf and colleagues document the rapid deployment of hybrid regional workshops between 22 and 25. But the core lesson is that scientific training can be standardized without being disconnected from local realities. As I mentioned before, AfricaBP try to share procedures, operational guidelines to maintain the quality of the training over all the African regions. So we can make the training accessible and relevant to all the Africans, whatever they are taking these trainings. The third landmark connects the genomic to the African bioeconomy. We would like to show to the African people that sequencing is not a luxurious work. It has a real economic impact. Here, we are given an example of the simulation of 1,000 American Genome Project. And the study reports a benefit cost ratio of 3.29 and an estimated net present value of $20 million over 10 years. The core message here is that local genomic investments create downstream value. It can reduce dependence on foreign services. It can retain expenditure within the country, and it can develop skill deployment and strengthen the institutional base for future research and innovation. So by investing in sequencing, we are investing in the construction of a whole continent. The fourth landmark is it places biodiversity genomics within the One Health framework. We know that human health, animal health, and ecosystem health are deeply interconnected, and changes in climate, habitats, and human activity can alter the condition in which disease emerge or spread. And this is specifically important for African countries, where the borders between the habitats, between the human habitat and the other habitat is not clear in many cases. And the genomic surveys would provide information that can help a researcher understand this relationship and respond more efficiently to zoonotic diseases and threats. And we cite here the work by Kuja and colleagues that argues clearly that biodiversity genomics can become part of critical surveillance infrastructure for One Health. The fifth landmark examined the wider microeconomic effect of genomic investment. We tried to make simulation for many African countries using GTAP model to explore how sequencing induced productivity shock can influence the national economy. The reported cost ratio was different according to the countries, the economical structure of each country. Here specifically we cite the benefit cost ratio 2020, 22.14, to show that effect of sequencing extend beyond the laboratory or the biotechnological sector. It impacts cross utilities, construction, electronic equipment, heavy manufacturing, transport, and communication. This illustrates the aforementioned idea of sectoral fingerprint. The fingerprint of sequencing, of investment in sequencing, is very wide, and it could reach the far sectors of all the economy. The last landmark is it connects the scientific activity to the global biodiversity policy through what we named the theory of change that begins with genomic data generation, but it does not end there. Data must be governed through an ethical, legal, and social framework, including access and benefit sharing, which will be the theme of the second day, and in which many experts will say their words. So these protections are essential for maintaining sovereignty over African biological resources and respecting traditional knowledge. I will not say more here because we have all the day that will speak about that. Of course, AfricaBP Open Institute could not reach all these goals if it doesn't take two things in consideration. The first one is, of course, the EI aspect, because we know that the EI now is needed for any scientific endeavor. So through its data science and EI subcommittees, there is an initiative of developing artificial intelligence and machine learning capacity for biodiversity research. We already some specialized training in machine learning for biologists has been done in Mohammed V University and others, but we believe that EI can help researcher analyze large and complex ecological and genomic dataset more efficiently to identify the patterns and to accelerate the discovery. And we believe that including this aspect in Open Institute vision is mandatory for any development in the future. The last and second point, Africa BP's Open Institute is taken in consideration is the African DSC databank, which addresses a fundamental question. Where will African genomic data be stored, governed, and transformed into value? I believe that this aspect will be discussed widely the next day. I will just say a word about it, because we know that having African DSC will overcome many problems concerning the limit local access and commercialization, the weakened control over intellectual properties, and increase the risk that value is created as well. So this DAC in the context of Open Institute and AfricaBP is very important for the success of AfricaBP. And I reach the last word I would like to say is what I named AfricaBP engine, which brings together 5 mutually reinforcing elements: vision, policy, capacity, prosperity, and discovery. The opportunity is to safeguard Africa's biodiversity while building scientific leadership, responsible governments, and economic value on the continent, because we believe that economic value is of great importance to our continent. That's all. Thank you very much for your attention.
So thank you very much, Professor, for that insightful presentation. I will take the first question. If you look at the core mandate of your institute is actually development of a curriculum Probably at what level of training is this curriculum? Is it undergraduate? Is it master's? Is it PhD level? Because our mandate as MKU is to offer training, and from our previous speakers, the short course, even though they are good, they might not be adequate enough to equip the trainees. So we want to go for the long-term training. So maybe you can share your thoughts and your experience on curriculum development. Yeah.
Yeah, thank you very much for the question. I think it's important one. So for the curriculum, we don't exclude anyone, but we give priorities. We prioritize PhD students first because we know that they are in big need of the training. But we can include master students too when they need that. So this is our first strategy. But generally speaking, we don't exclude anyone who is interested in learning. For your second question, of course, this training are short-term trainings for 2 days or for 3 days generally. Of course, they are not enough to build skills and capacities in bioinformatics and biostatistics and all what we need for studying genomic biodiversity. First, we believe that starting for training, because we always students one day, and we remember our first training that opened our eyes on something very interesting and something that really shaped our career forever. Sometimes when we do training just for 2 or 3 days, we start to know exactly what we want to do in our life, what is very interesting for us, and what moves us a lot. And after that, we have our eyes open on many, many other things. This is the first point. So we believe that the philosophy of having training at least for 2 or 3 days, it's a big enhancer to open the eyes of its students on what exists in there, what they want to do in their scientific career. And it opened the doors to many, many other things. Second things, we believe that in the future, of course, exchange between African countries for trainings. And we started already something with South Africa and with South Africa and other countries, for example, to exchange students for a long-term training. For example, some students, for example, from Kenya, that comes to Morocco with a professor that is specialized in one thematic and vice versa. So for some months, and that could be very, very helpful. So you're right, we are starting with short-term trainings, but we are, in our philosophy, we have trainings for long terms too.
Okay, thank you. So any feedback from the audience? Maybe I could take 2, 3 questions, burning questions. Okay. Yeah, because the first session we were told we need to be very argumentative. Yeah. So this was coming from the Vice Chancellor himself. So
yeah, maybe you can ask online if there are any questions online.
Do you have anyone online joining us with a question?
I think we have Aragao. Aragao, do you want to speak? We have some question here. Someone is asking, Boabit, can you share your thoughts on how many capacities have been built in this direction for genomic and bioinformatics?
Yeah. I presented this, I think, slide here. If we take a cumulative number, we are about 1,300, the number of people that was trained, but in many areas of biodiversity genomics, including bioinformatics, programming, molecular biology, biobanking, many, many aspects. So if we take all that together, we are now is about 1,300 across the African continent.
Yeah. So just to also support what Professor Boabit has said, this workshop started, the actual workshop started in 2022, and we've organized since 2022. We've organized over 110 workshops, both the regional workshops and the practicals, and we've trained over 1,300 people cumulatively. and we've reached over in terms of awareness. When I say training, I mean actual practical, going to a practical site, having a lab, and then working with the lab, in small groups of 10 and 15. So that is a huge thing to do on the continent. Then we've reached over 15,000 people who have attended the day 1, day 2. So, and when I say training, I mean people who have been trained day 3 to 5. And of course, with thousands of people we've reached online particularly. So that's just to give you a context. We also published, we have a preprint that described this in, I think the preprint was authored by Josiah Kuja, who is on site now, and I think Boabit also mentioned that. So we describe all of this context in that paper, and of course things have changed since then because we've trained an additional 100 people who are not part of this 1,300 people that Boabit just mentioned.
Yeah.
We have another someone that wants to ask question, Mateta.
First, I would like to thank Professor for a nice presentation. But I have a question regarding the training, the Africa Open Institute training, where you mentioned that he trained, it is primarily based for PhD students, but they also take master students. So I was wondering, do they also consider undergraduate students? Because, for example, I am an undergraduate student doing bioinformatics. So I think I would benefit from this training. So I was actually wondering if they also take undergraduate students.
I think, Boabi, do you want to go for it, or should I? Boabi, you're mute if you're talking.
Can you hear me now?
Yes, please.
Yes, you can hear.
Okay, sorry. Yes, of course, as I mentioned before, we have some priorities, to start with PhD students, master, and we, for example, I have experience when I did the training in Python programming. I had places, or I took PhD students, and after I took master students, and I got more 10 seats. I took undergraduate students, and I choose from those that are interested in bioinformatics and all. So to answer your question short, yes, it's possible. We don't close the door, but just according to the place we have some priorities. So we start with PhD students, master students, and we can go to undergraduate students, because just we believe that those people are in need to use bioinformatics in their work, but we can go to undergraduate students. I don't know if, thank God, you would like to add something.
So we have a selection criteria, a general selection criteria, which we've published in Nature Genetics back in 2023. So we don't the criteria does not differentiate between an undergraduate or a postgraduate or a professor. It just says, do you have a project you're working on, or will you have a project in the near future? So because what we don't want to do is to train people and then they come, they see the technique, and then they don't give back to their institution. So we discourage that, and the general criteria discourage that. So oftentimes, and I think in support of what Professor Boabit has said, is you might find it's actually people doing research, or PhD students, or early career researchers, so you have to have a project, or you have to have the intention of having a project, so that when you are trained, because all of this training costs money. And we work with our sponsors to kind of bring the training. So, and what we want to do is to see that feeds back into their institutions, into their project, and that creates the right ecosystem on the continent. So I will also share the selection criteria. It's published, it's in the public domain that we use, and that is the baseline. And of course, the practical sites then decide on additional specific selection criteria.
Okay, I think that marks the end of the online question. So let's applaud Professor for that insightful presentation.
Thank you.
Yeah. So, ladies and gentlemen, allow me to introduce our second speaker of the day. This is Dr. George Michuki. Dr. George Michuki is a medical microbiologist and genomics expert in Kenya. He's the founder and CEO of the Africa Genome Center and Consultancy Company, the TAGCC, specializing in genomics, bioinformatics, and biological software development. As a founder and director of NextGen Molecular Lab, he spearheads molecular diagnostics, precision medicine services, and health software solutions. Daktari's impactful work includes advising the Kenya National Multi-Agency Command Center on COVID-19 and the Ministry of Health from 2020 to 2022. His career highlights encompass establishing Africa's first genomics platform at ILRI. This was in the year 2009, where he utilized the next-gen sequencing technologies for pathogen surveillance and discovery. In addition, he is a member of the Biotechnology Research Program Committee at KEMRI and a member of the Precision Medicine Task Force at the Ministry of Health, Kenya, and an accomplished academician. He has delivered lectures and training across various scientific disciplines at local and international levels. Daktari's visionary leadership and entrepreneurial spirit have not only revolutionized the genomics landscape, but also driven the creation of cutting-edge facilities and protocols, making him a proven innovator whose contribution promises significant advancement in genomic research and technology. So we are happy to have you here today, Daktari, and please, karibu.
Thank you so much. Okay. So I was asked to talk about AI, and I'll go direct to it. And what I'll be sharing today is not the norm academic stories or shares. I'll be trying to challenge you and also confirm, because what AfricaBP is doing, I believe this is what we need for Africa. And what I'll be sharing is just to enforce and to show the value and value proposition in terms of what is AfricaBP achieving based on what we already have existing. And so project sovereignty index for Kenya at that percentage. And then I know we had someone talk of AI as artificial insemination and artificial intelligence. So today I'll also be using DSI interchangeably with data sovereignty index as well as digital sequence index. Now, this is something that I just pulled data. Went into NCBI, pulled out all the data submitted between the year 2020 and 2026. And this data is filtering off COVID sequences, just everything else other than COVID. And something interesting, a lot of project authorships include Africans, and some not even as first author, but the work has been done in Africa. Then where's the data? From all these studies done, the sequences, where are they? Who owns the sequences? And that is where I think AfricaBP is trying to build on. How do we make, as users of our data, how do we retain sovereignty of our data? And that data that I pulled out, I've made it accessible. You can actually scan that QR code and you'll be able to stream it directly from my laptop just to play around with it. And there's a reason I've allowed you to play around with it, because I'll not share everything about it. But as you play around with it, you'll be able to see a lot more. And the data is pulled out from two databases, the nucleotide database and the SRA database. Why nucleotide? You know very well that most of us, when you sequence one sample and you have 2 reads, you've probably sponsored yourself. Where do you submit that? To NCBI nucleotide database. But when you have good funding, where do you submit the massive project data? Mostly the SRA database. Now, data is the foundation of artificial intelligence. All this data that has been uploaded is being used globally for AI model trainings. So the question is, when you submit that data, it's your data, it's being used for AI model training. How do you get the benefits? You probably just did a paper, but you shared your entire data. You released it, and it means the ownership changed from you to the recipient or to the holder of the data. So what do we do, and how can we change that? From the data, you will notice that a total of 5 million reads have been submitted in those years, excluding COVID sequences. And for your information, I would challenge you to also repeat and probably see if you'll get the same information. But I'll give you a caveat. For you to pull out data from NCBI database, they only allow you to do 3 requests per second. And if you have an account, you can only do 10 requests per second. And so this data took me 3 weeks to pull, not because of low compute power, but because of the limited requests that you are allowed to do. Now, based on this, you can see how much of the data that is from Kenya alone was analyzed on local platform, and you'll see what I mean by that. It's only about 15%. So 85% gone. Is it collaborative, or what is the structure of arrangement in terms of that sharing? And these are the policies we are saying as AfricaBP is actually trying to pass. How do we have policies and bring policies that allow gain or getting advantage of the data by Africans who are actually generating the data? And this is one of the section. You can go to the last tab on that if you've managed to connect, and you'll see this information. There's a reason I highlighted that section. What do you see? Gut metagenomics. Sequenced on what platform? MySeq. Where is it being sequenced? University of California. Which are the countries giving that data? Kenya inclusive, and many other African countries. How many MySeqs do we have in Africa? Kenya alone, how many MySeqs do we have in Kenya? Professor Gitaka's lab has an exec. It's more superior than the MySeq. So why do we have all these samples, simple microbial data being analyzed in California, which means a sample was shipped from these countries into another lab to be sequenced using a platform that we have locally? And so this is where I bring in the challenge. Why aren't we using our local platforms, yet we have the platforms? Next slide, same thing. I've highlighted something being done in Rwanda. Hot springs metagenomics being done somewhere in Asia. Why is hot springs metagenomics being done on MySeq, being done on the Nanopore, being shipped to be done elsewhere? When we have our students, we have our PhD students, master's PhD students who can actually do this work locally using the platforms we have locally. And this is the question. You do it out there, then eventually you submit the data, or the data is actually not submitted by you. Actually, what you see there, the submitter is those universities. However, the source of the sample is the countries you're seeing. So this is a challenge I'm trying to pose, and I'm sure this is what AfricaBP is trying to drive, because all these trainings is to train you so that you can be able to do this using the platforms that we have locally. And this should be our motivation. And I'll share what this means. Why is this very important? It means we are not driving our own agenda. And if we need to drive our own agenda, then we need to start having this ownership. I know most people will give me this answer. It is expensive. What comes before? Is it the egg or the hen? Why is it expensive? It is expensive, one, because if Illumina is to sell only 10 kits to Africa, why would they price it cheap? So do you expect Illumina to lower the prices when you are only ordering 5? Twist the coin. If all these samples were sequenced in Africa, that's 5 million samples. If you told Illumina today, as AfricaBP, all the granted projects, all the funded projects will be sequenced in Africa in platforms that are in Africa, it means you're telling Illumina we have 5 million samples that need to be sequenced in Africa. What is the price you're going to give us for the sequencing kits? Will they give you a discount? So what is making the costs of reagents expensive? Is it Illumina or is it our mode of operations? It's basically our mode of operations. So we need to change our mode of operations and use the local capacity that we have in Africa if we are going to scale down the costs. And this is the way to go. So what next? How do we start solving some of these problems? And this is where I know entities like National Commission for Science and Technology, NACOSTI, they're working on policies. And some of these policies are actually to help. And some of the things, and these are proposals, I'm not saying this is what exists, these are proposals. To me, can we have a way of monitoring what is being sequenced out? Because this is where we are talking of digital sequence information. How do we monitor what is being sequenced out? And so it means for everything that has to be shipped out for sequencing, can it be known? Why is it going? Is it because Professor Gitaka's lab cannot sequence? Or is it because you are just sending for the sake of it? Two, there is a UN convention that was actually passed. And this convention says that even data that is submitted in public databases, because again, you still have to publish if you are to graduate for your master's PhD, and some of the papers will require you to submit the data in public databases. But I checked, and some of the data submitted from here, they are not tagged specifically to indicate the country. Some I could pick them because maybe someone said sample collected from Murang'a, so I know that is from Kenya. However, someone may not know. You've heard of people saying, I'm going to Lagos in Kenya. I'm sure you've heard that. Yeah, so it means what we are losing is when this UN convention was passed, it says that for any multinational or any industry that uses digital sequence data from the public on the databases, the revenue from anything they develop needs to be shared back to where the sample source came from. So if you're not putting your geotag, or you have sent your sample and the person submitting puts the geotag from their country, then you're making Kenya, Nigeria, Tanzania, or wherever lose on that revenue coming back. And so there's need to ensure that you actually do put your country geotag when you're uploading. And the way to go is, can we have a platform, I don't know, by a regulator or by a university, the AI challenge, develop a system that would actually help trace, track, and see how much is going that is not geotagged, so that you pick out what needs to be geotagged. And how I would look at it in terms of how do we develop these, I would look at policies being developed, which would actually help us come up with strategies, or actually I would call these a strategy. And for me, it means we need to be a bit more stringent in terms of what needs to go out. Only needs to go out if it is necessary. But am I saying you hold up on data? No. Have the data with the right information, properly geotagged, and where possible, if you can analyze data locally, that is the best option. And so it means we also need to start building, because I think right now we have so many sequencers around. It is a high time now we think, how do we build on servers, how do we build on compute to be able to analyze this kind of data, which will help us now come up with structures like federated learning. What is federated learning? It means you have your data held by you, but accessible by other researchers and other teams. And because it is federated, it means whatever I use your data for, you get to know what I'm using it for. And so if there is anything commercial coming out of that data, you benefit from that output. And so these are some of the things we're supposed to be thinking in terms of how do we become sovereign, in terms of how does our data help us. And you will notice that I believe we have the scientists, and this is, even when you look at the project sovereignty index, quite high. It means we have the scientists, and we have the sequencing platforms, which we are not using. And so the intellectual leadership exists, but what we need is, can we build on structures of holding our data if we want to develop proper AI? If we want to revolutionize AI and use AI, because AI is data. Without data, you have no AI. If you use wrong data, wrong AI model. So the AI model is as accurate as the data is accurate. The AI model will work only if you have the true data. There's my biome company, which if you check, if you can Google mybiome.com, something of the sort. If you check what they are selling, they're actually selling, you basically sequence your microbiome from stool sample, and based on your sequence data, they are able to tell you your dietary requirements and what you need to take. What are they using? Probably the many gut microbiome data that has been uploaded is what they have used to train their AI models. And now they are selling a product that we are buying it at even a higher cost here in Africa. Yet it's probably something that has been built and trained using your own data. And so that is where the issue of sovereignty comes in. Just to highlight, I know I've said we have a lot of these platforms. There is an additional one, which is a Novasec X+. We've noticed that a lot of people are doing microbiomes and small genomes. And the question is, are we studying hosts' microbiome interactions? You probably not, a plant or an organism is not behaving the way it should be because it is not able to associate with the normal microbiome. Why? And so we also need to look at that. And the platform that we have is a Novasec X+, which is open. For us, we are commercial, 100% commercial, which means you have a project, you want to use the platform, we are open to allow you to use it. We have a compute server with GPUs and running AI models from most of the data that we are generating. And so you cannot say there's no capacity. And to me, this does not mean it stops here. This is just to say it is possible. And so it means as a private company, if we are able to get a server with GPUs, how much more of education institutions, how much more research institutions. And this is where we are trying to build, we want to also build something we're calling Viwingu, which is a federated data that we want to host in partnership with BioLinks Africa. And the reason we are doing this is from a pilot study that we did. And from that pilot, I can share something about it. We did a pilot, which was just a call where we asked innovators with ideas to apply. That was about a year ago. And we got applicants who were mostly from academia. And one of the successful applicants was a PhD project where they had done their PhD on prostate cancer. They did some cell cultures and stopped at that and published. So this application, we reviewed and we onboarded them in. And what we onboarded them now is to use genomics and repeat what they did just to see, can we identify unique biomarkers that can be used for detection of prostate cancer. At the same time, we did some pharmacogenomics testings using the same DNA. Now, this is what I want to share. We got very interesting results on pharmacogenomics data. And this is where we found that the population we had from Meru, 80% of them had mutations on the MR11 gene, which confers them resistance to apalutamide. 80%. I'm sure those who are in the medical field, you know how much apalutamide is prescribed the moment you are diagnosed with especially castration prostate cancer. So if at germline, not somatic, at germline, someone is resistant and we have this, is the drug being given to the patient? Is it helping them? No. Now, from the same data, we got mutations that can be helpful in identifying prostate cancer, positive from negative. And from that, we've developed a diagnostic test that just uses blood to detect if you have prostate cancer or not. And that is currently now undergoing patent registration. Why am I sharing this? We would have just uploaded that data to NCBI and publish the great paper that says 80% of the population is resistant to apalutamide. Nice, good paper. But then from the same data, an innovation. Now, why am I sharing this? We can't train all of you 1,000 to work in Gitaka's lab or in my lab or in another. How many labs do we have? But we can train you to be innovators and use the existing capacity to develop. This is a PhD student who had abandoned the project at that, but now we are developing a product that will end up being a company. They will have their own company manufacturing and selling diagnostic kits for prostate cancer. How many diagnostic kits can be developed from this forum? How many tools can be developed from this forum? So it's an issue about mindset switch. Let's not always think, I want to do this because I want to be a professor. Yes, we need professors, but at the same time, we can't all be professors. I'm a student of Professor Anwigai, and Cecilia was my student, so you can see the difference. I did not take the university line, but I still work with Professor Anwigai. But at the same time, I took a different channel through her mentorship, yet it's still producing and generating. So there are many things you can do with genomics, and now with AI, sometimes all you need is your computer and access to this data. So can we do much more? The other thing is, I've already shared about the prostate. I've shared we did some training last year at TAGCC where we were training on library prep and sequencing. And intentionally, I said, we have to do something real. And the training involved sequencing a bee genome. We did the library, sequenced, analyzed, and had the genome in 3 days. This was meant to be a proof that you can get real data, real results in Kenya with our capacity. I don't think we sent anything out other than just completing, and were able to submit, and the genome was accepted at NCBI. To me, that was a proof of a point that yes, locally done and acceptable internationally. And that was done in 3 days. So these are possibilities that exist. It's a matter of us just doing. And for now, with the setup of the new NovaSec, I know nobody knows this, but we are going to be unveiling this week the first whole human genomes done in Kenya, analyzed in Kenya by Kenyans, and in Kenya and hosted in Kenya. So this is something that is coming up this week, done from our lab using the NovaSec X Plus. And so what am I saying? If we can do that and we can generate that, what prevents you, if there's a plant you are working on and you want to see how it's interacting with the microbiome you've been working on, what prevents you from doing it? All you need to do is talk to us. I know most of you are asking, what does it cost? What does it take? You can scan that, and that will help you even cost your project. We've done it. It's using an AI model. So you can design your project based on that, know what coverage you need and what it will cost you. That is just a simple AI engine helping you design your projects. And thank you.
So can I get an extra mic? Yeah, thank you so much, Daktari, for that presentation. I think we can open up his presentation to a few questions from the audience.
Thank you very much for that insightful presentation. My question goes to, based on your experience with the genomics, what parts of the workflow benefits more from the AI model? Do you add like a analytical layer in the pipeline, or maybe you can comment about that? Thank you.
So I'll give a practical example. So when we did the prostate work, for instance, from each patient, we got about 100,000 plus unique mutations. Now, 100,000 plus from each patient. If you are to now analyze, to pick, because you're not going to do a diagnostic tool using the 100,000, you need to pick the most optimal, the most effective. Now, traditionally, it means we would have had to divide those 100,000 sites to probably reasonable size of maybe 100, 100, 100, and 100, and then write a grant to look for PhD students who will look at each set. So imagine how many PhD students, how many years, and how many failures before you get the most accurate and the top 10. We used an AI model, which we trained, ran the samples, and within 3 days, actually over a weekend, by Monday, we had the top 10 hits. And now those top 10 is what now we went for validation. So that is where AI reduces what you can do for 3, 4, 5 years or 10 years to a few days. I hope that answers.
Very well, thank you.
Any other question? Maybe at the back.
Okay. Thank you, Dr. Michoke, for that wonderful presentation. I have a question. I mean, as a global digital sequence information policy is being developed, maybe how do we ensure as African countries don't end up with restriction of accessing the data, yet securing fair benefits while others still are commercializing it? Thank you.
I think I would first applaud AfricaBP. I think that is one of the things, because this conversation is happening because of AfricaBP. And this is where it starts. Starts as a conversation, starts with people understanding, starts with the trainings, people seeing the value, and then now you sit in forums. Because, for instance, like what was passed at the UN Convention, it is because of scientists who fronted those points and policy suggestions, who are scientists from Africa. And so this is where it begins. So I think right now what we need to do is probably maybe change to a higher gear, but we've already started the process and we need to get more of us on board to actually achieve this. And this is a professor who I like to quote. He said, if you want to understand the biodiversity of the Amazon forest, you don't carry the Amazon forest to the north. You bring the tools and the abilities to the Amazon forest. The African biodiversity, it's the origin of humanity. Why are we carrying it out? Let people come study their origin, where the origin is. Thank you.
Thank you. And maybe a follow-up on that. How do we ensure that we move from the lab, where we do the sequences and generate data, and how will the implementers, or maybe an application in the health field, how will the health workers maybe be accessing that data to be able to know or to identify the risk of pandemic outbreaks? Thank you.
Thank you so much. I'll tell you things that are already happening. I sit in the precision medicine task force, and it's one of the reasons we are trying to see how do we make the data federated. Federated here means, yeah, you own the data, but it's accessible. So that's one. Two, I also sit in a committee at the National Commission for Science and Technology, NACOSTI. We are also trying to come up with a biotechnology policy and a genomics policy that will see how this can also be federated. And thirdly, on our own side, as I mentioned, we have a partnership with BioLinks Africa. We did a first call. We'll probably be doing another one soon. We're trying to fundraise where we open for innovators. So as an innovator, you have an idea, you have this thing that you believe can be commercialized. We want to open up to see, can you be incubated to access the data and to use the platform that we have to actually move your idea to a product to market. Another institution is Kenya National Innovation Agency. If you check Kenya, they always do calls for innovators. How many of genomics experts are applying for those calls? Very few. So we need to see more of you applying to those. So they are starting to have opportunities. And the more outcomes and outputs we have from these small opportunities, they will create more opportunities. Thank you.
Dr. George, we have one more question online, and this is to all the people who are present and also those online. Kindly tell us which institution you're coming from and just give your name. So Masi Mulandi, you're next. In 1-2 minutes, just give your question and tell us which institution. And happy to see you for the second year running.
Thank you very much. My name is Mercy Mueni. I'm currently a PhD student in Japan, and I'm working on climate change and with the rumen microbiome. And it's very good presentation. I'm recently working with the Kenyan native plants and trying to see how it affects the rumen. So if I'm working with Kenyan plants in my next objective, I'll be sure to outsource your services. Now, my question is, what do you think about, there is NCBI in Japan, they have a database for sequences, and I heard Prof. Badaul talk about sequencing database for Africa. So do you think that if we own our data, what do you think about Africa's own sequencing database, especially in regards to AI?
Thank you. I know Japan has their own database. I know Europe has their own database, and the US government has their own database. But when you look at it, they are interoperable. You're able to see the Japan sequences on the NCBI. You're able to see the European database data on the NCBI. So it's a high time we also as Africa have our own, which means it doesn't mean we block it or we lock it from access. It just means it is well annotated and categorically stated as African origin, but still interoperable and communicating with the other. That's what I'm talking about, federated data. Because if you have data and you lock it, then what? You may not be able to exploit that data wholly alone. You need other minds, other thought leaders to actually also mine the data. So the whole idea is about having the data, having proper tags known that this data belongs to who. And whoever owns the data benefits from any output coming from the data. Thank you.
And perhaps let me just chime in and say that as Africans, it's time for us to have this conversation about when are we going to have our own database, and where is it going to be located, and which institution is going to host it, and what are the accessibility questions around the data sovereignty, because we have to talk about it. And this is one of the questions that we want to have tomorrow, so that we can hear, not from Professor Mwegai's point of view or from Professor Jesse's, what are the Africans saying? That's a conversation that is happening right now. And what would we like for ourselves? And what are our experiences? The good experiences, the efficient experiences, and the mistakes, because I think we have made some mistakes that we have to acknowledge. And then from there, we will be able to now further and say, let us start our own. Because again, there are resources that will need to be committed, and we will now need to come together as nations, as institutions, to be able to say how we want to move forward. Did I hear thank God online? If not, thank you very much. I think this has been a very, very enlightening conversation. Dr. George, we're always proud to see what you're doing, and you're very humble in saying, oh, we just did this and we did this. I know the investment that you have made as an individual has been huge, and I call upon all of us to make sure that you're busy, the instrument is busy, so that we can make sure that the investment actually pays back. So thank you very much, and we're proud to see what you're doing.
Thank you.
I hand over to the MC.
Okay, thank you so much for your presentation. It's really been inspiring, and I just have one question. From the beginning of the day, the talks have been like African data is missing, especially on the global genomic platforms. And if you look at databases like the Cancer Genome Atlas, is one huge genome database that is used to look for mutations that causes different types of cancers. And this is the data that we rely on even for diagnosis and for prescription of drugs. And if you go to that database, you realize that the African genomes are actually missing. It's mostly the Caucasians. So do you think there's a lot we are missing when it comes to cancer treatment and diagnosis, as well as prescription? And also considering the fact that Africans are quite diverse. Just an expert opinion.
Thank you for that question. A very good question. And I like switching to politics on that. Why? We are moving at the age where drugs are being designed based on genomes. Right now, the new drugs that are coming out for cancer treatment are actually genome targeting, and they've been designed based on Caucasian genomes. So you know what that means? They are coming with a label, not suitable for children under the age of 5 and not suitable for the people of color. I usually tell our politicians, so you think you will mess things and fly to the US and get treatment? You will find that superior drug is not suitable for the people of color. So it is a high time even politicians realize with the less than 1% human genome representation in the database for Cancer Atlas, they need to start chipping in and making regulations that are favorable to ensure we start generating this data into that database. Thank you.
Okay, I think we can applaud Daktari for the way he handled it. So, ladies and gents, allow me to introduce our third speaker. That is Dr. Elekia Anguko. He's a Kenyan molecular virologist, a computational biologist, and the genomics lead and PI at the Department of Genomics, Bioinformatics, and Computational Biology within the KEMRI CGHR, which is located in Kisumu. Daktari is also, again, PI on various protocols. He leads advanced genomic sequencing, pathogen surveillance, and computational host pathogen studies in Kisumu. He's also the scientific director of the GENA initiative. Probably he'll tell us what the GENA initiative in full means. Spearheading regional capacity building in computational biology and advanced data tools across Africa. So he collaborates with a number of partners. This includes the US CDC and the Bill and Melinda Gates Foundation on AI-driven metagenomics and epidemic preparedness. So thank you so much, Daktari, and we welcome you to your presentation. Thank you. So is it available?
Elika, can you hear us?
It's online, yeah. Hello, Daktari, can you hear us? Maybe you can put your presentation on presentation mode.
Elika, if you're speaking, you're muted. We cannot hear you.
Yeah, so we can see your slides. But we can't hear you.
So try put your slides into presentation mode, please.
Yeah,
That's good
perfect,
now.
yeah.
No, it's a
You
sound.
can go on, yeah.
We can't hear you. If you're talking, we can't hear you. So until we hear you, that's when we can start. But the presentation is good.
Still on mute, Doc.
We've asked you to unmute yourself, so kindly unmute. Yes, I think now it's okay.
Apologies. I hope you can hear me now.
Yes, yes. Go on, please. Thank you.
Okay. Thank you for the opportunity, and sincere apologies. I couldn't make it to Nairobi because of other competing interests, but I'm happy to be here and speak about the work that we're doing at Cambridge CGHR and beyond Cambridge, what we're doing at the GENA initiative. So pretty exciting to be in the space of virology and artificial intelligence. Been quite an exciting journey and working with partners from across the world just to see Africa becomes a frontier in AI and genomics. I chose a different mode of presentation just to look into the scope of genomics, where we are at the world and as a country. So first, we get to see that the market for genomics is evolving globally, and good numbers there in terms of income, in terms of growth by 2033. But the biggest challenge at this point still remains data volume, where we are generating genomic sequencing pretty fast, and the costs are going down day by day, and it's becoming more widespread. And at the same time, the volumes is growing faster than our ability to manually interpret this data. Unfortunately, we have challenges with data complexity, where genomes are isolated, where genomes are not isolated sequences. So their meaning depends on time, geography, host, and other factors. And in most cases, you realize that the time between collecting a specimen and sequencing the sample and putting out the data out here is quite long. So by the time we really throw this data into databases, it's either too late or at that point it's not really that meaningful. The other global challenge that we're having is this data is distributed across different repositories, laboratories, countries, and surveillance systems. And then the data is also in different formats. Metadata quality issues, standards, and levels of completeness make integration quite difficult. And then the final biggest global challenge is how can we translate this data into action? The challenge is always, can we analyze these sequences? Can we turn genomic signals into knowledge that supports scientific and biodiversity decisions? When you look into the LMIC problem, we've made so much progress as a continent, and this is with regard to pathogen surveillance. So much work happening in Morocco. Thanks to the Institute Pasteur, they are always my point of reference when it comes to how they have advanced surveillance and integration. So much work happening in South Africa, DRC, Kenya, and Uganda. But if you look into all these databases, Kenya, Morocco, South Africa, Uganda, and Nigeria, and even Ghana, they are doing the most when it comes to generating data. Now, the key challenges in Africa, at this point, we have unequal access to sequencing and lab capacity. Yeah, we've done pretty well post-pandemic, but we are still seeing these challenges persist. There's also so much fragmented data, whereby this disconnected genomic, clinical, AP, and biodiversity data. One of the big challenges at this point is also limited compute and storage. And it's a big issue even in established research institutions. And then the genomic interpretation gap, whereby we're generating lots of data, we're training a lot of people on wet lab. But when it comes to analysis and interpretation, that's where the problem begins. And then at this point, as much as everyone is speaking about AI, we have limited AI capacity. Africa lags behind when it comes to developing models, tools, and even AI expertise. And I'll speak about this as my presentation continues. So artificial intelligence and foundational models or foundation models. So we've seen the sequence revolutions, data tsunami from the African Genome Project, the pandemic surge in NGS activities across Africa and across the world. But then immediately after this, we had the rise of artificial intelligence, whereby we're seeing machine learning, deep learning, and complex workflows that have potential to advance genomic works globally. At this point, everyone is aware of general purpose AI, for instance, Claude, OpenAI models. They have done a pretty good job when it comes to democratizing genomics by making sequencing analysis, annotation, variant interpretation, and phylogenetic workflows more accessible. Anyone can query GPT, anyone can query Cloud, anyone with basic knowledge or basic training in command line can still now make sense out of data without struggle, unlike before. But at the heart of AI and foundation models is deep learning. It's one of my favorite things to study and analyze. So this was pioneered by Hinton, LeCun, and Bengio way in the 80s. And their work in neural networks has laid foundation for today's AI revolution. So something about deep learning, it automatically learns latent features from raw generic data, any sort of data. And it is well suited for sorts of integration from proteomic data to multi-omics integration. Most of the foundational models that we have at the moment are built on deep learning architectures. Unfortunately, as the previous speaker said, we still need a lot of data to make these models effective because they require large data sets just for them to give us reliable outputs. A simple dive into deep learning and how it works. So you need just data, a neural network, a learning system, and finally prediction. And it's pretty simple on paper until you start doing it. Now, what you need at the core of it is data input, in this case, DNA sequences, and then you have multiple neural networks, layers of artificial neural networks that process your data. This is followed by featured learning, where early layers learn simple patterns, deeper layers learn increasingly complex patterns. This is succeeded by training, where the model compares its prediction with the correct answer and adjusts millions to billions of parameters to reduce error. And finally, we have the prediction layer, where the trained model applies what's learned to new unseen data. Unfortunately, deep learning is very powerful, but very resource intensive. So what are some of the challenges with deep learning? One thing is large data sets. We've spoken about this in the previous talks. So massive data, well-trained, curated data. Africa still has a massive data gap for production, standardization, QC measures, guiding genomics and bioinformatics. We've had these conversations with my collaborators from the West, just how best can we come together and figure out how do we standardize systems so that whatever is happening at Institute Pasteur is happening in Kisumu, is happening in Nairobi, is happening in Kigali. The other challenge is compute requirements. GPUs, accelerators can be very expensive. We are yet to get there as a continent when it comes just having good compute that can handle deep learning models. And then specialized expertise. Interestingly, the AI era will be driven by computer scientists. Or worst case scenario, people are willing to drop lab work to pivot into this space. And it will require teamwork between different expertise, data scientists, computer scientists, lab scientists, to come up with innovations that can help us make use of the data that we have. And then infrastructure is still a challenge. Storage, networking, and cloud access matter. And this points to compute. And then there's the aspect of reproducibility and bias, whereby models can reflect gaps in training data. We've seen initial models that were trained in data from Europe coming to Africa, and they failed to deliver their objectives. So looking into where Africa should begin, it's pretty good to be ambitious. It's pretty good to speak about what we can do as a continent. And at this point, when you look into statistics, this is somehow old, but it communicates something. So the role of AI in key African industries. There's so much focus on fintech, some solutions in agri-tech, and Europe, East Asia, North America, Indo-Pacific are leading in AI impacting the GDP worldwide. Unfortunately, the sub-Saharan Africa, we are at the bottom, 1.3%. If you go into tech startups as 2021, you realize we may fall under other because genomics is not really accounted for. And this boils down to lack of innovation or spaces that can allow genomic scientists to work together and push conversations forward. So much funding is in fintech, unfortunately, yet genomics and public health are the ones that drive the economy and hold the world together. Diving into the AI and genomics market, now we're seeing 2023 to 2033, steady growth, 19% moving forward. But there is so much focus on drug discovery and development, precision medicine, diagnostics, other applications. So as much as genomics cross-cuts all these spaces, to an extent also innovators in this space, especially in the public health research space, they likely to fall under other applications. Unfortunately still, most of these innovations will be built for the West. Not so much is happening in these spaces of Africa beyond using the support AI infrastructure. So where should Africa begin? We look back to what happened, the post-COVID surge in genomics applications, expanded sequencing capacity across Africa. That was visible. There was support financially to do that. But over time, cost of sequencing, infrastructure, specialized expertise, that's a big challenge in a continent where we're heavily relying on donor funding, and most of these donors have their own objectives. And that's why, as the previous speaker, Prof, said, some of this data ends up in California because there was an agreement that will ship this specimen to California, unfortunately. So AI inequality is projected to affect LMICs despite efforts to democratize AI, especially in areas that require deep expertise, such as public health and research. Then, as Africans, in my experience and expertise, one thing that comes clean is we do not need to build large AI models from scratch. We can basically integrate AI into existing genomic workflows and progressively scale. So there exist lightweight solutions that require more manageable investment, cloud infrastructure, workflow automation, data integration, data sharing, and knowledge democratization. Unfortunately, also, we yet to get there in terms of training. So for instance, the AI engineers or the computer scientists are stuck in their field. I, as a genomic scientist and a virologist, I'm stuck in my field. There exists no platform where we can work together, computer scientists, genomic scientists, virologists, AI engineers, and even data scientists, just to work together to build systems where everything communicates. And the goal is, can we push innovation? Can we make Africa a priority when it comes to AI innovation? So I'll speak about a case study of PAN, the Gender Initiative. So I'm the co-founder of Gender Initiative, where I'm also the scientific director. And the goal for the Gender Initiative was just to build innovation, platforms beyond what's existing. And it's grounded on genomic data, bioinformatics, artificial intelligence, genomic intelligence, and actionable insights. So one of our first innovations that we're in the testing phase, and we are hoping to roll out in September, primarily focuses on influenza and RSV. That is my domain expertise. But pretty exciting is the foundations that have been used in designing PAN can be used in other spaces, biodiversity, anything else. We just need to go and tweak the infrastructure that builds the backend. So the opportunity, and this cross-cuts across different spaces. So we have NCBI, we have GSAID, we have Pathoplexus. These platforms contain vast amount of underutilized data. But making sense out of this data requires specialized expertise, computing, downloading, and reanalysis. My previous speaker just said it took him some time to go back to all these databases to download data and make sense out of it. It was only possible because he has expertise that can do this. So what happens to that public health, a professional somewhere in the DAB, a policymaker at Africa CDC, they don't have this expertise. So PAN's approach is quite different. We try to bring analysis and intelligence closer to data through APIs and automated workflows. And as I said prior, the current focus is RSV and influenza, but there's opportunity to expand this beyond these pathogens because the infrastructure allows easy tweaking just to accommodate other needs, especially in Africa. So unlike using deep learning, the crazy models that are out here, we've utilized a simple bioinformatics foundation by Ruby, which is an open source Ruby library. It allows for pragmatic sequence processing, analysis, database interaction, and phylogenetic workflows, creating a computational layer between genomic repositories and AI. And then we've added a second AI layer that consists of natural language processing and generative AI, built on these bioinformatics workflows to support automated analysis, interpretation, natural language querying, and knowledge discovery. So finally, we have democratization, whereby we are allowing even non-experts to interact with genomic data, interpret it across academia, research, policy, and public health. So 3 simple layers: AI-powered analysis, sequence comparison, and then real-time surveillance. So pretty exciting is everyone is depositing, for instance, RSV genomes to GSAID and NCBI. But at any point, no one can understand what's happening globally. So to do that, you have to go back to NCBI and GSAID, get this data, download, with good expertise, reanalyze all the genomes, and then finally say genotype AD.1 is circulating, and we are seeing these changes and all that. But with platforms like PAN, we are allowing this to happen by simple queries, because by a simple query, the calls are made to all the databases based on the time frame that you want to analyze your data. And then you get outputs that are at expert level, that are at policy level, and finally, even at natural language. So it's allowing even beginner people who just wants to understand the natural language of surveillance, the natural language of biodiversity, without really needing technical expertise. So, I mean, Jenna's vision has been to build open, scalable genomic AI systems that transform growing biological data sets into actionable genomic intelligence. And the vision was to create an environment where software, AI engineers, computational biologists, data scientists, and lab scientists innovate together. And part of the vision as the AI advances, so beyond platforms like PAN, beyond platforms that can support integration, beyond platforms that can monitor mutations in real time, we looking beyond the single reference, where we have this vision where we want to shift from reference-based analysis towards learning from genomic diversity. So it allows data being sequenced at MKU. If someone has 48 genomes at MKU, and there's this huge database with built diversity, they can throw their genomes into the database, and these 48 genomes can be compared against thousands of genomes globally. And with this, we are trying to accommodate the complexity of microbial populations and even biodiversity as the science progresses. We are allowing data to learn from thousands of genomes, whereby we use deep learning to identify patterns, relationships, and emerging signals across expanding genomic data sets. So this shifts the whole approach from comparison to intelligence. At the moment, we are always focused on how does this sequence differ from the reference. So you're sequencing influenza and reference genome. So what's the difference? Are we seeing mutations? Are we seeing anything? Are we seeing vaccine escapes? But then we're hoping for a place where we can say, what does this sequence reveal in the context of 10,000 genomes from Africa? Are we missing out something that the reference-based analysis cannot give us? So looking into this whole landscape, as I wind up, is the key takeaways and future opportunities. So Africa can begin integrating genomic, environmental, ecological, geographic, and climate data sets to better understand its biological resources. AI can make these diverse data sets easier to query, connect, and interpret without requiring every user to be a bioinformatics specialist, without requiring every institution to have high compute power, without requiring people to literally take 2-3 weeks to make sense of what's happening. And at the same time, in spaces of surveillance, it can allow to compare the time spent between when a sample was collected to when its genome was deposited on GSAID or NCBI. So this will allow continuous integration and monitoring, which can support early detection of changes in species, ecosystems, pathogens, and also biodiversity. And as I said prior, most of these foundations are built on frameworks that can allow tweaking of the models to accommodate different needs. As long as the input data is sequences, then just tweaks are done in the back end, and all is just work towards accomplishing the project's objective. So platforms such as PAN demonstrate how existing data and computational infrastructure can be connected to create new forms of biological intelligence. But then the opportunity to build an African ecosystem where data not only generated and stored, but continuously integrated, queried, and monitored requires knowledge between, different domains. So computer scientists, public health people, genomic scientists, AI engineers, and also data scientists. Building such an ecosystem will allow Africa to innovate, act on its own data, and then consequently will go just from generating data and depositing it in databases to creating opportunities through AI. Unfortunately, deep learning still has barriers, and Africa just needs to start small. And PAN, amongst other innovations, generate some of the approaches that we can use to navigate the cost of compute and creating opportunities where organizations can now easily start making sense of their data. Thank you.
So thank you very much for that insightful presentation. So I think I'll ask the first question. For the PAN platform, you've tried out probably mostly viral genomes. So my question is, have you tried out pathogens that have a bigger genome, like certain bacteria, using the PAN platform? And what was your experience?
Yes, so we are still in the developmental stage, and we're just piloting, testing genomes. We are hoping to deploy this latest October. We've started with viruses because that is the domain expertise, and it's easy for a virologist like me to say, okay, this is not really making sense, and guide the computer scientist on the best way forward. We yet to consider expanding into bacteria, but it's an opportunity for the future. And the main challenge that I see also as PAN expands is will require some level of compute, considering that viruses are smaller genomes compared to bacteria. But how the whole framework is built, there's opportunity for focusing on also more on bacteria, but this will require also investing in more resources, specifically compute.
Okay, thank you. So we can take a few questions from the audience.
Thank you so much, Dr. for the amazing presentation. I just have one in relation to what my colleague has asked. I'm eager to know if PAN has capacity to foster pharmacogenetic surveillance for vaccines that are currently in deployment phases. I know KEMRI has been very pivotal in spearheading vaccine rollout for malaria and influenza. Do we have any data that supports pharmacogenetic surveillance for the vaccines currently in deployment? Oh, sorry, I forgot basic protocol. I'm Dr. Sarah Kitui from the Center of Malaria elimination here at Mount Kenya University. I'm also affiliated to the University of Embu.
Yes, thank you, Doc. So one way to navigate the growth of PAN was to make this platform purely open source. Challenge that we had at start was it's being built purely by genomic scientists who are heavy on virus surveillance, bioinformatics who are heavy on just analyzing data. There's opportunity to expand into pharmacogenomics. And one goal was just to invite more contributors, invite more experts, because this solution is built for Africa. And looking forward, there's opportunity to expand this into those spaces, because just looking into the framework and being part of vaccine trials in Kenya, especially RSV and influenza, those questions came in, but it also boils down to the need for more compute. So yeah, there's opportunity, but this should come in the later phases. But at the same time, we're inviting collaboration. If you feel like this platform really suits your needs, we sit together with the computer scientists, with the software engineers, with everyone on the platform, and then we like, how can we advance this so that it can take care of pharmacogenomics, it can take care of everything beyond viruses. I hope that answers the question.
Thank you. Yes, it does. Thank you.
Thank you.
Any question online?
Yes, we have a question from Arago Amare. Please unmute yourself and ask your question. Arago Amare.
Arago, could you please unmute? I think you can go ahead and
Yeah, if there's no other question, let's go back to the floor. Moderator, any question on the floor? There's a question there next to Dr. Cecilia.
Hello, my name is Edwin, and I'm from Mount Kenya University Center of Malaria Elimination. AI is here to stay and seems to be a very pertinent part of research going forward. Do you think that we should now introduce AI as a course at the university level, at a degree level, or how do you advise?
I mean, it's time we have AI become part of our curriculum. Unfortunately, when you run through this society and ask anyone, what is AI? They'll tell you it's ChatGPT. I mean, thanks to Lamartman, he's done a good job taking AI mainstream. But beyond querying and beyond that, it's time we push our curriculum towards the fundamentals of AI. And I'm happy that it's happening in Europe, it's happening in the Americas. So it's time we also pivot towards that as a continent. But this will require people working together and coming up with a curriculum that is not more theoretical, but allows people to really have the expertise that is needed to drive Africa's future in the AI era. If we don't do that, unfortunately, we'll remain consumers rather than producers of models and innovative tools.
I think we would want to conclude our questions at that particular point. Let's applaud Daktari. So with this, ladies and gents, allow me to hand over the session to my colleague, Dr. Protas Omondi, who will be chairing the next session. So thank you so much for your cooperation.
Thank you very much. So please, let's appreciate Professor Clement for the moderation of that session. Thank you very much. So my name is Protas Omondi. I'm a postdoc fellow here at the Center for Malaria Elimination. And it is my pleasure to welcome you to session 2, focusing on AI tools and emerging technologies in biodiversity and integrative genomics. So we have an excellent lineup of 3 presenters, and because of limitation of time, I'm going to keep an eye on time so that we ensure all presenters have the time they deserve. So with that, let us begin. Our first presenter is Mr. Paul Gashora, founder and CEO, Project Mocha, Nairobi, Kenya, who will be taking us through presentation titled From Living Organisms to Investable Digital Assets. Please join me in welcoming Mr. Paul Gashora.
Paul, you can go ahead and share your presentation and unmute yourself so you can speak. I think you have the right to unmute yourself.
Hello, good afternoon, everyone.
Good afternoon, we can hear you.
Thank you. Can you see my presentation?
Not yet. Please share.
We can see you. Yes, now
Yes,
we can see your presentation.
perfect.
Okay. So let me first take the moment to appreciate the convening of these sessions. I'm very much honored to be in this gathering with the doctors and professors and the many of you who are from the field of academics and research to just bring forth my thesis and the work that we have been doing around biological assets and making them visible to the current financial infrastructure. So a brief introduction, I'm Paul Gashora. I'm a coffee farmer, third generation coffee farmer, in fact. Over and above that, I'm the founder of Project Mocha. And Project Mocha ideally is we are looking at being able to create digital records of the existing organisms. This is, in our particular use case, we are looking at coffee, the coffee production, the coffee trees, and looking at how we can create digital twins that allow farmers to access markets, access capital, and also capture biodata that can be used to improve their production at the farm level. So today's presentation is, as introduced, is around from living organisms to investable digital assets. And as you can see, there's this image of a coffee tree. That's someone picking coffee at the farm. And a question I'd like to pose to the audience is, does this tree have value? And if yes, where is this value recognized? So we are probably one of the, we are considered, Africa is considered part of the global south, nowadays being called the global majority. But nonetheless, this is a term coined to show that from an economic perspective, we are below the food chain globally. And this is in contrast to the fact that we here in Africa are not necessarily asset poor. What is actually the fact is that our assets are invisible to the existing financial infrastructure. So we have, Africa has one of the largest collections of biodiversity, bioorganisms. By land size, Africa accounts for nearly over 20% of the global land mass. Yet we are still, I think probably the poorest continent. So if you actually look aside, not just the bio organisms, if you look at the land being one of the core factors of production, we should be at least carrying at very least 25% of the global asset base or global production capacity or global revenues. However, we are considered the dark continent. This is because our value remains outside the existing balance sheets. Banks, financial institutions cannot see our land, cannot see our trees, cannot see the biodiversity that we have, our forests, our carbon data. The other thing is, so for an item to be captured within a balance sheet, there are core things that need to come together. We need to be able to identify this asset. We need to be able to assess the condition, assign and verify where the rights and obligations, who owns it, and the cash flows or revenue that's generated, the productivity of this asset. All this is not currently visible. And so we have the case of this coffee tree where the farmer knows that the coffee tree has value. However, the banks do not see it. The country, when assessing the GDP, they cannot see the tree. They see the value of coffee being sold, but the tree itself is not seen. The coffee buyers are not seeing it. The global economy doesn't see it. So to just a few numbers I'd like to throw around is Africa contains, as I said, a quarter of the world's land area. When it comes to arable land, we account for close to 20% of the global arable land. Yet of the arable land, we are able to generate income in terms of productivity, less than 5% of when you compare to the other continents in terms of their sizes and the amount of productivity and income that they are able to generate from the same piece of land. So we have currently roughly about 350 billion that is being generated in revenue from agriculture, from an asset base that is worth 6.3 trillion. I know some of the figures they're coming from reports, research done by World Bank, and this is considering that there's still a lot of our assets that have not been measured and captured. So an estimate is that we have a quarter of the global natural capital in terms of our asset base, but then we are generating revenue of only 350 billion from the same. By contrast, looking at the other factor of production, in this case, labor, currently we're looking at Africa holding almost a quarter of the population, and then by 2050, accounting for almost 2.5 billion in the population. And this is not the aging population that we are seeing in most of the countries. Over 60% of our population is below 25 years. So we have two very core, two of the key pillars of production of the four. This is land, we have labor, the entrepreneurship and capital, with a huge gap around the capital. And so the challenge we currently face is how do we create visibility around our wealth? We have the land, we have the minerals, and this is, I know we are talking about agricultural bioassets, but if you're to look at the wealth also compounded by the kind of minerals that we have, we are probably the richest continent. So where is this gap whereby we are asset rich but cash poor? It's in the fact that these assets are not visible. And so the visibility comes in the form of, there's a gap in terms of our infrastructure. We are the least trusted population. And with this, the fact that we do not have infrastructure to, on our own, project our own value, then those who are looking from outside cannot see us, and we are thus perceived to be highly risky. We are undervalued, and we are also exploited. So what we find existing today is we have data sets in as much as, like for example, from a biodiversity, we have data sets which from a global perspective, Africa data set accounts for 1% of the biodata. These data sets are still nonetheless fragmented. And whatever is happening in Kenya is not being reflected in the work that's happening in the Congo, is not being reflected in the work that's happening in Nigeria. And it's automatically also stems from the fact that even we ourselves do not trust ourselves. We do not have even the infrastructure. It's much cheaper to fly from Kenya to Dubai than from Kenya to Ethiopia. The same accounts the same way we don't have actual on-the-ground infrastructure, we also do not have the digital infrastructure that is required. We also do not have like a standardized way of measurement in terms of being able to the protocols that if I present a data set, if I present valuation of farms, possibly in Nyeri County, will my data be accepted by someone who is in Egypt or someone in the UK? We don't have clear rights around ownership, around the rights and obligations of our assets. From Kenya, from those who are in Kenya, we know that land is a very tricky issue. Buying land is actually a high-risk activity. You need a lawyer, you need to talk to the chief, you need to get to the ground, you need to go to the lands office, you need surveyors, and all this is to ascertain whether you can actually purchase a certain piece of land. And even with all that, you'll still get scammed. And after you've gotten scammed, we've seen cases of many people who, even after presenting their issues, we have multiple title deeds, and these cases are stuck in the courtrooms for years, and there's no progression in terms of being able to adjudicate and clearly determine that either an error or a fraud took place, and being able to give the legitimate ownership of a certain piece of land to a person. Scientific outputs, we have outputs, but we are not able to create actionable, underwritable data or from the scientific outputs. So we have, I believe from your perspective as a scientist, there's a lot of data that you're processing, be it from healthcare, from a disease perspective, from the agri, or from like generally the biodata. However, with all the data that we are working on, how much of it can be translated to a balance sheet or can be translated into economic value that a bank can take a claim against, or a farmer can use to insure their piece of land? So, and lastly, which has been a key detriment as a continent, is we have our value leaves. We sell items raw, others are exploited, and we are not able to retain the production, the value of our own assets. We sell, for example, here in Kenya, we sell 95% of our coffee raw, that means unprocessed in terms of the green bean. And 70% of the value of the coffee accrues after it has left the Kenyan continent, the Kenyan country, and the custody of our country. So we sell at a throwaway price, and as a matter of fact, we're not able to have our own identity around the coffee we produce. Our coffee, for example, is used to blend other coffees as opposed to creating a Kenyan brand. So what we require in this particular case is to come up with infrastructure that we can all share in terms of access and in terms of adding data and verifying this data. We call these trustless protocols that can be used to verify and project our actual wealth. And for this, it's very easy to come and say we're going to build a certain platform. However, before we get there, the platform in itself is not sufficient to solve the trust problems. because we need to first come up with the standards of measurement. How do we get data into this platform or into these databases? What methodology will I use to say that farmer A has 100 coffee trees, and these coffee trees are worth 1,000 100,000 Kenya shillings for these 1,000 trees. Likewise, once we've come up with this unified way, these protocols to measure and capture and recognize our assets, then our record keeping has to be interoperable. We ought to have different actors able to access the same record, access the same data, and be able to infer different use cases from the same. The government should be able to access the same data that the farmer is accessing, that the bank is accessing, that the coffee buyer will also access. And all this data is not ideally not to be in one silo, one database, but different databases ought to be able to communicate with one another. Rights and permissions are a key challenge, as I mentioned about the piece of land. But even as we make digital infrastructure and digital assets, we have to first clarify the ownership, the rights and obligations of our actual assets, and ensure that as we open up our digital borders, we do not get into a situation where we are compromising the rights of ownership of the inherent and the indigenous communities. Verification and assurance, and I'll touch that also with in terms of claims settlement. It should be, our infrastructure should allow us to have a bank is able to verify that the farmer has an identity. This particular farmer actually owns this piece of land. They have these assets, which are their coffee trees, their avocado trees, the indigenous trees, and they are able to either as a farmer, either borrow and the lending institution lend to this farmer based on this data that is provided. And they should be able to rely on it with a degree of confidence, as they are currently doing when it comes to the current, let's say, asset financing, where they are able to look and see this is a vehicle, here's the ownership documents, we have the logbooks, we can actually verify who owns this asset. We can see that the asset is there, and then we can also now put our financial claim to it. We can lend against it, and also be able to receive our settlement out of it. If I'm lending as a bank, I should be able to know that I'll be able to receive my money back out of this. So this will be, ideally, these are the key tenets of the kind of biological infrastructure that is necessary, or we need to be building or are building, to ensure that we can bridge the trust barrier that we currently have. I'd like to touch on the role of government, because currently we are politics accounts for possibly over 70% of our economy and our social life. So governments have a key role to play in being able to spearhead the formation of this infrastructure. Infrastructure, I mean, we're looking at not just the AI in terms of the models, but also looking at having the data centers, having capacity to create our own models, having the capacity to build our own blockchains, and having government creating an enabling environment where even the private actors can invest in this case. If you look at the AI economy in the last couple of years, spending towards AI has more than doubled year by year. If you take case in point what's happening in the US and China. However, if you look at what is happening here in Africa, we are not at the position to spend this kind of money. And as the previous speaker said, that we are left in a position where we are consuming the technologies as opposed to being at a position where we are producing them. So government has a key role in providing this enabling environment. ensuring that the rails are built. They do not necessarily have to build the rails, but creating an environment where an investor can come in and put in a data center. People can come and set up the models here. We can have electricity and energy production investment taking place and have this infrastructure invested and built. It is critical for our policymakers to realize that since we are so much far behind, we need to be proactive in terms of the kind of policies and the laws that are made. Not just taking what has been done in the developed countries and copy pasting and just adding license fees on top, but looking at how to spur growth, spur in production, spur innovation, and also spur investment. So I look at this in three key buckets. And the first bucket is observation. Are we able to look and see what we have? Are we able to capture our assets? Can we tell the kind of biodiversity of Kenyan coffee farms? How many trees? What's the canopy what's the canopy assessment of coffee farms in Kenya? What is the biodiversity? How many insects do we have, and how are they contributing towards the production of the coffee? What is are we able to capture like rainfall and weather patterns accurately? So this is where we are looking at observing, and this requires both sensors, the IoT sensors, and also the study of when being able to capture. These are the omics, like being able to, the genomics, which quite a number of you are looking at, remote sensing, and having IoT and mobile sensors to capture this data. Once we have the data, are we able to analyze it and read it and make useful interpretation, make this data make sense to us? This is where we classify it, come up with risk parameters and the risk ourselves. Because at the end of the day, what you're fighting against is the perceived lack of trust and perceived high risk that we have as a continent. Are we able to detect different anomalies in the data that has been collected, and we are able to analyze and create data that information and enable the decision making? Finally is the storing and sharing of this data and the actionable data. And this is where the ledgers come in. There's been a lot of growth within the blockchain space, within the AI space, and within the IoT space. And so we really do not need to be thinking around creating or coming up with new ways of doing it. It's just being able to do it ourselves as well. So having our data stored in blockchains and having not just the data itself, but do we have rights and contracts that can transfer rights from person A to person B? Because a coffee farmer, can a buyer from Europe get onto a platform, click and buy coffee, and be certain that they will receive delivery of this coffee? So it's not just having the data on a platform, but are the rights provided for on this platform, are they actually enforceable, both digitally and in the real world? So in terms of just a simple practical example of how this works is, we enroll a farmer, create a farmer identity. So get to the field, get to know the farmer, get to know the piece of land that he has, get to do an audit of their trees, of which from a finance perspective, these are actually assets. So create an identity that can be used by the farmer, and they can be used by third parties like the government, by buyers, by financiers to transact and trade. We then need to be able to continuously observe and generate data around the same farm, so that we're able to not only see what is currently there, but also come to a point of projection, and also in terms of improving the farm, where we are looking at precision farming. We do not need to spray the entire piece of land with a herbicide or with a pesticide. We just need to get to know that a certain block has a certain issue and we need a certain intervention done. And this is where AI really would come in play to analyze and consume this data and give us actionable intel. From a transaction perspective, we need to be able to prove that certain actions have taken place. Can we prove that this data, the coffee, the farmer, can we prove that his historical production is there? And can this provenance be trusted? Once we are able to get there, then the finance will definitely trickle in. The buyers, the funders will come in. Currently, we'll see that here in Kenya, CBK reports that less than 5% of the bank's loan portfolio is in agriculture as a whole, while agriculture accounts for over 20%. Why do we have such a gap between our GDP and production and the amount of money going to it? It's the fact that the banks, the lenders, are not able to satisfy their risk, and that this provenance, that what we claim as farmers, what we claim as the producers is true, and that our data can be relied on. And finally, there will be the payments and settlements, where coffee is sent, the coffee is received, a buyer pays, and the money gets to the farmer. And whoever who was involved in investing or providing capital, this amount is received. All of this, the technology is there. However, can we able to apply it and implement it in our case? So in terms of bioassets, I see there are 4 core areas where bioassets fall in, which is looking at how do we improve our food and agriculture. And this looks at increasing our production, getting capital into industries, insurance. We are currently not able to insure crops adequately, insure livestock adequately. Trade finance, how these are the low-hanging fruits of having provable data that can be shared globally in a trusted way. Then we're looking at nature and resilience. We have some of the largest bioecosystems, from forests to water bodies, marshlands, carbon sinks. How are we able to verify these ecological outcomes and create wealth out of it and be able to earn out of it? And there's a biological knowledge. There's so much research that needs to be done. We do not have sufficient data sets on African species and specimens. So how do we fund ourselves to ensure that we are capturing this data, and this data can be used as knowledge, and we can become now the providers of knowledge? And finally,
wind up, Paul.
Okay. And lastly would be the programmable finance. With all this data put in place, can we automate contracts? This is through tokenization and smart contracts, where we do not need to rely on you seeing me or a court executing a contract, but all this can be executed automatically. So I'd like to conclude that we have an opportunity to leapfrog the current establishment, leapfrog our global north continents, and by being able to adopt these technologies, but then localize them to our own issues, to our own needs, specifically creating an environment, creating data that can be trusted, and de-risking ourselves. Thank you.
Thank you very much, Paul. Let's appreciate Paul. Thank you for that insightful presentation on how we can convert living organisms to digital assets. So kindly allow me to allow only one question because of time, please. Any online question, please? So if not, maybe, sorry, yes, please.
Oh, thank you very much, Paul. Once again, I'm Dr. Kitui from CME, Mount Kenya University. You've given us a very elaborate presentation on what you're doing, and I'm just wondering if where you sit, you looking for like collaborations to front some of what you're doing from Project Umoka, sorry, because when Dr. Michuki was presenting on health associated aspect and of course the role of African genomes, he mentioned something to do with biotagging of genomic data. And you just mentioned that some of our coffee products are normally shipped to European markets and maybe blended with other coffee products from other markets. Is there room for collaboration, especially for biotagging of our coffee products, so that then when they end up blended into other products across the world, we're able to tell that this has a Kenyan origin? Where do we sit where the labeling and the tracing of Kenyan coffee products is concerned?
Thank you, Dr. Tutui, for the question. And I'd like to say that collaboration is a necessity. Unfortunately, we are so far behind in terms of adoption of the technologies. And not just adoption, but we do not have the capacity to go it alone. So we are left with only one option, which is to come together and collaborate. And that's why I had emphasized on the role of government, because it would be ideally a really good neutral player that will allow different players to come together. However, as we look at the progression of our political systems, I think it's left up to us to find ways to collaborate and to see how do we make our technologies, make our work interoperable. The work that you're doing in genomics has a significant outcome in how do I price my coffee? How do I prove that I'm an organic farmer and my coffee has a specific identity compared to coffee from another region? So we really need to come together. We need to bring different fields of knowledge, fields of study, the investors, the people who are in IT, the people who are the farmers, researchers, to come together and have a concerted effort in being able to project our wealth, to recognize it, measure it, and present it as it should be.
Thank you very much, Paul. Let's give Paul another round of applause. Our next presentation brings us directly to research. Ms. Mudhoni Silverson, a founder, AI and Insights, will speak about from data to discovery, applying AI and emerging technologies in biodiversity and genomics research. Ms. Mudhoni, the floor is yours. Karibu. If you're able to share your slides.
Mudhoni, we can hear you, but you're very faint. Okay. Could you maybe add your volume?
Right, I'll do that. I'm sharing my slides as well. Okay, so welcome to today's session.
We can't see your presentation. Have you shared it? Yeah, it's loading. Yeah, it's okay now.
Perfect.
You can see it now.
Okay, thank you so much.
Please
Good
go ahead.
Good afternoon, everyone. I'm sorry if it's a bit faint, but it's a great honor to be here with you all and to welcome you to this session. I think we'll rush through it in the interest of time, but I'd like to highlight, as my previous colleagues have mentioned, that we are living in an extraordinary moment where AI, genomics, and biodiversity in data are coming together to help us transform how we understand life. So today is not just about learning new tools, but it's also about turning data into discovery and then turning this discovery into impact for Africa. So we'll get started as our moderator highlighted. My name is Modoni Wanyuike. I work as a customer engineer helping companies and individuals identify and get value from AI. And we can see that when it comes to data and discovery, it's important for us to explore how we apply AI and emerging technologies in biodiversity and genomics. So we'll be looking at taking us a step further to see how AI is reshaping and how we are turning biological data into insight. AI helps us to reveal patterns, to identify relationships and signals that would be otherwise incredibly difficult to uncover manually. But this is not simply about having more data. It's also about having smarter data that is connected in smarter ways so that we are able to answer the bigger scientific questions, and we are also able to see the three powerful shifts that we are experiencing. The first being that biology is now generating data at an extraordinary scale. So from genomes to proteins to biodiversity observations in images and environmental measurements, satellite data, and even sound, AI is giving us new ways to make sense of all that complexity, to identify patterns, to make predictions, and also to answer questions. And thirdly, emerging technologies are connecting these previously separate sources of information. So the opportunity before us today is enormous. And the question is no longer how much data do we have, but what can we discover from it? And that's where our journey today will be focused on. When we look at the data explosion, we can see that our challenge as researchers is no longer simply generating data, but being able to understand the data that we generate. We have genomes, we have proteins, we have images, we have satellite imagery, we have sound data, but more data does not automatically mean more knowledge. The more powerful discoveries will happen when we can connect the relationship between the data sets. And this is where AI becomes transformative, where it can help us to identify patterns and possibilities that are too complex to uncover manually. But this is not to say that AI replaces the researcher or the scientist. AI reveals the patterns, but the scientists provide the meaning. And every promising insight still has to be tested, to be validated, and grounded in scientific evidence. So our goal is not to have the machines discover for us, but our goal is to enable us to discover more. And perhaps this is the reminder that we have today that technology gives us greater capacity, but human curiosity gives that capacity purpose. I'll repeat that as we transition. That technology gives us greater capacity, but our human curiosity is what gives that capacity purpose. Then we'll look into the discovery pipeline. So how do we move data to discovery? And I think of this as quite a simple journey. The first is where we begin with data. These are the raw biological observations that we collect. And then we use AI to identify patterns and relationships that are hidden in the data. Then as researchers, we turn those patterns into an understanding. We are able to decode biological meaning, and then this can translate into hypotheses, which we can validate, and then we get to the discovery phase where we are able to uncover new scientific knowledge. So AI is not the destination, but it's an accelerator in this process. The scientist remains at the heart of it, asking the right questions, providing the right context, and also testing evidence that ultimately turns to discovery. I just added this slide in case we have a too long didn't read moment in the presentation, and this is just to highlight that the key areas where AI thrives is where we have large data sets, where we are also looking at pattern recognition or repetitive tasks, and where we are looking for real-time insights. So the opportunity for us as researchers is to discover new opportunities, new species, new variations, and new information, to also predict the distribution of species, as well as changes in the ecosystem, to detect threats early using historical data. We can be able to detect poaching activity or deforestation or destruction of invasive species. And then finally, just to highlight that the key challenge within the opportunity of AI in biodiversity is that data continues to be quite fragmented, and in some areas, as we have been told, for example, Dr. George highlighted that only 1% of the African genome is represented in the Cancer Atlas. That means that some data is still hard to access. Then there's still the challenge of limited high-quality labeled data, then the skills and infrastructure gaps. And what this means for us is that AI will not replace experts, but it's going to empower them so that there is collaborations that empower big impact for tomorrow. I also added a snippet of a prompt vault in case you're looking for some prompts that you could use or that you'd want to take away from this session, as I was supposed to highlight on some of the practical aspects of this, is that if you're looking, for example, at literature and knowledge synthesis, you can use a prompt such as summarize the latest research on this topic in biodiversity and highlight the key findings, the gaps, and the future directions. So that really accelerates your path to research. If you're at the data and insight stage, you could use a prompt like to analyze this data set and identify key patterns, trends, and anomalies. If you're looking at threat detection, identify signs of deforestation or habitat loss in this satellite imagery, and summarize the key hotspots, and you can use this for early warning, for conservation and monitoring. I'll be sharing these slides so you can be able to check more of the prompts that can help you in your research journey. Then I think the previous speakers have very clearly highlighted this, and that is the purpose of AI for both biodiversity and beyond. So I'll keep that slide for now, but you can look into it later and go into this concept where we are saying that we are moving from analysis to augmented discovery. We have the traditional scientific workflow where you begin with questions, then do your experiments, your analysis, and then you're able to conclude and publish your findings. But now with an AI augmented workflow, you're able to add AI at the different levels of your research. This means that you can analyze data sets more rapidly, you can identify non-obvious patterns, and then every discovery creates new questions and makes the process more iterative. Then, so this means that AI's greatest contribution to science will not be answering questions, will not just be answering questions faster, but it will also help us to discover which questions to ask next. Then within biodiversity, AI will help us to make the invisible information visible. A genome is more than a sequence, and sequencing tells us what is there. The real scientific challenge is understanding what it means and how it functions and how it's going to influence biological outcomes. So AI is increasingly being applied across this journey to help us interpret genetic variation, to identify patterns across genomes, to model gene to gene and gene to environment relationships, and finally, to generate hypotheses about biological mechanisms. So on this, I would like to highlight that the key points around biodiversity and making the invisible visible is that modern genomics will continue to produce enormous volumes of sequence data. And the difficult part in this is connecting the sequence to the biological function. So we can use AI to learn the patterns across large and complex biological data sets. And then this helps us within our validation journey and ensures that our experimentation and validation processes remain essential and provide scientific proof. Then still, we now have, thanks to AI, a new biodiversity data layer. This means we are able to use camera traps, for example, to study species and their behavior. We can use satellite images to study habitats and environmental change. We can use environmental temperature, moisture, and conditions. And even in some cases, we can use the sensors on our mobile phone, which are able to collect quite an array of data. So sequencing gives us the letters of the biological language, and AI is going to help us to understand the grammar behind this. When we connect the signals of life, we begin to see the ecosystem and not just the species. Sorry, if I went back here. When we connect the signals of life, we begin to see the ecosystem and not just the species within it. So as we conclude, we have to highlight that the new scientific stack that we are looking at that helps us in the next generation of biological research will not just depend on one technology, but a convergence of multiple technologies working together. That means being able to look at genomics within the sequence and molecular layer, then being able to look at AI and foundational models for prediction and data analysis, then also using cloud computing to be able to scale our research and also for efficient storage and collaboration, and then technology like computer vision to help us analyze images and to identify species. So as we come to the end of my presentation, we can now see clearly that there are infinite possibilities that are within our pipeline. And this is because science is helping to evolve new technologies that are more personalized and more connected, meaning we can have smart hospitals and smart hospital care, remote patient monitoring, robotics in healthcare, and also advanced data and analytics. Then these are a few questions that we must ask, and the fact is clear that technology will enhance biodiversity, healthcare, but compassionate professionals remain at the heart of every patient experience. So for us to have better a better scientific workflow, we have to look at combining our technology with empathy, improving our communication, supporting continuous learning and innovation, and then looking at privacy and quality care and focusing on better outcomes. So I think in the interest of time, that's the highlight of today's presentation, and I hope that from it, you've been able to get a few tools or tips on how to go about handling AI in biodiversity. Thank you so much, and back to you in MKU.
Thank you very much, Mdoni. Let's clap for Mdoni. So thank you for that interesting talk on how we can apply AI to transform our data into reasonable insights. So I'll take two questions from Mdoni, please. Okay, maybe I can go first. So I like the fact that you've said we should not just be consumers. So as African researchers, what's your advice on how we can now become innovators and not just consumers of AI? Maybe you can comment about that. Thank you.
Mdoni, please go ahead.
You're muted if you're speaking.
Just unmute yourself.
I think you can talk. No, you're not muted.
You're muted still.
Sorry about that, and thank you for your question. I think as African researchers, the opportunity we have for innovation is, as we've mentioned, is that there are many gaps within our data sets. I think where we can start, and this is a practice that I've seen us apply in industry, where you need to create AI models in areas that there are data gaps, is that as a researcher, you can begin by creating synthetic data sets. So use AI to create synthetic data sets, which in my opinion, work better than probably using existing data sets that have many gaps. So I would say start there. And then this will help you within your path of research to be able to use AI to fill in gaps that you encounter on the way.
Thank you very much. Let's give Mdoni another round of applause. Thank you. So now we go to, it is my honor to invite our next speaker, Dr. Ansia Van Coller. She's a senior scientist, South Africa Medical Research Council, and our presentation promises us to give us more practical insights. And the title is AI-Assisted Bioinformatics, Practical Experience from Genome Assembly and Annotation Projects. Please welcome, join me in welcoming Dr. Ansia Van Coller. You're welcome.
Oh, thank you. I'm just waiting so that I can share my screen. I think the previous presenter will need to stop sharing first.
Yeah, you can try now. Try and share your screen. Yeah.
All right. Can you see it
that's
now?
good. Yes, please proceed.
Okay. Excellent. Thank you very much. Move all the way. Oh. Okay. All right. So thank you very much for the invitation to present at this workshop. I just feel like I need to give a bit of a disclaimer. So I'm a bioinformatician. So I am, as the previous speaker mentioned, I'm more of a consumer of AI. So yeah, I have a lot of experience with sequencing analysis, including genome assembly and annotation. So I'm not at all involved in any kind of AI policy development or the actual building of AI systems. So I can only comment on my experiences so far when working with some of these various different AI tools during my genome assembly and annotation projects. And where these tools have been very helpful and where they can sometimes mislead us a little bit. So I think based off what the previous speakers have mentioned, I think at this point we all know that AI is very important and it'll be very essential for African biodiversity genomics going forward. It'll be very useful when looking at things like biodiversity monitoring, One Health approaches, when we're looking at conservation genomics. It helps a lot with population level studies and adaptive variation, and also in genome assembly and annotation projects, such as in large-scale sequencing initiatives, like AfricaBP. And as AfricaBP continues to grow, I think that AI-assisted workflows will become increasingly more important because as sequencing is becoming cheaper, the bottleneck is starting to shift from, trying to put money together to do the sequencing, but rather to finding the time to actually analyze the data and interpret it. So there is therefore currently a shortage of specialized bioinformaticians that can work on this data, but AI is helping a lot with this hurdle. So when I'm talking about AI in bioinformatics, I'm mainly referring to these three different types of AI, not just ChatGPT, which I think a lot of people are much more familiar with, but I'm also talking about machine learning algorithms, things like random forest algorithms, which can spot patterns in data, which can be very useful for gene predictions, etc. Also talking about very deep learning, like multi-neural network approaches, which can analyze very complex data, including images, and it can be used to streamline analysis. And then, of course, we do also need to talk about generative AI, so the large language models, which are these text mining tools that can scan millions of sources to help us extract valuable information. So this would be the ChatGPT, Claude, etc. So AI is actually already built into a lot of our pipelines, and I think a lot of people probably don't even think about that already. But here's the very basic pipeline that I use for genome assembly and annotation, and I've just put little stars to indicate where in this pipeline I have incorporated AI-based tools, and you can see it's a majority of the process, and it really does help speed up the process all the way from base calling, genome assembly, all the way down to genome annotation, and I will talk in a bit more detail on each of these topics. Starting with base calling. So I work mostly with Oxford Nanopore Technology long-read sequencing data. And to do base calling for ONT data, you do need to use the neural base callers. So it's not even an option, it is actually the default. So you have to use these AI models to turn your electrical current information as the molecule moves through the nanopore, and then the neural networks translate that into an actual DNA sequence. And so ONT's sequencing quality has improved significantly over the last 10 years. And if we look back in 2015, your average quality score was around 10, whereas nowadays it's quite easy to achieve a quality score of above 25. So it is at this point comparable to a lot of the short read techniques. And this improvement over the last 10 years is not just driven by the changes in the chemistry of their flow cells, but it is substantially driven by the improvements of the AI models that are used for base calling. So in essence, a lot of not just genome assemblies, but all types of sequencing analysis-based projects are already AI assisted from the moment that these raw sequences are translated into sequence data. After base calling, I also like to do additional error correction steps, even though the quality of the ONT data has improved significantly. I like to use a tool known as HERO, haplotype aware error correction tool, which basically compares all of the raw reads and then uses the deep learning approach to distinguish between true variants and sequencing errors while preserving haplotype information. And then you can improve the quality of your raw reads, which can then improve the quality of your assembly downstream. I've just shown here a picture from the HERO publication showing in humans and other model organisms how this polishing can improve the quality score from like 20 to 40 and above. So this has been a very useful tool, and I think if you consider that at this point we haven't really done any analysis yet, and yet AI has significantly improved the quality of our data and therefore our downstream analysis. So for genome assembly, I think it's still interesting to note that not all high-performing bioinformatics tools at the moment are AI-based. Sometimes, the classical algorithms, they still dominate and they can still outperform the newer AI-based approaches. So the top long-read genome assemblers currently, such as Hi-Fi or Fly, Canoe, even Zerko. So these are not traditional AI-based models. They use graph-based, like the brain graph algorithms or statistical overlap layout consensus and probabilistic error correction. So these are very complex algorithms, but they kind of predate AI. Here I've shown an example of the approach followed by Hi-Fiism. It also incorporates an error correction step, and then it generates the complex graph structure, which has been simplified here. But then essentially you can from that generate your primary assembly, and you can also do haplotype resolved assemblies in some cases if you have sufficient data. But so while the pre-AI graph-based genome assemblers still perform the best at the moment, I do still apply AI to assist with the interpretation of my assembly metrics, particularly when looking at the QC outputs like QAST, BUSCO, Mercury, or Genome Scope, because AI can help to explain some of the metrics and identify potential issues with my assembly. Like I might ask, does my genome seem fragmented or incomplete based on my QAST or BUSCO report? Is the genome size as predicted or expected, or is there something that looks funny? But unfortunately, AI doesn't help much when it comes to deciding whether what I'm seeing is biologically correct. It's also not very good at understanding species-specific biology when it comes to novel or like non-model organism species. It cannot really diagnose complex contamination, although it can sometimes hint towards that. And also, it's not always useful for making final scientific conclusions. After generating an assembly, we normally do polishing steps as well. This isn't always necessary because a lot of the assemblers produce very high quality, with high quality scores, like individual base scores. But in general, polishing is still recommended. So there are many different polishing tools. Not all of them are AI-based, but I wanted to show an example of one of the AI-based tools that I do apply, called Deep Polisher. So here you would align your raw reads back to your assembly, and then it uses the alignments, and it considers the matches and the mismatches, as well as the individual base qualities and the overall mapping quality. And then it uses this transformer-based deep learning approach to identify potential errors, and then generate a consensus sequence or polished assembly with a reduction in the error rates and a significantly improved quality values, as in the quality or the confidence assigned to each base in the assembly. But again, I think it's important to mention here that a higher quality value for a particular base isn't always indicative of biological accuracy. It just means that what you're seeing in your assembly was also seen in your raw reads. So you might just be assigning a higher confidence to some type of contamination. So we also need to look at all the different QC metrics. For instance, BUSCO, which measures conserved genes, which might give a better indication of the completeness of your assembly in terms of a biological context. I apologize, my screen seems to be stuck. There we go. So once you have a polished assembly, if possible, we like to do chromosome level scaffolding by incorporating 3D chromatin conformation information. So that would be something like Hi-C, Omni-C, or Poor-C data, which we can then use to scaffold the assemblies up to chromosome level. I put an example here on the side of how Hi-C data is generated for those that are not as familiar with this type of data. Essentially, we do preserve the chromosome conformation, and we do cross-linking, which connects DNA bits that are in close proximity to each other. And then after proximity ligation, we'll end up with a DNA library where the opposite ends of a DNA strand will correspond to different regions of your genome. And then you can visualize this as a type of heat map, kind of like this. In this example, you can see the clear clustering of chromosomes and limited interaction between the different chromosomes to show that this is a good quality chromosome level assembly. But this chromosome scale assembly curation has always been a very manual process. So you would visualize your Hi-C contact matrix like this, and you would manually go inspect and identify regions that look like not quite right. For instance, if we see these dark spots over here, that could indicate that this part of the genome belongs over here. So that would require some manual rearrangement. So you would have to, go look at every part of this assembly manually and try to identify all the misjoins, inversions, misplaced contigs, potential duplications. And then you would manually split the contigs, reorder them, or swap them around if they are in the wrong orientation. And then you would regenerate the map and redo this process until you end up with a good looking assembly. So this can be a bit of a challenge because when we're doing chromosome level assemblies, it presents a bit of a bottleneck. So usually, or beforehand, the biggest holdup of generating these genomes is the actual assembly process because of how complex it is. But now, with increased computing power, that this actual assembly process doesn't take as long. But the part that takes the longest is the manual curation, where we sit for many days or months sometimes inspecting these Hi-C plots to see if we can find the error and make sure that we represent the genome appropriately. And this does take a lot of expertise and training to do this kind of work. But recently, Pretext Map AI has been released, and I found this very helpful. So it employs AI models, and it helps to highlight suspicious regions and identify likely assembly errors. And then it can direct the attention of the curator to help prioritize corrections. So it does significantly speed up the process. So I've just inserted an example here of where I used Pretext Map AI to generate the assembly that I showed earlier as well. So when I aligned the Hi-C data to my assembly and generated a heat map, it looked like this. And as you can see, it is still quite messy. There's a lot of rearrangements required. We can see long distance interactions over the genome. And then when I put it into Pretext Map AI, it was able to apply the scaffolding, and several AI suggested rearrangements. And I was able to go from this to this in less than a day. So it has significantly sped up the capacity to do these manual curations. There are also some completely AI-based pipelines for integrating Hi-C data, such as the Auto Hi-C pipeline, which employs pre-trained neural networks to help produce these chromosome-level assemblies. I want to say the major drawback of this is that you do require a reference genome of a closely related species in order to generate a high confidence report. And again, because it uses these pre-trained models that have been trained on model organisms, this pipeline might not be as useful for non-model organisms with more complex genome structures. So now moving on to annotation. So during the assembly process, we now have managed to piece together the millions and millions of fragments of DNA into a complete genomic sequence. But at the end of the day, that just provides us with a string of letters. I'll put just a little example here of one of my genomes. At the end of the day, it's just a grouping of A's and T's and C's and G's. It's still pretty much unreadable. So we do need to do annotation to add structure and meaning to the data. And so we do this through structural annotation, whereby we identify the exact locations of genes, which regions are coding, which regions are non-coding, which regions are repetitive, etc. After that, we also do functional annotation, where we try to assign biological meaning to these proteins to see what actual functions are for these proteins and what roles that they may play in survival or disease. And then after we have our annotated genome, this does make it a lot easier to do further studies, such as in identifying metabolic pathways, doing comparative genomic studies, transcriptomics, etc. So I've mainly worked with the more traditional genome annotation tools, such as EGAPX or Breaker3. So some of these are starting to increasingly integrate some machine learning methods, and the functional annotation tools also integrate these machine learning methods to help assign meaning to the genome. And it has been extremely useful. There are also several tools that are completely AI-based, such as Helixer or Tiberius. So these are deep neural network tools that can identify ab initio gene models. So it can identify regions that look like exons or introns based on pre-trained models. There's also Gaia, which is the genomic AI annotator, which uses a language model, a GLM2, to identify potential properties of proteins. And also DeepGo Plus, which operates similarly, but it can predict the gene ontology terms and the protein functions faster than the conventional BLAST workflows used in, for instance, Eggnog Mapper. But again, we need to consider that the prediction confidence is not always the biological truth, and a lot of these AI models are built on model organisms, and so may not be relevant for non-model unique organisms. And some of these AI tools, I think Helixer in particular, can often overpredict genes, and some of these protein prediction tools can also jump to some incorrect conclusions with confidence. I also use generative AI in my daily bioinformatics. It is incredibly useful for coding assistance when I'm writing Python scripts or looking at my Nextflow or my SnakeMake pipelines. So this has helped a lot for improving the times that it takes to develop my pipelines, and it's allowed me to better document the building of these pipelines. But yeah, it's very important to always review the code, because it very confidently can produce incorrect code. But that being said, it is also still very useful for troubleshooting. It can very it is to apply these generative AIs in that context. Again, it's also useful for documentation, creating SOPs. I've found it incredibly useful for some data interpretation as well, in terms of summarizing my outputs and providing additional context, which has been very useful for training students and for presenting data. But yes, I always want to just highlight, and I think I've probably repeated this before, but we need to remember that AI is a tool and is not an authority that will replace us. It is really good with pattern recognition, helping to design pipelines. It's useful for documentation, literature discovery, and summary of literature, and summary of your results. and it's been very useful to learn how new tools work. But unfortunately, I do not think AI will be able to replace actual scientific validation, biological interpretation, experimental evidence, the detection of subtle assembly artifacts or other errors. It is not very good at always understanding the specific context of your data set and your species-specific biology, especially if you're working with non-model organisms. And it really struggles to distinguish truth from plausible sounding errors. So yeah, I consider AI to be my friend, but sometimes we don't always get along, and I can spend hours and hours trying to explain context to ChatGPT or Copilot to see if it will help me interpret my data. So we need to also consider the responsible use of AI in the African context. So we do have that scientific responsibility to verify all AI outputs, and we make sure that we maintain reproducibility by keeping track of the versions of the tools that we use and the parameters that we use them. And we always need to keep experts in the loop. So we cannot just let AI run off unsupervised, because it might just keep building on nonsensical outputs. And we also have a big responsibility towards the data. So as we all know, Africa contains some of the world's most diverse plant, wildlife and pathogens, so high diversity and lots of endemic species. So many genome projects can generate a lot of, data which is unpublished but have substantial scientific and potential economic value. So we need to consider this before we upload data into any AI system, so that we can protect our own genomes and our sensitive metadata, to ensure that we also comply with institutional policies and legislation. I think we also have a community responsibility to increase capacity in Africa for the use of AI. We do need to share skills and not just the tools, and promote the use of transparent and reproducible science. So to just summarize and conclude on what I've said in this presentation, AI is already embedded in most modern sequencing workflows. It's very useful, especially for genome assemblies and annotations. And generative AI can dramatically increase productivity. But human expertise does remain essential for interpretation. And so I do not think AI will be replacing bioinformatician anytime soon, but it certainly helps to amplify their capabilities. So instead of now spending many years on generating and annotating a single genome, we could probably do that in just a month, increasing our capabilities. And yeah, the African genomics initiatives such as AfricaBP is very well positioned to benefit from the AI-assisted workflows. Yeah, AI works best when we're looking at identifying complex patterns where there's lots of repetitive inspection requiring expert attention. So, if you think of base calling and polishing, where millions and millions of individual reads are interrogated at very specific positions, using the AI tools really speeds up the time. But humans remain essential when making biological interpretation and evaluating novel genomes and validating the outputs. So to conclude, I just have these 5 points, and that is, we should treat AI as a collaborator, not an oracle. So it can accelerate our work, but it does not absolve us of judgment. We do need to validate our own taxa, because a lot of these AI tools are built off model organisms, and so these sometimes do not hold up onto our underrepresented African species. We need to keep humans in the loop, because AI can make some costly errors, even though it assigns a very high confidence to it. We need to make sure that we govern our data as deliberately as we analyze it, to make sure that we protect our data and we do not accidentally handed over. And we need to make our science more reproducible. So if we do use these AI tools, we need to make sure that we keep track of which versions we use and the parameters, etc., so that that analysis can be reproduced. And that brings me to the end of my talk. Thank you all so much for your attention.
Thank you very much. Let's clap for Dr. Ansia. So I think this has been very useful, especially to most of us who still rely on traditional bioinformatics tool. Here is a chance to collaborate and enhance our analysis. So I will allow only one question because of time. Just one, please. Yes, we have one here.
So thank you so much for that insightful presentation. Again, my name is Edwin, and I'm from Mount Kenya University. My question is that the long-read sequencing platforms, such as Oxford Nanopore, they have traditionally been shown to have generating a lot of errors during the base calling steps. So I don't know if the incorporation of AI is helping to solve these problems, or it's generating the same errors. Maybe from your experience, what do you see?
So I think ONT data has come a very long way. So they do constantly improve their AI base caller. So their default base caller is AI, it's a deep neural network based base caller. And they have improved this model substantially over time, but that does not mean that it performs well on all organisms. But I think the quality has improved a lot. So I also still like to do the additional error correction steps using tools such as Hero, but there are also others that can kind of try and make up for the errors introduced during the sequencing process. I hope that answers the question.
Thank you.
Thanks.
So I think that marks the end of our presentation for session 2. I would like to thank all of us, all our speakers, and our participants. This has been great. And I believe one thing that maybe all of us we've learned or we've been reminded is that technology alone is not enough. Like we've heard, we have the infrastructure. We also have high quality data from our continent. But one important thing that we need is we need skilled researchers. So this is an opportunity for us. And also we need strong collaboration. So I think particularly for African researchers, this is our opportunity. Let's collaborate and learn these new skills. With that, I would like to thank all speakers and all our participating, those who asked questions and all those who are very keen. So let's give ourselves a round of applause. Unless there is any other announcement, I would like to invite us for lunch and exhibitions. So let's enjoy. Yeah, so we get back here after 30 minutes. Thank you very much.
So to our speakers who are online, we can take a short break of 30 minutes. We'll be back here on the top of the hour to start with the afternoon sessions. I do wish you a good lunch break. Thank you.
Researchers can do everything from interrogating the hardest regions and inherited disorders to powering population-scale precision medicine.
Good afternoon. Welcome to the afternoon session. We are going to have young scientist oral presenters. We have around 5 of them. We are going to be very strict on time because we are running behind the schedule. And we're going to start with Dr. Josephat Jenga from Mount Kenya University. You have 15 minutes.
Good afternoon. Help me with the pointer, kindly. Okay, so good afternoon once again, those here physically and those online. My name is Josephat Jenga Geshure. I'll be making this presentation. It's on food and nutrition, so it's good that it's just coming after lunch. I'm gonna be brief because of the time. I did this work at the University of Pretoria, and we did in a team with Dr. Adeline Pretorius and Professor Elna B. So this is just a snapshot of the work that we did. We are just about to send this paper for publication, to submit it. So as an introduction, we know that body mass index, when it exceeds 25, you are considered to be overweight, and when it goes more than 30, it goes to obesity. And we have so many ways of addressing obesity and overweight, and functional food has offered to be a very practical means, since it's somehow cost-effective strategy when you look at just diets, and this is where we have things like prebiotics, and such. So what is probiotic yogurt? These yogurts are products which are enriched with a beneficial organism. We know that naturally and scientifically, yogurt should have at least two organisms. So when we add others outside those two, we refer it to as the probiotic yogurt. But if it just has the two organisms, that's just the conventional yogurt. So in this study, what we did, we wanted to see about adding these strains of yogurt and checking how they impact the gut microorganism. We know that our gut has a lot of organisms. So we wanted to see how is it going to impact these trillions of organisms. Our objective was to check the impact of regular consumption of yogurt, but specifically we wanted to see the abundance shift, and we also wanted to see if there is any promotion on other like how the organisms relate themselves. For the study, we started with getting the ethical clearance, where once we got it, we went to do the sampling. We had 8 people in each of those categories, 18 people, and we categorized them into 9. And these were women who were between 25 and 55 years. And for the control, we gave them the normal yogurt with the Streptococcus, Thermophilus, and the Lactobacillus bulgaricus. For the intervention, it had the same 2 organisms, but we enriched it with Rhamnosus, and we enriched it with Bifidobacterium. So they were supposed to take 100 ml consumption of this yogurt each day. And what we did the sample collection at week 1, at day 0 and at day 12. And what we did for the data collection, we collected fecal samples from them. And from these fecal samples, DNA was extracted at INCABA, and then amplicon sequencing was done at Mr. DNA. And then we used just the normal bioinformatic pipelines in R and Python. So the main part which I'm presenting today are the results. We have the microbial community, then we're going to look at diversity, we're going to look at co-occurrence, and going to look at implication when it comes to health. When it comes to abundance, we find that there's a general trend. There's a general trend of organisms on the phylum. It's important to note that these genera in this phylum, they behave differently based on where they are within the body. And some of them will be having beneficial organisms. They'll be having the benefits. That means there will be things like digestion of carbohydrates, long-chain carbohydrates. They will also come with the bit of reducing the other organisms that may affect products that will interfere with your gut. Organisms that will cause like your gut to have tiny holes, let me call them the tiny holes. You find that there was an enrichment of such organisms. Thank you. So I can stand here. I'm quite short-sighted, I can see there. So we find that there's some of the organisms which are enriched, and these organisms are quite good, and there are those whose numbers went down. But basically, this is the comparison. But even when you just look at it in general, you find that. Okay. Oh, sorry. Basically, you find that conventional yogurt will increase the organisms, but probiotic increases by a greater margin. And also there's differences in how they enrich, as I mentioned. The thing is here, you realize that conventional will increase some of the Akmasias, Shigella, Clostridia, Succinivibrio, cocci. And when you enrich it with bifidobacterium, those will still be enriched, but now it also increases the rumnococci. Rumnococci are very good, bifidobacterium, and also the bacteriocidium. We're going to talk about the clustering. So on the alpha and the beta diversities, you find that probiotic yogurt has a profound way of directing a given community. That is, it's like it moves the organisms, the type and the number of organisms in one direction, which is beneficial to the body. And then also, when you look at those diversities, there is one thing we call the richness. So we want to see the number of organisms. So both yogurts increase, but probiotic increases very much. And also we want to see about balancing of these communities, because you don't know, as I said, probiotic may reduce the unhealthy ones. So you find that the probiotic, it has a way of bringing about stability and resilience. And this shift can be shown when you look at the beta scores. And here we just try to do the eclipses and use the NMSDS, non-metric dimension scaling, and the principal coordinate analysis. And you can see that yogurt in general does a good job, but probiotic does a better job. When you look at co-occurrence, you find that when organisms certain organisms tend to stay with certain organisms. So when you enrich a certain organism, it will enrich a certain group of organisms. So you find that, as I said, the probiotic yogurt tends to make the co-occurrence more compact. So that means that probiotic yogurt, daily consumption, it's going to really drive your microbiome in a certain way. And also, to explain this, you can look at this. I know it's quite condensed, but if you look at just which organisms, you realize that when you enrich bifidobacterium, it enriches certain organisms. When you enrich streptococcus, it enriches certain organisms. When you enrich lactobacillus, it enriches certain organisms. But just to summarize is that you find that things like streptococcus, we say that it prefers the upper gut. And when you detect these organisms, which are commonly found on the upper gut, since we collected the fecal samples, being enriched, it means that this streptococcus was able to survive even the gut. Remember that nowadays people are told that if you want to take some certain medicine, they have to be coated so that they can bypass the stomach acid. So when you find that when you enrich these organisms, even if the stomach acid is going to destroy some of them, there is still a good number that reaches the lower gut. Bifidobacterium prefers to stay on the lower gut, and it stays with a lot of those, especially the rhamnococci and those. So it still continued enriching them. And lactobacillus, it's quite diverse organism. It's one of the most diverse organisms, and it's able to survive throughout. So this said, let's look at the relevance to this. It's a 12-week study, so we want to see. It's always good to be cautious when you tell someone about diet, so that it doesn't affect. You don't over-exaggerate. You realize that the 10 genuses, the 10 genera, will account for 64% of your organisms, and as you increase, the number increases. But when you look at the Wilcoxon, which tells us about the significance, what we call the significance. And when you look at the Cohen's D, where you are looking at the actual effect size of these organisms, you find that still within the 12-week study, there is increases, as we said, but statistically, when you look at the effect change, you still find that they are small. So this is something to be cautious when it comes to making any conclusion on this. But when you look at the statistical significance, you will realize that the p-value is very small when you say about your gut consumption. So in conclusion, we say that daily consumption of yogurt does a good job to your organisms. And when you do even the probiotic, it's better. And we look at clustering, and we are able to see that we are able to do a better when you look at the functional properties, when you're looking at things like carbohydrate fermentation, short-chain fatty acid production. And this short-chain fatty acid, we concentrated on the butyrate producers. So you are able to see which organisms, rhamnococci, Lactospiraceae, bifidobacterium, which were also enriched in this study. The thing is here, you want to make it to be a healthy diet. So in as much as we know that you are increasing your health, you also want a stable diet. So when it comes to probiotic, it increased the richness and the evenness, the stability and the resilience, which are also very, very critical. There are shifts which are visible when you look at the communities, and then also you want to have a more compact study. So as I said, statistical significance increased as you increase the number of organisms. But remember that for 16S, which forms the major part of this study, you can only go to the genus. We still have some data we are analyzing on the metagenome, and which we are able to go to the strain level. Maybe that one I'll present in another forum. Thank you very much.
Kindly a round of applause for Josephat. We are going to take questions after all the presenters. Kindly note down your questions. I will invite them once again. Our next presenter is Mr. Daddy Helefu.
Daddy Helefu, can you hear us? You're
Yes,
able
I
to
can hear you.
Okay, great. Are you able to share your slides? Please go ahead and do so. Great.
Are
We
you
can
able
see
to
your.
see my slide?
Yes, yes. Please go ahead. You
Okay,
can also
thank
turn
you.
on your video if you're able to.
Oh, it's quite not possible because my video is inactive.
All right, carry on then.
Okay. So my presentation title is about the meta-barcoding assessment of algae symbionts diversity of corals under restoration in southern coast of Kenya. But my name is Daddy Mbwana Helefu. I'm a researcher working with Kwani University. I'm also MSc in bioinformatics. I'm a Tanzanian, by the way, working with National Institute for Medical Research. Starting with the introduction, these coral reefs are the vital marine ecosystem that supports biodiversity, fisheries, and livelihood. And due to rising of the sea temperature, they disrupt these coral symbionts. Due to the effect that happened with El Nino south oscillation, that causes bleaching and up to 95% of coral death in Western Indian Ocean. So recent studies show that the microbiome and genetic adaptations that may boost this coral resilience to these stresses. And Kenya have started establishing that for effective reef restoration and conservation, which is being done in Pwani University. Starting with the symbiosis cycle, these corals, they act with these holobiomes to work together for crucial disease resilience and distress. So actually they start with the spawning, fertilization, then with the accusation, and then settlement to the development. That will impact in metabolic efficiency and the thermotolerance. So if you disrupt the coral symbionts, you will lead to death of these corals, and which will also be impacted with bleaching due to effects that may happen. Normally, due to these heat waves and some human activities that are conducted in these oceans, so it leads us to find a gap to do the genetic diversity of these host tolerant families of Symbiotineaceae. So we come up to do this research on coral symbiont diversity. We actually wanted to assess the algae symbionts diversity of corals under restoration on the southern coast of Kenya. And then we had to do specifically to characterize these algae symbionts and then to evaluate the community composition of these algae symbionts between the coral species subjected to different experimental treatments. Where is it done? It was conducted at Shimoni, Kenya. We specifically go to collection site from Firefly, Lipipa, and Kikuyu House, and Mkwiro. The study design was a cross-sectional study. When we talk about the sample size, we only had 210 coral samples that were collected from the 10 coral species. You can appreciate their names here. These experiments. So different experiments has different treatments with the numbers of samples, which we were about to get only 210. The method, we did sampling DNA isolation, and we amplified it only on ITS2 because we are specifically going for algae symbionts. We went for library preparation and then sequencing using MySeq. And then we do the quality check. If they pass, it means we are going for downstream analysis, diversity, and community composition. In bioinformatics analysis, we started with the Sympoto framework. This is the good framework for this algae symbionts annotation. It's established, but it's well known that you can do annotation with this Sympoto framework. And it comes up to answer our objective 1 about diversity, and then objective 2 about the community composition. And also we were about to do it in coral hosts. We did composition and also clustering. And also about the community composition, now we had to do in treatment, interacted with these coral hosts. We had almost 4 treatments, and then we combined it with our statistical analysis. What we got is the Symbiodiniaceae as a family, community composition by these coral hosts. So we went down to get the Symbiodiniaceae profile that they were characterized in terms of colors. You can see the C105 up to C15 AFT. These are the profiles that were different in terms of color. They are green color, and also the C3 to C3Z. You can see their color is bluish, and also the D1 to D6 and others. These were the other Symbiodiniaceae profile. So what we got is almost the dominated C3Z, about 72.6%, but it was on the Acropora tenuis. You can see it from the figure there. The Acropora species and Stylophora pestillata species dominated also by Durustinium D1, respectively. All of them, they were dominated. So we had to also to see other species that were very low relative abundances, as you can see there, like the C310. You can see the amount is very low. And other rare symbionts contributed minimally. They were explained in others. For clustering, we clustered all the coral species. So the overall, we got Cladocopium dominated. And also we got some few points of the less sampled species. And statistically, the difference in community were unlikely to have occurred by chance. So community composition with species and treatment interaction with these algae symbionts, as we have seen here, the preaching responses, cradocopium was still dominating, and we have seen some not bleached colonies. They have some few Durustinium D1 and the low abundances of D1 UK. These are the symbiogeneous profile of Durustinium. In mineral accretions, actually we got Durustinium profile that were visually seen in Stylophora pistillata across all treatments that you can look on it. Going forward to other treatments like coral gardening. Coral gardening was overwhelmed with this cradocopium, and we went to tidal zones because we had intertidal to intertidal. We were changing the tidal environment from natural to intertidal, from natural to subtidal. And actually, the Acropora tenuis was dominated with cradocopium profile, particularly C3Z. And there is only a mixed community in natural intertidal. You can see there is a mixed in Acropora tenuis, the mixed community of this symbiogeneous profile. What we can conclude is cradocopium and Durustinium were predominant. So ecologically complementarity between this productivity optimized and the stress tolerant lineages. And there is tight clustering signals stable for symbiotic partnerships and broader dispersion. What we can recommend is a long-term multi-seasonal monitoring should be implemented. And also the future research should integrate this metagenomics with physiological performance metrics. Yeah, I want to acknowledge Pan University, the Revolution Association, Wageningen, and the National Institute for Medical Research, Tanzania. This is my contact. Thank you.
Thank you, Daddy. Our next presenter is Edwin Magomere. Put down your questions, those that will be directed to Daddy.
So as they set up, I want to make a disclaimer. This presentation has nothing to do with the bioinformatics, nothing to do with the AI, and nothing to do with genomics. But it's a preliminary towards doing some genomic works. And so yeah, okay, this one. So my name is Edwin Magomere, and I currently work at the Gitaka Lab as a postdoc. And I'm going to present on HIV-1 and HIV-2 interactions, reduces cell infectivity and viral replication in vitro. Okay. Yeah, so HIV still is a major burden, with currently about 40.8 million people who are currently living with the disease. And I don't know why it's going back. Probably there's a problem. So it is one, right? Okay. So if you look at the continent of Africa, it actually bears the largest burden. Out of 40 million people, about 26 reside on the continent of Africa. And if you look deeply, you'll see that out of the 26 that reside on the continent of Africa, 21 million people come from east and southern part of the continent, while only 5 million people come from the western part of the continent. And so the question that we are still trying to understand is that what is it that is at the interplay that is causing, the West African epidemic, being less severe compared to what we see in east and southern part of the continent. Wow, it's going back. I don't know why it's going back. Okay, let me go. Yes, so in literature, there are a few suggestions that could somehow explain what we see, the difference we see in terms of epidemics in the east and what we see in the western part of the continent. There is a proposition that host genetics, that people from east and south could have a predisposing genetic variations. We have behavioral factors, we have viral transmissibility, because if you look at the different viruses circulating in east and western part of the continent, the subtypes are quite different. And so there is also the issue of replication capacity, and it's possible that HIV type 2 is inhibiting HIV type 1, because in the western part of the continent, there is, in addition to HIV type 1, there is HIV type 2. And the West African epidemic, as I've mentioned, it has it's quite unique. It has both HIV type 1 and type 2, and the major circulating subtype is actually a recombinant form that is called CRF02AG. So that is also proposed to be one of possible reason why we could see the difference in and so when you look at the HIV is actually in the genus of lentivirus and family of Retroviridinae, and I want to emphasize that there are two major types, that is HIV type 1 and HIV type 2. And if you look at these different types, what you see is that type 1 seems to be a bit more diverse compared to type 2. In terms of the grouping, HIV type 1 has group M, group N, group O, and group P. And you could see that group M has other subtypes from A to about K. And within the subtypes, there is what we call recombinations, forming recombinant forms. I've given examples of the CRF02 and CRF01. And all these contribute to the different epidemics we have just seen both in east and western part of the continent. So if you compare the genome structure of HIV1 and 2, what you see is that they are quite similar, largely similar, with only two proteins that are different. We have protein VPX that is only present in HIV type 2, and protein VPU that is only present in HIV type 1. And if you look at the distribution of HIV1 and 2, what you see that is that HIV type 1 is distributed across the continent, but there is the epidemics in different parts of the continent are driven by different subtypes. You could see that in the southern part, you have majority being subtype C, about 98% is subtype C. When you come to the western part of the continent, you see there is a recombinant form CRF02AG, and to the east, we have, I think, C and D that are dominating the epidemics. So this is one proposition that is possible that they could be contributing to the differential distribution of the burden compared to HIV, compared to West, if you compare between West and East and as well as South. So if you look at HIV type 2, what you see is that it's quite restricted to the western part of the continent. It's not found in any other part of the continent apart from the western part of the continent. And so the rationale of this study is that from previous observational studies, we have seen that there is reduced disease progression or development when individuals are infected with HIV type 2. And also, there is also this natural protection that individuals are able to gain when they are infected with HIV type 2 and as well as HIV type 1. And this study showed that it was a long-term cohort study, about 19 years. People were followed up for about 19 years, and they saw that those individuals who had HIV type 1 alone, they progressed faster to the disease, to the symptomatic phase of the disease, compared to those individuals who had HIV dual infections, which means when you have both viruses, you take a bit longer time to progress to the asymptomatic phase of the disease. And here again, we we don't understand what is it that HIV type 2 is doing. How is it able to modify the disease projection of HIV type 1? And so we looked into literature and summarized what had been reported before. So we wrote this review paper and published it, and we were able to show a few findings. We saw that these individuals who have both HIV 1 and 2, usually they tend to present with an immune system that is already primed. So for an individual who has HIV type 2, his immune system is usually primed, and they are ready to, to respond to HIV type 1 when it comes in later. And then we also had issues with some protein, HIV type 2 proteins have been reported to limit HIV type 1 replication. So all this put together, we proposed that we need to also look at the replicative capacity of HIV type 1 when it coexists with HIV type 2. And to do that, we could measure the expression of unspliced HIV1 RNA as well as multiple spliced HIV RNA. That will help us to really understand how HIV type 1 is able to replicate in presence of HIV type 2, as well as when it is existing without HIV type 2 in monoinfections. So our first research question was, how does HIV2 influence HIV1 viral infectivity during dual infections? And the second is, how does HIV2 influence HIV1 transcription as well as splicing? That is during dual infections. So the main aim was to investigate interactions between HIV1 and HIV2 during dual infections. And the first objective was to assess viral infectivity in single as well as dual infections. And we looked at evaluating the effect of dual infection on HIV1 transcription as well as splicing. So the first objective to assess HIV infectivity. And so to do this, it's an in vitro experiment. So we have we have two cell lines that we used, U87, that is CCR5 positive, and TZMBL cells that are that are susceptible to infection with both HIV1 and 2. And we also have the medium we use is the complete DMEM, and a list of about 6 viruses that we used for these experiments. 4 of them are HIV type 1 and 2 are HIV type 2. So to do this, we first of all maintain our cells in culture. Then we infect them with either HIV type 1 or type 2 for a period of about 72 to 96 hours. Then we go ahead to harvest the viruses. After harvesting, again, you have to establish the TCID50, which is the concentration of the infective viral particles. So we went on to do a TCID50 experiment using this crystal violet staining. Then we went on to do to use luciferase assay to further quantify the viral concentration, and did ELISA assays again to just confirm that. So this is an experiment layout of the first experiment. So what you see there is a 96 well plate, and the first side that I've highlighted is where we did our single infections with either HIV1 or HIV2. Then in between there you see it's uninfected cells, that is a negative control. Then we went on to do dual infections, where now you introduce those viruses, at the same time, simultaneous dual infections. You introduce them at the same time. And then, so when you do that, what you see on the x-axis is the type of infection. We have HIV1, monoinfection, dual infection, and HIV2 monoinfections. And on the y-axis you have the relative light units, that is the signals for infection. So you could see the signals are higher when you have HIV monoinfections compared to dual infections. That was across, most of the subtypes, apart from that subtype, that is a recombinant form, which is actually the one dominating the West African epidemic. You could see that when we do dual infections, you don't see a lot of reduction. And so these experiments are one-time point measurements. So we wanted to confirm if you could, replicate, you could see this in culture over a period of time. So we repeated the same experiment, but now we harvest cells at different time points and measure the luciferase activity. And what we saw is that you see that for single infection, you will see high signals of infectivity, but for dual infections, the signals are a bit lower, and you could see that after 72 hours, you could see there's some sort of a reduction in infectivity signal. So that tells us that after 72 hours, the cells are almost dead, and that's why you see the signal is now coming down. But you could see that the reduction in infectivity in the dual infections was consistently lower in the dual infections. And so we then wanted to look at the effect if you do these dual infections in a sequential manner. You introduce HIV-1 first, then introduce HIV type 2, or you introduce HIV type 2 first, then introduce HIV type 1 first. So these are an experiment layout. In this first quadrant, we have single infections only. We have dual infections where you have HIV type 1 coming in first. Then here you have dual infections where you have HIV type 2 coming in first. So what we saw is that this was just a diagnostic PCR to confirm infectivity. So what we saw again is that there is reduction in infectivity for dual infections across all subtypes. And you could also see when you reverse the order, now you start with HIV type 1, that reduction disappears. So, sorry. Yeah, so the last objective, we're just looking at the transcription and splicing. So we basically, you extract RNA from the cells, that would be the HIV, cell-associated HIV RNA, as well as RNA from the supernatant. That will give you an estimate of viral load. So what has been released into the supernatant after dual and mono infections. So we quantified using qPCR, and what we saw is that when you quantify the viral load in the supernatant, again, during dual infections, there is reduced viral load compared to, mono infections. And just want to go to the last part. So here we looked at the simultaneous dual infections. Now we are looking at the cell-associated and splice HIV-1 RNA as well as multiply splice HIV-1 RNA. What you see here is a reduction, is a negative fold change. So relative to the mono infection, you see a reduction in expression levels of HIV and splice HIV RNA, as well as multiply splice HIV-1 RNA is reduced apart from subtype B. And I will explain that. The same for sequential infections, we see reduction apart from subtype B, and you could see what I'm highlighting there. Subtype B has been shown to be quite high, to have a high replicative capacity, and therefore it's possible that's why it's unable to, to reduce even when you put it in a dual infection. So basically, I mean, I rushed to the presentation, but what I've explained is that when you have dual infections, there's a possibility of the two interacting at infectivity at that stage of cell entry, and when they interact, possibly HIV2 could inhibiting HIV1 interaction with the receptors and reducing its ability to infect cells. And at the same time, if you look at the transcription stage, we see there is a reduction in levels of unspliced HIV RNA as well as multiple spliced HIV RNA, which could tell us that possibly there is a reduction in transcription as well as splicing, and both events would lead to lower viral loads. So this work has been published, and now going forward, we want to try and sequence the isolates from dual infections to see if we pick some recombinant forms, and just to see if that could also explain why we see reduced infectivity in dual infections. Thank you.
Thank you, Dr. Magomere. Our fourth presenter is Dr. Protas Omundi.
Good afternoon. As he shares my presentation, maybe I can use this time to introduce myself once again. So I'm Protas, a postdoc fellow in malaria epidemiology, genomics, and data science. So my work sits at the intersection between malaria epidemiology, genomics, and data science. And today, I would like to talk about some work that we've been doing here at the Center for Malaria Elimination here at KMKU locally. And that's why I've decided to say this is building a local capacity for malaria genomic surveillance in Africa. So I will use Plasmodium falciparum in our case, which we've been yes. So I will talk about this parasite called Plasmodium falciparum. And as you know, this parasite is complex. And it's not just a static, it doesn't have just a static genome. This genome changes as a result of various factors, including drug pressure, host immunity, interventions that we are using. We also have the vector ecology. We know the mosquitoes, they drive transmission, and also the transmission dynamics. So, this, our work, it provides a window into selection and how we can understand the population change for the parasites. So, looking at Africa as now where we are, we know we have a lot of biological data. And not just data, we have a lot of diversity. For instance, if you look at us here in Kenya, in the lake endemic region, we have high data for pathogen diversity. We have a lot of complex transmission systems. As we know, it's an endemic region. We also have a lot of host pathogen interaction, and more of evolutionary responses, and also the spatial data, because we always do surveillance when it comes to malaria. So this gives us an opportunity to build the capacity. And we know one of the challenges that we've had, which it's not now a major challenge, is the infrastructure. We know we have sequencing machines in Kenya. We have the next 1,000 here at MKU. And we also heard about the Novasec. So we need to use this opportunity to build computational resources, bioinformatics, expertise, large-scale integration, and more importantly, we need to utilize AI and machine learning capacity. So the sequencing African diversity, it gives us an opportunity, not just to collect data and generate data, but also to understand what this data tells us. So maybe we'll go to our case where we have a lot of, we always have challenges. And this usually starts from the field. We usually have very difficult samples because most of the individuals in an endemic region, we have very low parasitemia. So a lot of samples, they contain high human DNA, but low parasite DNA. So what we usually do is usually targeted, targeted PCR, where we're able to get the DNA, PCR or target PCR. Then we are able to sequence and, of course, analyze the data locally, not sending the sequences outside for analysis. So with this, we are not only able to generate like binary data, presence or absence of mutation, we are able to understand how common are these mutations, the different combinations, which are like the haplotypes, mixed infection, and also within an individual, we can able to tell the diversity using the deep sequencing, which we are now doing here in Kenya. So this transition is good because it gives us that opportunity to extract biological intelligence locally. And just more on our study that we did. So in this case, we had a total of 129 isolates. These were extracted from placental samples. And as you can see, 128 passed the sequencing and the downstream analysis. So because this was targeted, our question was very clear. We were more directly to these mutations, the markers. The PFDHFR is a marker that is associated with SP resistance. Those who know SP is very common, sulfadoxine pyrimethamine, and also the PFMDR, which is also a marker associated with the partner drug for ACTs. So what did our deep sequencing reveal beyond just simple mutation? Like I said, we are able to know all this, we are able to answer all these questions. Now, like I said, we were able to interrogate the sequences more deeper and not just understanding whether the mutant, whether it's mutant or wild. If you look at classic example here, this is a sample from one individual. You can see just a simple visualization. We are able to identify more than 27,000 reads at both loci. And all of them, they suggest there is that change of A to T, which is a mutation that is very relevant to antimalarial drugs. So deep interpretation, we are able to know even the minor variants. There are those variants that now are emerging. So with this technology, we are able to learn even more. Now, so this is more on that data. And as you can see, our data set shows a highly prevalent resistance for that DHFR gene and also the 184 for MDR. So 82% of the isolates carried the classical mutant, which contains 3 haplotypes. So we were able to identify those individuals who have different combinations of the mutation, and that's the one we are defining as haplotypes. These haplotypes are known to have different levels of resistance. And more, we were also able to identify hidden diversity. There is those markers that are now emerging and they are circulating. 164 is a very good example. It's very rare in Africa, but it's now emerging. And its combination with the triple mutants that I mentioned, now resistance is even high. So this deep sequencing exposes more on the genetic diversity rather than just the binary mutation calls. And now, our next step, we are now doing targeted. We want to move to whole genome sequencing. Targeted sequencing, of course, it answers focused questions. Plasmodium falciparum is not so big. It's just 23 MB, but it has a lot of complex information, that genome. We need to sequence the whole of this parasite. And because of the challenge that I mentioned, for instance, I said the DNA we got from the placenta has got more than even 99% of human DNA. You extract DNA, you find very good concentration, but the parasite DNA is very low. So we have come up with a technique that we borrowed from colleagues from London. And this low parastemia issue, we are using a selective whole genome amplification using the Equify 29 parasite. This one, we're able to enrich the parasite DNA because it has primer specific to the parasite, but it's not targeted. It's more of a long range PCR, so it's able to generate very long reads of the parasite. Then from there, we move to genome-wide sequencing, and of course, we infer on a population scale. So mostly we deal with huge number of samples. So you can imagine like this study, we are focusing more than 100 samples. Whole genome for 100 samples, I think it's not easy. So we are able to answer many questions, like I mentioned. And one important, we will be able to even know the selection and the evolution of these parasites as we move towards elimination. So now this is where we are. So as we move to huge data sets, like I've mentioned, we also have a lot of challenges which will apply basically to our data sets. it's going to be huge data, so we're going to have a lot of analytical challenge. And generating this data, like we heard from the previous presentation, we need these AI models, machine learning tools, to be able to generate resistant prediction, population structure. We can be able even to map all the parasite population that are circulating in Kenya. For instance, I've heard in Wajir, now Vivax is very common. For those who know the implication of Vivax, you know we need to do something. So we can be able also to know the emerging variants. We have those parasites that are known to be like the dominant ones. When they are emerging, if you're able to track them, the interventions will be more directly and also more impactful compared to the way we usually approach without this information. So the goal is we need to generate data locally, we need to generate biological intelligence locally, and also we need to make those decisions because they apply to us. I think that marks the end of my presentation. Thank you very much.
Thank you, Protas. All the young scientists, you're asked to kindly come at the front with a seat, take questions from the listeners. So you can lift up your hand if you have a question.
Good afternoon. Thank you so much, young researchers, for the wonderful presentations. My question goes to Dr. Josephat. Thank you for the insightful presentation on probiotic and the potential it has for a lot of things. I got a bit curious about Streptococcus, which you said has a preference for upper gut. Have you found something about whether it could limit the growth of H. pylori or other pathogens in the upper gut?
Oh, hello. Streptococcus, actually there is a lot of studies which has shown that it is good on the mouth flora. When you want to address gum diseases, it's a potential thing. On H. pylori, I haven't seen a study on H. pylori, but I know H. pylori, one of the things is that it destroys the gut, and you need a lot of especially the bifidobacterium is going to address that one more. Thank you.
Thank you. My name is Lorin Onyango from University of Nairobi. My question goes to Dr. Magomere. You mentioned that people from western part of the continent are experiencing HIV type 2. Why so? Why not type 1? And does it mean they also experience dual infection?
Thank you. I didn't get the last part of your question.
Do you think they also experience dual infection for type 1 and 2?
Okay, so thank you. So yes, I say that HIV2 is circulating in West Africa as well as HIV type 1. So both of them are there. And yes, we have samples of people who have both HIV 1 and 2. So one can have both infections. Usually HIV type 2 is more of asymptomatic. You don't see the symptoms. So they're like, even the viral load usually it's very low. So someone can be carrying type 2, but they're not aware. They end up getting type 1 on top of that. Yeah, and one thing that has been observed is that those who have both 1 and 2 tend to have a slow disease, a disease that is progressing more slowly than the people with type 1 alone. And that was one of the questions we are trying to answer in our study. Why is it that when you have 2 viruses causing the same disease, we were expecting that you would have a more severe disease, right? But then you see the opposite. So yeah, we have people who have both viruses.
Okay, so we'll take another 3 questions and then you guys can answer.
Thank you. My question goes to Dr. Plotters. You've done a presentation or linking what you have currently, and then we are integrating AI and and the impact of AI in sequencing. So currently, it takes a very short time, for example, to detect resistance of antimalarials. Yet it takes a long time for government or the health ministry to take an action. So what do you think should be done to bridge that gap from what we get from the results or from the lab, for example, drug resistance, and the implementation or what should be done in the field to deal with that problem? Thank you.
Bramwell, you're online. Unmute yourself and ask your question. Bramwell Wanjala.
Okay, thank you, colleagues, for the nice presentations. My name is Bramwell Wanjala from Calro Biotechnology. I have a question to Joseph on probiotics. I think there are several indices that determine weight apart from BMI. Did you consider that? And the other thing is a comment. In the wake of people getting health conscious, there is other fermented drinks like kefir and kombucha. What will be your take on that? Thank you.
My question is to Josephat again about the probiotic and the conventional yogurt. Right now we have a lot of CA colon. What is any relation between the probiotic yogurt, because you've talked of its immunological benefit? So what is the relation with the CA colon? Secondly, now there are a lot of companies that are manufacturing yogurt. So how, and most of the companies will label as a probiotic. So when purchasing this yogurt, how sure should we be that for sure it's a probiotic yogurt and it's not a conventional yogurt? Thank you.
One more question.
My name is Sam Momani from Kenya Institute of Primate Research. So my question goes to Josephat too. What is the rationale of using 100 ml of the probiotic yogurt? And if I got it right, why did you decide to collect the samples during day 12? Thank you.
Okay, last one, and then we'll not take more questions. 2 more. 2 last ones. I hope you're noting them.
So my name is Gishane Maina and my question is to Edwin. So do you think for a patient with type 1 HIV, it would be beneficial to get infected with the HIV type 2? Would it lower the virulence?
Thank you, Sifuna Martin. My question was to Dr. Magomere, but the question has just been asked because I was also thinking of him giving a conclusion or some advice or some suggestions and recommendations for the future about the use of type 2 to counter type 1. So I remember we mentioned this aspect during his first presentation, and I thought that maybe he will be able to clarify on it on the subsequent presentations, but he was not able to bring it out clearly today. So my question now is the repeat of what has just been said. Do we take type 2 as a preventive measure for type 1, which is more virulent and more dangerous? Thank you.
Thank you.
All right. Over to you to answer the questions.
Let me go first. So I'll try to address all the questions, but in no specific order. I'll start with the reason of 100 ml. there was no rationale, but we assume that 100 ml per daily consumption every day is supposed to have a benefit. We know that also this was just one of the interventions. We couldn't account for things like their lifestyle, their diet, which will definitely also affect the analysis. But that we were able to filter out when we were doing the analysis, whereby we do the non-metric dimension scaling when you're doing the principal coordinate analysis, and you're able to see things like the stress level was very low. I didn't mention about that in the results. So it meant that whatever you are able to see is as a result of what the microorganism is doing only, because we were able to account for the others in the analysis. But it doesn't mean that only diet is accountable for weight gain. The other thing is that the other question was on the colon. Yes, Bifidobacterium really enriches ruminococci, and most of those, what are they? The fibers, and you're able to ferment, and once you ferment those ones in the stomach, you're also able to get some more nutrients from that food. Otherwise, it would have gone to waste. So the more you have them in the stomach, and it passes the stomach to the gut, it really helps to bring the health benefit. But remember that at this point, you're not looking at the health benefit as such. You are looking at just the benefit of enriching other organisms. And that's why in the last slide, I mentioned about the real life and what the statistics show. So when statistics says that this is what you expect and it is significant, the P is 0. But when you look at the effect change, it's still minimal. You cannot make a conclusive conclusion to say that when you take probiotic yogurt, you are able to enrich your gut. And then, yes, I know, especially in our market here, you find a lot of products called probiotics. Kombucha actually is probiotic. It depends with what the definition of yogurt says. Yogurt basically should have Streptococcus, Thermophilus, and Lactobacillus bulgaricus. So if those two are present, you're not supposed to call it probiotic. It's just normal yogurt. Otherwise, you will call it something else. So, but when you find that only those two organisms, you call it probiotic, that's commercial fraud. So I wouldn't advise on the brands and anything, but I know there are those brands which they will just have those two organisms, but they call it probiotic. It's just about consumption, consumer awareness and consumer, education. Thank you.
Okay, even though the Q&A session is done, Lynette was talking about cancer of the colon.
Yes, we just looked at the overweight. We were looking at weight, and this study was part of. It was quite a big study. It's funny that when I was just about to move, that's when the slide went down. So there was another one slide which wasn't on the slide. Yes, but we never looked at cancer of the colon. But what we did, we collected also blood samples. We did their weight. We did a lot of other things on the health sciences. But those ones I wasn't able to really show here because it was a big study. And on the cancer, I know this was something that we were discussing about. If we realize that someone had cancer, what do we do as a researcher when it comes to ethics? Because there are so many things that we saw on those, but we cannot present most of them in this forum. Thank you.
Thank you very much, young scientist. Oh, there's still some more questions? Okay.
Dr. Sifuna asked whether we should infect people with HIV type 2. the kind of data I presented here does not allow us to make that conclusion, but we have additional data that we used clinical samples. We collected samples from people who have actually HIV 1 and 2, and we've done a lot of immunology assays, and we've seen here, those people who have both infections have some they present some immune markers that we selected for about 2 of them that we are still working on to validate that. And so, yeah, it's good. We can learn lessons from HIV type 2 and apply them to HIV type 1, but we cannot infect people with HIV type 2. It's not ethical. So one way we can do is to look at how HIV type 2 primes immune responses. And then we could go on and elicit similar immune responses in individuals, even before they're infected with HIV type 1, so that they have that protection advantage, but not really using a live virus to infect people. Yeah, or maybe you can develop a vaccine that mimics HIV type 2, which could be injected and give that immune responses, immune response profiles that would protect them from getting HIV type 1. Yeah, I think that's the best way I can answer that question. Thank you.
Okay, I will also go for mine. So thank you for that question, and I think it's very important because I've had it even before. So I will use a quick example to answer you. So there is a time we did a presentation on the progress of malaria in Homa Bay, and we did the presentation to the CEC of Health. We presented very interesting results, very complex results. But when it came to the comments, she told us that my boss is a politician. How will I go and tell her what you've just presented here? So what I'm trying to say is, as scientists, we also have a role of communication. We need to communicate to stakeholders, and we communicate in a way that they will understand the impact of the results that we have. A good example is the Mamodex project that we are doing. We are trying to model the currently available interventions so that we can advise the policymakers and the implementers which interventions work well, where, and when. So I think that's a very good question. Thank you.
Okay. I think they have done a really good job in answering those questions. Give them a round of applause. We are now quickly transitioning into the next session, headed by Dr. Sarah Naulika Kitui.
Thank you very much, Dr. Rachel, for the introduction. Yes, my name is Dr. Sarah Kitui. I'm a researcher here at Mount Kenya University, and I dabble as a lecturer at the University of Embu. I'll be your session chair for this 4th and last session of the day on industry partnerships. Now, throughout the day, we've had some very good insights from, of course, all the presenters on the transformative role of artificial intelligence, biodiversity genomics, and bioinformatics. And this session will now bring us to a new dimension of transformation, where we're exploring the role of industry partners and just how they convert research knowledge into scalable solutions that are sustainable for Africa. So we're privileged to have about 4 industry partners for this session. And during this engagement, I'll urge each one of us to not only listen to those solutions and the technologies that they have, but also open yourself to opportunities for collaboration, especially with regard to support for research, access to technology, and just how we can collectively move together to enhance research by Africa for Africa. So I'll be keen on time as well. So I'll have our first presenter from Inkaba, East Africa. If the presenter is here, please move forward and take the stage. Inkaba has been very pivotal in biotechnology and genomics, and they'll be speaking to us about just what they do. Thank you.
Good afternoon once more. Yeah, it has been a very good day since morning. One thing that I noticed that is not very common in all other workshops like this. You had a blessing of having 2 DVCs here this morning and the VC. It's not that easy. Mount Kenya, you guys are very lucky, and please take advantage of that and do more and more research, because you'll be supported all the way. I also want to take this opportunity to recognize Profan. Profan is actually my personal mentor. I was in class in 2006, that's 20 years ago, where Prof. Ann was my lecturer. Prof, this is what I became. Yeah. Yeah, so, okay. So this is the easiest session and also the most important one. I'm going to be very brief on this one. This is the business side of the work that you're doing. We are the guys who provide the reagents. We are the guys who provide the equipment that you use and also the services. I'm going to introduce myself as William Nyakundi. I'm the Managing Director for Inkaba Biotech, East and Central Africa. Inkaba Biotech started back in the year 2000. That's when the idea was born. It was born by a scientist who came from Switzerland to do his postdoc in South Africa. But he faced a lot of challenges in getting reagents and also services. So he thought about it, went back to Switzerland, talked to the Swiss government and other friends, and came up with the idea and started Inkaba Biotech. So Inkaba is derived from, is it Kosa? Kosa name, meaning rare and precious, which is South African. This one. The next slide. Yeah, over the years, from 2002, in the year 2010, the first subsidiary was started, which is Inkaba Biotech East Africa, which is based in Riverside, just here in Westlands. And in the year 2015, we had the Inkaba Biotech West Africa. From these 2 subsidiaries, we have now fully operational branch services in Uganda, Tanzania, and we also have representation up to the Congo. Also, another thing I should mention is when you guys are attending this African BioGenome Project, just know that this is a very, very, very big workshop which is going on. When I went to Congo for the first time, as I was going to open an office there, I had them speak about Africa BioGenome. I was shocked. So you guys, the work that you're presenting here, it's the best. And also where this work is being published is the best of the best journals. The next slide. So what we do basically as Inkaba Biotech, we are a distributor company. We go to the manufacturers, most of them who are based in Europe. We go to them, get contracts with them, and then we come and supply their products on their behalf right here in the African soil. Why are we very so important in the distributorship business? For example, you've bought a kit, let's say for example an extraction kit, and it doesn't work for you. Probably it's faulty, or maybe you didn't understand how to use the kit. We are there as representation in the country to visit your lab and work with you so that you can get better results from the item that you purchased. So if the item is faulty, which in some cases, rare cases, it usually happens, when you have local representation here, we are able to exchange the kit for you at no cost at all, rather than you buying directly from Europe, whereby you don't know whether you'll be able to get a replacement of the faulty kit also. So these are the major brands that we present as Inkaba Biotech. We love yellow so much, yellow and green. I think you've seen it outside there. So Zymo Research is the brand which deals with extraction kits that we supply in the local market, direct competitor to QIAGEN. We also have PacBio, which is for sequencing. We have the latest technologies in the Vega, the benchtop Hi-Fi sequencer. And we also have Eppendorf, ProteinTech for your proteins, Glentham for your biochemicals, Quantum for your protein sequencers, Seegene for molecular diagnostics, New England Biolabs for your day-to-day molecular biology reagents. With New England Biolabs, we can work with ONT, we can work with Illumina, which we don't supply ourselves, but our friends and colleagues here who are also distributors, they supply those machines. We also have, we're also into plant diagnostics, whereby we supply for a company called BioRiba. How do you know that Inkaba Biotech is the one which has been subcontracted to do this business in Africa? We usually have, the next slide. We have contracts. We sign contracts. These contracts are yearly and we follow them to the letter. We are not supposed to do anything outside what we've agreed with the manufacturers. Talking about services, these are the major core services that we offer. We do bioinformatics. We have bioinformaticians who will look into your data and interpret it for you. Next generation sequencing services. We have Sanger sequencing services. Animal genetics. We also do animal genetics. For those of you who love dogs, German Shepherds, Caucasian Shepherds, if you want to know probably the color of your puppies, the DNA paternity of your dogs, cows, we do all that. Currently, we are working with a very big cattle farm here in Kenya, whereby we're doing this service for them. Oligosynthesis, this is the major thing that we started with in the year 2002. So I think we are the only company producing oligonucleotides here in Africa. Erie bought the machine, but they are not yet to operationalize it. It's not easy, it's a bit expensive, but we are proud to say that your oligos, as much as you order oligos and it takes up to 2 weeks to get to you, the oligos are produced overnight. It's an overnight thing. The only thing that makes it to be delivered within a week or two is the logistics and also the Kenyan custom laws. The next slide, please. Yeah. Recently, 2 years ago, we started a lab. We have a molecular lab, fully operational. We're doing sequencing there, we're doing trainings there, and we accept each and every one of you. If you don't have space here where you're working at currently, you can visit us in our labs here in Westlands, book an appointment, book space. If you're giving us work to do for you, come see how we're doing it so that you're able also to defend your thesis when the time comes. One thing you may agree with me is the sequencing bit, not everyone knows the protocol on how this is done, because mostly you just send and we send results and you're okay with it. Most of the time you can get away with it in defense, depending on the panel that you have, but it's good that you know how all this is being done. So not only in Kenya we have labs, we have a major lab in South Africa. It's like a reference lab for us. Whenever we want anything, they can always, if we are unable to do something here, we can always send it there. We also have another big lab in West Africa, which whereby we are also doing the same thing. We are just doing the same thing. We are not reinventing the wheel. That's training and also offering services for those people who cannot afford the sequencers. Next slide. Yeah, this is part of the capacity building that we are doing. These are the recent training which is ongoing in West Africa under the African Molecular Research Excellence Fellowship, whereby we have, currently we've trained around 142 since, that was since March, started in March, but our target is to do 500 and more. So this will also be done here in Kenya as well. And when that time comes, we are going to share with you so that you guys can participate as well. Yeah, we've been lucky. We've been with African BioGenome Project from the time it started. These are some of the students. You can see the faces. Once you come to our labs there, you'll always be smiling because the environment is good. Everything there is good. Yeah. So we usually take these photos and document them. And I'm looking forward to some of you also visiting us in our lab there. Next. Okay. Apart from distributorship and everything that we do, in Inkaba Biotech, we don't forget the communities. You know, in business, you make profits. But what do you do with the profits that you make? We give back to the society. And how do we give back? We have an Inkaba Biotech smart farm. This idea basically, first, it's being done in South Africa, but it's going to be implemented as well in East Africa and also in West Africa. We have eggs. I'm not going to explain the price. It's in rands, but this is what we do. We have all types of crops that we've grown. If you can go to the next slide, you'll be able to see, those are spinach, yeah? When did you last see a spinach the size of your arm, the leaf? So this is being done in a way that we are using technology to make sure that we give the best and also be the best in the market. Apart from that as well, just here in Kibra, there was a lot of insecurity. So Dr. Kalebi, you know Dr. Kalebi? We had a discussion with Dr. Kalebi. He had started a football team which comprises only of criminals by that time. Yeah, so there was a lot of insecurity in Kibra, and Dr. Kalebi thought about how am I going to make these areas safe? So he came up with a team, started a team, and we are actually the official sponsor. This is called Gogo Boys, for those of you who know Gogo Boys. We are the official sponsors of them, and also the other sponsors as well, like Red Cross as well, they sponsor the team. But we have been able to reduce the levels of criminals in those regions. Last but not least, this slide is a very special one. In PacBio, apart from being distributors, getting items from Europe, bringing them and supplying them on behalf of someone, we identified a gap whereby during the COVID season, we had problems getting even tips for us to do our COVID testing here. So in PacBio has decided to start manufacturing of plastics for molecular biology grade. This is not yet open. It will be open towards the end of the year. That's December 2026. They are still finalizing on the setup and everything. So we expect to get our first batch to try out, and we're going to share. With this initiative, we expect the price of plastics to go down, because what makes your plastics expensive is the duties and freight charges. When you fly plastics, it's expensive. It's more expensive than when you ship them via the sea. But via the sea, you have to wait for 60 days for the plastics to arrive. So that's the difference that we have. And also, as I finalize, the last slide is basically next generation sequencing. I know you've heard of Illumina. My good friend here is also, you work with Illumina, yeah? She works with Illumina. We are competitors, yeah, but we are selling this technology. This is the best benchtop sequencer that we have, and the most affordable. Why do I mean the best that we have? Let's go to the next slide. I'm not going to talk science. I'm just going to mention just a few things here and there. Okay, the next slide, please. You can see the size of that. So actually this machine is only, the height is 76.8 centimeters, the width being, that's 55 centimeters. That's almost slightly more than a ruler. So it's a benchtop. I know some of you prefer ONT, but what this machine can do, it's much more than what ONT can do. So if you need more information regarding this machine, please visit our stand. As businessmen, we prefer engaging on emails rather than me answering questions that you bring up, because I want to track so that I can follow you up even after 2 or 3 months. Okay. So, and also another thing you should know is there is nothing which has a fixed price. There is nothing which has a fixed price. So what you do, you have budgets, yeah? You have budgets. I can tell you, let's say, for example, I tell you this machine probably is, let's say $400,000 or $200,000, and you don't have that money at once. We can always discuss. Yeah, I can give you even a payment plan of 36 months as long as you keep on paying on a monthly basis. Yeah, we can always agree. Yeah, for your reagents, feel free. My email address, I gave you my email address there. The company email address is there. I'm also available on WhatsApp. You can reach me anytime. Visit our stand outside there, and let's push science ahead. Thank you very much.
Thank you so much, my friend, for speaking just within time. I appreciate that, and I appreciate the fact that we can always talk about the prize. Great. At the end of the session, please stay around. We might have a question or two for you after all the partner presenters have spoken. We now move on to our next industry partner, IKA. Please, if the representative is here. Yes, kindly. We'll appreciate if you can take maybe 10 minutes and then spare some time because we're closing today.
I'll do you a favor, I'll take 5.
Cool.
Well, I've heard about complex equipments here, genomic sequencing, LSMS and stuff like that. But I think there's a gap when it comes to research, and that is sample preparation. So before you get to downstream analysis, how you collect your sample, how you process it, how you homogenize it, having proper equipment is definitely a key factor. So I want to introduce you to ICA, or rather ICA Works Kenya. It's a German manufacturer of lab equipment based in Germany. And as you can see, we've got subsidiaries all over the world trying to support researchers with sample prep solutions. So maybe I can highlight those and we can move forward. So next slide, please. Next slide. Yep, I think you guys have seen that amazing poster over there, something that looks more of a pen. And that's one of the major technologies that are coming out of ICA Works. And it's an electronic pipette. And I know we're all used to using mechanical pipettes, and sometimes accuracy is always an issue. But I think ICA has decided to go ahead and introduce something that looks like a pen, something that is more personal, something that is more automated. So this is one of the slimmest pipettes that you can see in the market. It's automated with a couple of modes. So if we can move to the next slide. Oh, I see. Yep. Next slide, where we have the features for the modes. So as you can see, this is quite a pen with us discussing about how AI is coming into automating equipments. You have a pipette that can consolidate many processes that previously were not possible. So you can be able to do reverse pipetting. You can also be able to do multi-dispensing, which normally we use the multi-channel pipette. You can also be able to do dilution. And if you're not interested in using the electrical programming, you can also set it to manual pipetting. So this is a very tiny device that comes packed with so many features. And if you can go to the other slide, we can talk about the price. But let's talk about the accessories first. So it comes with a stand. So when you're not using it, you can place it on your stand. It also comes with a wireless controller, because you can also connect your app. There's an app for this ICA pen, which you can connect it with your pipette, and you can be able to multi-dispense and also engage in all those modes by using the software. And it's coming at a very quiet price. As you can see, it's coming at the price of a mechanical pipette. Some brands I know, I don't want to mention them, they really come quite pricey, as as Nkaba would tell you, but we are really trying to also avail accessible and affordable solutions to the African researchers, because I believe Africa's scientific discovery is based most importantly on the lab infrastructure. So if we build our infrastructure, making it affordable, then again, African pricing. So let's go to the other slide, please. Yeah, so again, another important device that is coming out of ICA for sample preparation is a handheld homogenizer. So some samples, such as plant samples or animal tissues, cannot be processed the way they are. They have to be maybe homogenized or grinded. So now we are providing a solution that is more mobile friendly rather. So this is a T10 handheld battery homogenizer, which when charged can give you 24 hours of working time. So as you can see, it's also compact, so it doesn't take much space in the lab. It's also compact, so you can also fix it in a fume hood in case you're dealing with the sensitive samples. And we can go to the next slide so we can discuss about some of these dispersing element. So you get two options. One is disposable, just to avoid cross-contamination. And the other one is autoclavable, so in case price again is an issue, and you don't suffer from issues regarding contamination. You can use the reusable stainless steel disperser. And you can see most homogenizers out here are quite large, the ones you mostly see in industrial chemistry labs or analytical labs, but we have an equipment that can be able to homogenize samples in very small tubes. So this makes it easier for a scientist to just be able to prepare the samples properly. Another technology that is coming out of ICA, probably released this year, is the cooling and shaking incubator. So what we are doing here is we're consolidating a couple of processes, putting them into one equipment. So you can be able to shake, you can be able to cool, and you can also be able to incubate your samples all in one device, making it easier for the, for the scientists to be able to create a reproducible process, which makes it easier in terms of saving time and productivity. ICA is also well known for these shakers. These are not new equipments or new technologies. Incaba has been probably supplying the Vortex 2 for quite a long time in Africa, but we've got a wide array of shakers, including some that can also be able to handle well plates, as well as others that can be able to also heat and cool as well. So these are just some of the shakers in our portfolio, and yeah. Most importantly is a bioreactor. I'm not sure if this technology has really immersed here itself in African laboratories, but for people who are really interested in cultivations of bacteria, cells, and other biological microorganisms, this is a bioreactor, rather a benchtop bioreactor, that makes it easier for the scientists to be able to create a reproducible biological process. Instead of only creating or growing a cell, the scientists can be able to monitor certain processes, such as the amount of oxygen that is getting into their samples, the pH, as well as also shaking or agitating. So therefore it gives the scientists more control over the entire process rather than the outcome. So that's much of the new technologies that are coming out of ICA, from liquid dispensing all the way to incubation. And the goal of ICA Africa is to also avail these equipments to researchers. So we are based here in Kenya, in Westland. We also have a lab, similar to what Incaba has. And in case you guys are interested in visiting us, having some sort of application consultation, or maybe even like doing some demonstrations of our products, the goal for ICA is just not to be an equipment seller, but rather a partner when it comes to, providing these solutions to African scientists. These are so many solutions that are more technology focused rather than what we are used to, and we're making it easier for scientists to be able to have these solutions. So with that few remarks, I want to say thank you.
Thank you very much for being so precise with time. I really appreciate that. We're moving with speed. I must apologize for having skipped Exela Biotechnologies. I think they were meant to come after Incaba, but I didn't have the program with me, so kindly let's have the representative from Exela Biotechnologies coming over. Thank you, my friend Kaleche. The floor is all yours.
Good evening. What is it? Good afternoon. Good evening. My name is Christine Kaleche. I'm an application specialist at Exela Biotechnologies Limited. So Exela Biotechnologies Limited is a biotech company. We supply life science products. We are based in Westlands, Nairobi, next to Sarit. We have a regional presence in Kenya and the rest of East Africa, Uganda, Tanzania, Ethiopia. Our product portfolio spans from molecular biology products, ELISA kits, molecular oncology for cancer testing and screening, centrifugation, biosafety cabinets, cold chain, that's freezers and refrigerators, and lab consumables. So we partner with our world-class manufacturers such as Altona Diagnostics, Beckman, Hamoy Diagnostics, Biocatis, Panadea, RBC Bioscience, Sadit, and ESCO. We talk more about each as we go on. So in Africa, there's so much genomics opportunity like human genomics, pathogen genomics, other genomics, precision medicine, One Health surveillance. The main issue, the main problem that we have in Africa, it's the infrastructure challenge across the chain, from sample collection, preservation, all the way to translating it into impact. So for us at Exela, we know that genomics is not just a sequencing problem, it is an ecosystem problem. So next, that's why as Exela, we come in with an integrated approach to enable the genomics ecosystem. So our partners, BioCartis, we have fully automated molecular testing. We talk about it more as we go. Is a company in Belgium for precision medicine. RBC Biosciences is a company that manufactures automation, machines for automated nucleic acid extraction, also manual kits from Taiwan. Amway Diagnostic, a company from Singapore for comprehensive cancer mutation detection. SADET consumables, Beckman Coulter, for all of us who are familiar with centrifuges from Beckman, and also flow cytometers. And we'll talk more about this banner here as we go on. So BioCartis makes molecular testing accessible from SABU to research. So we have this machine, it's called IDELA. So, we push in more into precision medicine, whereby we're moving from like one size fits all approach to now looking at a person's specific genome. And so looking into that and then doing targeted therapy for several cancers, lung cancer, thyroid, blood cancers, breast cancer, brain cancers. And now for just those mutations that you can see, for example, EGFR testing has been very revolutionizing when it comes to lung cancer. If a patient tests positive for EGFR, now the clinician is able to prescribe, targeted therapy for that. So this machine, next, is a compact, closed system that allows for, it is a PCR machine actually. Just put a sample, which now it's very versatile when it comes to samples, FFPE, blood samples, stool, urine. Just put a sample in a cartridge in the machine. It does all the PCR work for you, and then you're able to get the results in under 3 hours. On Beckman Coulter, I've talked about centrifugation, for that all of us who've worked in most of these big labs, Allegra, centrifuges, and I know most of us who have worked in genomics, we've used Ampure. With the people who are distributing Ampure for cleanup, nucleic acid extraction support, and also for RNA also in size selection products. On Beckman, let me talk also on automation. We also have equipment for automating liquid handling. Next. For RBC Biosciences, we have these machines for automated nucleic acid extraction. We have MagCo machine that is able to run 16 samples with MagCo EDA, run 48 samples. And then we have MagCo Giga, which is a new machine with the liquid biopsy testing, especially for cancer. We need to, the tumor cells less in blood. So it allows for extracting from 8 ml of blood. So now enabling automation makes sure that our genomic data is reproducible, is held, and can be traced. For the MagCo machines, we have the different automation kits, which use with the cartridges, depending on the type of sample that you have. And also for the students, we've been very flexible to provide kits which are very affordable, pocket friendly, and this spans from DNA extraction, RNA extraction, and such. So if you're a student and you have projects going on, you can talk to me. So we also partner with Altona Diagnostics. For those of us who read more about the ongoing Ebola We have a kit called the PhyloScreen, RealStar PhyloScreen. PhyloScreen, it's been approved by the WHO for screening, and even recently it's been listed for emergency use. It's called the RealStar PhyloScreen, and also we have the RealStar Phylotype for typing the Ebola. Next. So for Hatona, it's basically a PCR kit for respiratory infections. I want to like go through the whole list. Next, for fever, tropical and emerging infection, supporting dengue, malaria testing, hantavirus, chikungunya, all that. Next, and also for GIT infections. Our kit come with internal control to ensure that the whole PCR process works. Next. So for sample, we know that once you mess at the sample collection, you are done for. No amount of optimizing in the extraction, in the PCR, NGS, anything downstream. Once you mess from sample extraction, you're done for. So that's why we offer quality consumables, basically from even the pipette type, pipette tips, blood collection systems for disposal, PCR plates, and all that. So if you students and big labs, talk to me, very price friendly. Again, the infrastructure around genomics really has also to be physical. We also provide equipment from our partners, ESCO. ESCO is a company that manufactures lab freezers and refrigerators and biological safety cabinets, laminar flows, depending on the size of your lab. Also incubators and centrifuges and all that, just so to ensure the whole chain from biosafety, cold chain, sample storage, contamination control, all that contribute now to sustainable DSI. Us as Exela, we not only just supply equipment, solutions for genomics, we build a sustainable genomic ecosystem through multidisciplinary collaborations. Through academic partnership, we partner with the research institutions. For example, JQuat, we've recently collaborated with the JQuat to train students on extraction, PCR and such, and also Aga Khan University Hospital. A clinical integration, we work closely with hospitals, Aga Khan University Hospital, KUTRH, Nairobi West, MTRH, just to support the clinical diagnostics. We partner also with other partners in the industry. For example, we partnering with Kipre and Aga Khan to establish a center of excellence for flow cytometer. Also, we engage much in public health engagement by supporting national surveillance with the National Public Health Lab. We supporting also testing for Ebola across the region and supporting mobile response. So we have other products that I did not mention because I need to focus on the genomics. So in case you have any question, please find me at the booth. Thank you.
Thank you very much, my friend Kaleche, for precisely talking about what you do at Exela Biotechnologies. We really appreciate, especially you parting shot on the nature of collaborations that you support. We're now moving on to the last industry partner for the day, and that will be Mocha Limited or Mocha Limited. Paul was meant to be online, so Paul, if you could hear us, please. Paul? Right, looks like Paul is not available, so that then brings us to the end of the speaker sessions. We'll now allow for some engagement with our industry partners. They've basically been showing us what they do, what technological access they have, and this now gives us the opportunity to ask them questions, especially in the wake of the nature of collaborations that we could look forward to, the nature of research opportunities, the nature of technology access. This is the opportunity to speak to them. Perhaps just a quick one from me. Kalechi has given us a take into the nature of research collaborations that they are fostering with a couple of institutions, including JQuat, which is an academic institution. From my end, I dabble as an academic and researcher, and for a long time, people in academia have always been mistaken for being in pursuit of funding, and most partners in industry will not be so happy to listen to conversations about collaborations from people from academia, because they think we'll always be talking of how can you fund us. Now, thanks to support from various partners, of course, academia or academic institutions are no longer just talking about how you can support us. Here at Mount Kenya University, we actually have a very elaborate research infrastructure. We're doing good research, and we're open to collaborations. So my question is, what nature or what model of collaborations are most welcome to our industry partners? What exactly are you looking for in terms of the nature of collaborations that you want to foster with academic institutions, so that then we can come to you with the right strings of collaborations? We actually, the centers of knowledge generation, we have the infrastructure. What should we add on for us to pass as good collaborators? This goes to all our industry collaborators, INCABA, Excella, and IKA.
Good. So we start with Excella.
Yeah, sure. Thanks, Dr. Sarah. For us at Excella, when we talk about good partnerships, we're looking into our target client in the market. You know, we are a commercial company, other than like not just doing partnership for the sake of it. So we partner with hospitals. Our targets get the clinical people, the lab technologists, researchers, both in the universities, research institutions like Cambridge, and all that. Before we even talk business, we have talks. And what we want from our partnership is to be sustainable. Sustainable in the fact that we've put down, like, this is the path we're going to take on this project. We as Excella come in now to supply, to make it happen. We want to establish a lab. We come in as the suppliers to make sure that we've provided every equipment, every solution that you want, and not just like selling the equipment, but also training, that is capacity building, and also making sure that we do servicing in time, machines are not breaking. So what we need, and especially from the institutions that we work with, it's the commitment. The commitment, and to make sure that whatever we're doing is sustainable. Yes.
Thank you. We go to INCABA.
Okay, this is where you expect the answers to be the same, but I'm going to speak on the perspective of INCABA Biotech. When we talk of collaborations, we are looking at the whole range. Let's say, for example, it depends with the partner who we want to collaborate with. If it's a research institution, let's say the university is different from, let's say, a research institution like IRI. So at the university level, I expect you to negotiate on the pricing. So in terms of collaboration is when we are having collaboration is when we're talking from sample collection to post PCR. So once we negotiate and we agree on a certain price, that price remains specifically for that institution because we've negotiated. So those are the kind of collaborations. And also in terms of training, Mount Kenya is a research and training institution. So you're training students, and also at the end of it all, they are going to do their research projects. So if you call us in, we can have collaborations whereby we come and give a talk. Let's say, for example, during the proposal, defending proposals, before the students or the students now proceed to do their research, you can call us in. We don't charge for that. We can come when you're doing those presentations. We can be able to advise, because in most cases, when being first-time researchers, when you're doing your research, most of your protocol is copy and paste. And in most cases, you're going just to pick an item from there. Let's say, for example, let me take an example in the industry currently, where I didn't speak about sample collection. I didn't speak about PCR, but we have solutions for the whole process. We have so many extraction kits. You had my colleague from Excella saying they have affordable solutions. The solutions that they have affordable, we also have same solutions on these other end. It all depends. Let's say, for example, we have a DNA extraction from QIAGEN. Altona have their DNA extraction supplying for a certain company. In Kaba, we also have the Zymo research that I did explain. So during these presentations and defense of proposals, if you call us, you're not paying us to come and talk about this, but we are trying to tell the researchers how possible it is, and they should not have fixed minds. Since you copy-pasted something from somewhere, you want to use a QIAGEN DNA extraction kit and not a Zymo extraction DNA kit. They are all the same. What you look at is the pricing, that's your budget, and also the time it takes you to do your work. You may have an extraction kit which is taking you almost 30 minutes to do the extraction. You may have an extraction kit that wants you to add other components and you already purchased the kit. And there are kits which have the complete solution. So that's where we come in. Those are the kind of collaborations that we want. We want to engage and assist you so that when your data comes out, it's something which is presentable and can be also be used somewhere else, either in Africa or in Europe as well. I think that's it.
We have Aika.
Maybe you can just summarize the question for Aika.
Well, we actually engaging on the nature of collaborations that you expect from academic institutions.
Sorry, please come up again.
We're looking at the nature of collaborations that you as an industry partner will expect from academic institutions.
Okay. Thank you for this opportunity. My name is Jacqueline Mungai. I'm from ICA. Currently, I'm serving as the role of product management. Your question is a great one because we are a German manufacturer, so we have different facilities all over the world. So we have some that are production facilities, while some are simply sales offices. So for the academic industry, we know that we have people who are in the research field, people who are still learning, and they're trying to come up with different formulations. So at the HQ in Germany, we have a team known as R&D, Research and Development, and they usually rely on the feedback that we salespeople or people on the ground usually give. So you come up with something that is innovative, and then you give us your suggestions, and then they pull up, they gather this information, and then they come up with a product. Such an example is the ICA pen. It is the latest electronic pipette, very slim. This idea came from a certain source that we were waiting for them to give us consent. Yeah. So for academia, that is one way in which ICA can actually support people in the academic industry. So product development. Another way would be exposing yourself to research scientists or the same people in the same field of academia. So we have an applications lab at the HQ in Germany, and we're trying to replicate the same in all facilities at ICA. So we gather people who are from the same industries, and they come and collaborate or they share information. So we gather this knowledge from different kind of people, and then, there's that exchange of information. They get to learn. Yeah, that is what is coming at the top of my head right now.
Thank you very much. I really appreciate that. And being a mentor of postgraduate students, some of whom are seated here, I think they're very excited when they get to listen to some of the opportunities that they have with the work of academia industry partnerships. So we really appreciate all your engagement with us. You can spare the rest of the questions. I've just been notified that one of our industry partners is back online. That is Paul from Project Mocha. And Paul, if you can hear me, we're going to give you just about 10 minutes. We'll appreciate your precision when it comes to the 10 minutes. So please, you can start your presentation now, if you can hear me.
Yes, I can hear you. Thanks once again and good afternoon to everyone. Could you confirm whether you can see my slide on your end?
Yes, I can see your slides.
Okay. So I'll try as much as possible to rush this, given the time constraint. And so I just want to briefly take a walkthrough around Project Mocha, and that's our journey from tokenization of coffee trees to financing of verified production. CTOs been building the platform largely. And so let me get to this. It's one of my favorite slides, which is the farmer problem. And in this case, I take Kenya as a context. And on the left, you get to see a graph that shows two charts, which is one, the decline in the production of Kenyan coffee from our peak in the 1988, that was at 100,000 metric tons, to currently we're doing just between 40 and 50,000 metric tons as of 2024, 2025. In the same 3 decades, we've seen the global coffee consumption double. So it's a paradox that our production is decreasing, yet the production is increasing, the demand is increasing. So for Kenya accounts, coffee production is largely done by smallholder farmers. They account for 70% of the production. That's about 700,000 coffee farmers. And one thing I look at is the two figures on the right, which is the amount of lending that goes towards agriculture as a whole from our banking institutions. That's less than 5% within this 3-decade period. And the amount of time it takes farmers to get paid once they have delivered their coffee to cooperatives or to factories, which is anywhere between 6 to 18 months. And that kind of tells you why we're having this disproportional graph on the left, where the global demand is doubling and increasing, our production is decreasing. And on the flip side, we have the buyers who, from their standpoint, buyers do not have visibility on our production. They can't see our coffee, they can't see how much we're producing, they cannot see their farmers, they don't know anything, they're not able to source with visibility and traceability. We are left to dealing with our coffee auction, which has largely tried, but has, as we know, some biases in terms of how coffee is marketed there. The other issue is buyers face uncertainty in supply, whether this is from the weather, changing climate change, pest disease, poor farm management, which comes from the lack of funds going into the farms. And so we've seen our industry where buyers are carrying the financial risk, where it's the buyers who are sending farmers advances, buyers are committing capital towards training farmers, it's the buyers who are putting in money for cooperatives and factories to be built. And that ideally is not their core domain. Their work is and should be to purchase coffee at the best possible price. And so all that got us to was this production intelligence, where on the left side, we're looking at capturing data and making our farm production, our trees, our farmers visible. That's through satellite imagery, doing farmer registrations, conducting the registration of trees and digitizing these trees into tokens, capturing field data on how production is being done, aggregating all this into farmer profiles with records that can go through an intelligence layer. This is where now the AI comes in terms of being able to analyze and give prediction, or analyze and give insights into how we should manage the crop, or production purchase insights to the buyers, or even insight in terms of how funders can come in. And the result is being able to connect buyers. So the intelligence layer allows buyers to come and buy coffee. They provide traceability from all the way from the farm to the final coffee. We are able to now issue contracts where buyers can commit money early that goes towards assets, coffee trees that are ring-fenced, and they're able to acquire the crop from these coffee trees. It's allowing banks and lenders to now come in and start financing the production. Of course, it provides data towards sustainability, and key of it is we are able to expose the farmer to the final purchase price, where they are able to see exactly how much their coffee was sold for, and this money trickles down all the way back to them. So from a financial perspective, the opportunity is in the trillions of dollars in terms of now scaling this intelligence, where we are able to show all our assets. We are able to show our coffee trees. And this just coffee was, we started in coffee because this was my grandfather, as I said, I'm a third generation farmer. So I was born in a coffee farm. And so automatically I was naturally drawn to coffee. But the whole aspect of providing production intelligence cuts across the entire farm level, whether it's coffee, avocados, macadamia, or even indigenous trees for carbon credits. And so the whole aspect is, what we're trying to say is, by providing this intelligence, we're able to put together and mobilize capital now from the global capital markets to come into the farms. Why now? It's the world is increasingly demanding verified supply. The buyers are conscious. They want to know where is their coffee coming from. We have EUDR laws where they require that your coffee should not come from deforested land. We're also looking at changes within the capital landscape. We've increasingly seen the world adopt blockchain and more so stable coins, especially this year. The US has been spearheading regulation that has now allowed stable coins to become currency, and they're being used. Here in Kenya, we recently passed regulations again on virtual assets. We have an act in parliament, and the regulations were recently released by our regulators. So we've seen a shift in the capital landscape where now money is digital and operating on the blockchain. So the question is, if money is on the blockchain, where will this money move towards? Do we have assets on the blockchain so that they can have this productivity? And finally, of course, is the increase in the demand for coffee. Like we've seen more and more, the coffee culture is heavily taking root across the world. And so production data is becoming a financial asset, and we are positioning ourselves to be able to spearhead this for our country and for Africa across the board. Our novel innovation is around production backnodes, and to simplify this is it's using the different players and the data they provide, which is the farmers and their trees, the buyers and their demand for coffee, the investors and their need for yield, and bringing these players together via smart contracts where farmers commit trees and the production from their trees to access capital, and the buyers commit to buy the coffee that is produced, and from the money that the buyers pay goes to ensure that the investors are paid and the farmers get their money, and we are able to create this circle of production and jumpstart the flywheel when it comes to improving the productivity of our farms. And demand here closes the financing loop. We've realized largely that financiers want to know that someone is going to foot the bill. Like someone wants to buy the commodity and they're going to put money to buy the commodity for them to be able to step in and provide the working capital, which is a huge gap here in Africa. And so part of as part of this intelligence layer, we have also a marketplace where now farmers are able to list their coffees, and we are able to expose them to global coffee buyers and commodity traders. So far, we've focused on doing also local coffee production and coffee value addition. all the way to the point of selling the coffee cups. Like we roast our coffee, we mill our coffee, we roast it, we brew it and serve it at events. And this has, in the last like 1 year, we've been able to put together about 40 events and served 15,000 cups of coffee and generated some revenue in that. So the whole point is to demonstrate that with intelligence and especially the utilization of blockchain to verify and provide this provenance of the assets and the assets that we're front facing, that people consciously will want to buy. Let me touch a bit about on the global settlement, which is stable coins. And stable coins, I'm sure some of us have heard about them, but more and more we are going to get to hear about stable coins. Right now, we're predominantly dealing with dollarized stable coins, USDT, USDC, though we're looking at also even with our virtual asset service providers bill, having our own local currency stable coins. And in essence, this is local currency that is wired through the blockchain. Here in Kenya, we are really already attuned to this by our experience of using M-Pesa. We're already used to handling digital money. So the transition to stable coins will be, we will actually not realize that we are using a stable coin since we're already used to using digital currency. I got, there was a question around collaboration, and for us, it's mostly, we need to, especially from the research institutions who have the funding and the budget to extend the tools like ours, like now you are using drones, tokenizing trees, getting farm level data to collaborate towards using local service providers. So for example, we've gotten into this Darwin initiative with Oxford University and Strathmore to do mapping of 500 farms in nearly over 100 hectares. And we really appreciate that as opposed to getting service providers from outside the country, they were keen enough, they were open to working with us to bring this data to visibility. And this not only benefits us as a company, but also we are able to create that competence and the skill, and our data gets to be housed locally. And we are also able now to have access and be able to build around the data that we're going to collect. So opportunities where research is required, data is required, and you need expertise to capture this data, we will appreciate being your partners around this. And to sum up was why Project Mocha, it's around my grandfather. He planted his first coffee tree in the 40s. And to date, we are harvesting coffee from his trees that he planted. So we have the trees, farmers exist. When we are producing, we have global markets that are keen on picking up our demand. What's really missing in the middle is a verifiable and trusted layer where we don't have a number of middlemen coming in, but we have farmers directly being able to front face their production towards buyers, and buyers able to buy coffee. And this creating a commodity trade that financiers feel comfortable enough to come and put in their capital towards. Thank you. I feel like I've really read that.
Thank you so much, Paul. I'm very glad that you spoke with precision just within the 10 minutes that was allocated to you, and we don't take that for granted. I'm also happy that you've already taken care of my question on the nature of collaborations that Project Mocha will be looking for, and I'm not going to ask you that again. I think I can open the floor for all of us to take the opportunity of engaging with all the 4 industry partners. If anyone has a question, please, this is the moment for you to ask.
Good evening to all again. I'm Professor Sifuna Sudi. Others call us mimi ndiyo Sifuna. But that aside, my question is in direct connection with what Dr. Sarah has asked about the collaboration between academia and the industry partners that have joined us today. So when I heard of the collaboration, I understand that collaboration between academia and the industry is something like circular or a circle whereby the academia uses funds from the industry to generate knowledge. And then the industry uses knowledge, or rather converts that knowledge into products. And then the cycle continues. So the academia generates knowledge, and then consumer of this knowledge is the industry. And the industry uses that knowledge to produce something, then sell, therefore convert the knowledge into a product. And once they sell, they get money, and then this money is now used to buy the product from academia. That is now they are consuming the products of academia. But from the presentation that I've had today, I've realized that none of the collaborators talked of how they are consuming the knowledge of the research institutions or the academia in Africa. For instance, the products that they have presented to us, that's how they could now be consuming our knowledge, have all come from abroad, or the UK, no, not UK, but Europe, for instance, Germany, they have mentioned them here. So I was wondering, are they having any intentions of consuming the knowledge that is being generated by the African research institutes and academia to come up with products in a way that they can now be consuming the knowledge that is being generated by the African researchers and African research institutions, so that it's not just about the African Research Institute consuming their products and services in terms of product delivery, supplying products to the research institutions in Africa, that like what they have basically focused on, providing these research tools to you. But these research tools have been produced based on the knowledge generated by the outsiders from Africa. So I was wondering, do we have any mechanism or plans for today or in the future where you are going now to be consuming the knowledge, which is the product of the African researchers, to come up with something that now you can also be consuming from them, not just them consuming from you? Thank you.
I see we have another question online. Maybe we can take that and then they can be handled together. Marcy?
Oh, thank you very much. My name is Mercy, and as I previously mentioned, I'm a PhD student. My question goes to Paul Gashora. It's very interesting work you are doing, making our African data go all the way to the financial markets. But I'm wondering, how long do you think it will take to have actual financial impact using the digital coins? And yeah, I would also like to mention that, as previously somebody mentioned, that the connection between our data, I'm sorry, I'm losing my thought. The connection between the data and the market, as previously mentioned by the first Professor Chinguna, about I lost my thought. It's been a long day.
Was it about data sovereignty? That's what
Data.
Professor.
Yes, data sovereignty and providing a platform. Providing a platform, kind of a platform where African data can be stored, and that can be translated into the digital coin and monetization of data or something like this. It's a very complex topic with digital coins and all. So I would just like to get more information from Paul, what he thinks, how we can use our biotechnology information using his data. Thank you.
Paul, maybe you go first. You're muted. Are you
Okay, yeah. Thank you, Mercy, for the question. And let me start by saying, from a financial perspective, without looking at the digital coins, data is an asset. We saw China last year did papers around improving how we account for data as an asset. And right now, you can have your data as an asset on a balance sheet and assign a value to it. So let me just start there. Like, data is now considered a financial asset. So when it now comes to the financial markets and how financial markets work, it's about your balance sheet. There's this formula: assets is equals to capital plus liabilities. So here in Africa, the reason we are not able to access capital, the reason we are not able to access debt is because we do not have the assets recognized. So we have a very thin balance sheet compared to the kind of balance sheets that the developed markets have. We can't compare our balance sheet with that of the US. And that's why the US is able to attract capital, because they have a balance sheet. Likewise now with China. China is now the number 2 country in terms of their ability to monetize themselves. And so looking at what these other countries have done and what the advanced financial markets have done, we need to take a step back and see what kind of assets do we have that can fit into a balance sheet. And it's not the advanced derivatives that money markets have. It's not the stocks, because our companies are not yet there. You can't compare our top tech company here in Safaricom with the top company in the US. So the assets that we have that we can quickly put into the balance sheet that will change the narrative is our natural assets. It's the land that we have. It's our trees that have been producing. It's our minerals and all the mines that we have that honestly are plundered and taken away. Our oil, our gems, our gold, diamonds in Congo. Like, think about it. If you could have this visible in a balance sheet, then the equation would be, we have assets, and now would be able to attract capital, would be able to attract the liabilities. So what I'm happy about is the issue of digital currency is we're no longer talking about in the future we are going to. It's already happening. We've seen the adoption of digital currencies. Right now, cryptocurrencies are worth over $2 trillion. And right now we are at a bad market. There was a time it was close to $4 trillion. And we're seeing increase in more so stable coins, which is our fiat currencies put on a digital ledger. So the dollar, the yen, the Chinese yuan, and the Kenya shilling, having a digital repository, a digital ledger where we can transact them.
Paul, sitting on the edge of time, kindly will appreciate if you could summarize in the next
To
few
sum it up
seconds.
is the M-Pesa story. We're already using digital money. Like, if you look at how M-Pesa is structured, money does not leave your pocket to my pocket. It's a message that is handled on the Safaricom ledger, and all the money sits in banks. When I send you an M-Pesa message, it's just a text, but we've already learned how to use this as value. So we've already been using digital currency. So the question is, how do we adopt this technology to work for us, considering that we have significant structural gaps? We do not have the kind of capital markets, the kind of infrastructure the developed markets have. So how do we make it work for us in our own local context?
Thank you very much. I will reach out and talk more about the. Thank you.
Thank you.
Thank you so much, Mercy, for engaging with us, and we really appreciate Paul for taking care of that question. My friend from Enkabat, do you wish to respond to what Sifuna said?
Yeah. Before I forget, Prof, where are you? I was just good. Collaboration. It's a wide thing that we can do, but in this aspect, I was speaking about collaboration. Me and my friends, we are speaking about collaboration from the distributor side. So now there's collaboration from the manufacturer side, of which we've done it before, whereby they produce the kits, they give us the kits to give you the kits for free you try them out and give us results. The results that you give us, they use those results. Let's say, for example, it was Kenya or in Africa, samples were done in Africa. They use that to better the kit. And also just to finalize, also speaking about collaboration, I don't think there is any need for a university to buy a machine worth 500,000 US dollars. That's not a good investment. I'll tell you for free. From the industry, as much as I want to sell it, I'll tell you for free. University, it's not a good business. Research institutions like Emory, Erie, that's okay because they have the funds and they have the researchers working there on a daily basis. So where do we come in collaborations? If a university wants to buy an equipment, let's say, for example, the Vega system or the Revio system, which costs around 500,000 US dollars, we partner. We meet halfway, meet cost of the price, I meet cost of the price. And then we agree on where the equipment will be so that your students can access the equipment, and I can also access the equipment offering services. For the expensive equipments, it's easier to request for a service than to buy an instrument which in 2 years' time it will be obsolete. You know, one thing with manufacturers, sorry to telling you this, I hope they're not they're not listening to this. One thing with manufacturers, you buy an equipment today, 2 years down the line, they tell you they no longer support that technology. You'll be required to buy another machine, or maybe they'll tell you, like, you know what, the spare parts are no longer there. So if you can't have a spare part for the machine, what's the use of that machine? So when you're buying expensive machines, look at the after-sale service. If I cannot give you the after-sale service, please don't buy that machine. You will have a nice machine that you'll never utilize. That was my parting shot.
Thank you very much to all our industry partners. We really appreciate the time you've taken to engage with us. And I'm pretty sure all of us have been taking notes on just how to explore collaborations and, of course, support our research and, of course, foster technology access. Let's please give a good round of applause to all our partners in the industry. We appreciate all of you for taking your time, and we're looking forward to fostering the type of collaborations that you expect from us. I wish to hand over to my senior, Professor Anne.
A round of applause for Dr. for spearheading that a very, very, very engaging session. Partners, friends, colleagues, those of us who are online, we've been here since 8 o'clock this morning. That's 9+ hours. Can we give ourselves a round of applause? I think you've shown passion for science, but more importantly, the conversations were so, involving and enticing that you've not even wanted to leave the room. So we thank you so much. We promise an even more exciting day tomorrow, because we're talking about new and very, very, those conversations that the VC was telling us in the morning, the difficult conversations that have to be had among scientists, among ourselves, so that we can be able to reach those high-level boardrooms where decisions about us are being made in exclusion of where we are. So I'd like to tell you a little bit about tomorrow. Tomorrow, please be here at 8 o'clock. Those of us online, you can log in. We'll be open from 0800 East African time. The sessions will start promptly at 08:30, and we will proceed with the first session, which is discussing access and benefit sharing, DSI, and biodiversity governance in Africa. So thank you very much. Let's give ourselves a round of applause. For those of you who are online, we wish you a good evening, and we'll see you tomorrow. For those who are present, I invite you to have a cup of tea. It's ready and waiting, and have a very good evening. Thank you very much.